would like to be mentioned in the chat, we can do that for you, by the way.
Yeah, sure.
I will introduce you right before your talk, and then you will start. We can also start maybe checking the screen share. So we can start with Varun. Can you share your presentation so that we will check whether everything is working fine?
Yeah, just give me a sec.
Check whether everything is working. Okay, I think we can start now. So hello, everyone. Welcome to our session. My name is Ezgi. I'm a postdoctoral researcher at the University of Gießen, and I will be hosting the session. So in today's session, we have a great lineup of speakers, and we will talk about Alzheimer's disease, particularly the molecular mechanisms and state transitions that help us understand AD. So this is an interactive session, which means that you can come on stage if you want to ask your questions. If you want to come on stage, please let us know when you post your question. Otherwise, I will ask the questions for you. You can also use the chat to introduce yourself and tell about what you're doing as well. So we have, very exciting talks. We can get started. Our first speaker is Varun Ulanath. Varun is an undergraduate student at the RV College of Engineering in India. And Varun's talk is about the transcriptomic analysis of the brains of symptomatic and asymptomatic Alzheimer's patients. So the stage is yours, Varun.
Yeah, thank you. Can you see my screen? Okay, cool. Yeah, so good evening, everyone, or good morning, or good night, or good afternoon, wherever you are. So my name is Varun, and I'm an undergrad student in RV College of Engineering, India. My poster, as you can see, is titled Transcriptomic Analysis of the Entorhinal Regions of Symptomatic and Asymptomatic Alzheimer's Patients. So for those of you who aren't familiar, asymptomatic AD is a condition where patients have intact cognition. It is a condition where patients have intact cognition, but neuropathology similar to AD. Asymptomatic AD patients are highly likely to develop AD as well. So we can think of it as an intermediate disease state between the normal condition and AD. The intuition behind this project is that understanding the molecular mechanisms that govern asymptomatic AD could help us in early detection and treatment of a patient before AD symptoms develop. Yeah, so this is the basic workflow. We first took gene expression count data and again, some basic pre-processing such as removing batch effects and removing the bad quality probes were done. The data is then split into 3 study conditions: AD versus asymptomatic AD, as well as control, and asymptomatic AD versus control. Next, we performed a differential gene expression analysis using statistical tools and identified genes with a p-value of less than 0.05. We simultaneously performed co-expression studies. Then overrepresentation analysis and gene set enrichment analysis were done to identify the significant pathways and gene sets. Finally, protein interaction networks were constructed with the co-expressed genes. So moving on to the results. The first slide is for AD versus asymptomatic AD. We observed that in the volcano plots of the two conditions, of the two cases, most of the genes are overexpressed in the disease condition. Next, again, this is a table of the top 5 differentially expressed genes. In the protein interaction network, so each module actually represents genes which are similar, which have similar expression profiles or similar co-expression. We can see proteins of genes such as SNAP25 and FOS highly pronounced as hubs in these networks, and these are the classical genes related to AD. So moving on to the AD versus control case. So this is again very similar to the first case. Again, most genes are overexpressed, as you can see in the volcano plots. So finally, the last slide is for the asymptomatic AD versus control case, the one we're actually interested in. So again, most genes are overexpressed as compared to healthy individuals, but not as much as the AD conditions. We only obtained about 40 differentially expressed genes here, and again, the top 5 again. Interestingly, we obtained 5 different modules for the asymptomatic AD case, 5 different protein interaction networks, suggesting that multiple gene sets are involved. In module 4, you'll notice APOE4, gene synonymous with AD as hub. So studying the other genes involved would explain, or to an extent, explain how APOE4 works. APOE4 works. Next, so these are basically the Venn diagrams of the number of differentially expressed genes in each study condition. An interesting observation is that there is no single unique gene involved in asymptomatic AD. Also, about 5,000 genes are common in the AD versus asymptomatic AD and the AD versus control case. The Venn diagram on the right is of the significant pathways we saw during our analysis. We observed that about 17 pathways are unique to the asymptomatic AD versus control condition. So finally, the table to the left shows the classical perturb pathways known through literature and, of course, observed in our study. Again, the table to the right shows the significant pathways we observed in the asymptomatic AD versus control condition. Again, we saw several interesting pathways. So innate immunity pathways and neutrophil degranulation were shown to be affected, which indicates that there is an immunological aspect to asymptomatic AD onset as well, similar to AD. Coming to the conclusions, so again, it's consistent with the previous findings on genes and pathways involved in the onset of AD. Again, so the genes such as APOE4, FOS, SNAP25, and STC4 which have been shown to have altered expression levels in AD patients are highly pronounced in the current study results as well. With some further analysis, we actually identified 3 features or 3 salient features we observed in most of the protein interaction networks and the significant pathways. So first, we observe SIT1 as an interaction hub in the protein interaction networks of all cases. So further analysis showed that SIT1 is involved in a pathway that leads to AB top leg generation, which is an important biomarker of AD. Secondly, we observe SDC4 as a co-expression hub in all the cases as well. So by doing a similar analysis, we discovered that this gene is also involved in AB top leg generation. Finally, we observe CYBB as the most differentially expressed gene in symptomatic AD condition. So this gene is known to be involved in multiple sclerosis, but the function in AD is still being researched. Again, so a similar analysis was done for the temporal, frontal, and the cerebral regions of the brain as well. But we couldn't actually include it in the final presentation because of constraints of time and space. Yeah, so thank you.
Thank you very much. That was a great talk. Thanks a lot. So are there any questions? You can share your questions on the Q&A section. Also post your questions on the chat part as well. So for now, I'm not seeing any questions. Oh, someone has raised hand, I think.
Hello.
Hello.
Hi, nice talk. Thanks a lot. I have one question for the gene that was most differentially expressed in the asymptomatic AD. So could you talk a bit about the function of that gene, or speculate on the role, maybe for the outbreak of AD?
So CYBB is, I think that was the one which I mentioned,
Yes.
is actually, it provides instructions for making a protein called cytochrome B245 beta chain. So it's actually it's one subunit of a group of proteins that form an enzyme complex called NAD/PH oxidase, which again plays an essential role in the immune system. So again, we know that AD has an immunological aspect to it because of the degradation in the extracellular matrix and such. So is possibly the reason why it's differentially expressed.
Cool, thanks. That's very interesting. Yeah.
I actually have another question about cytochrome B. So I think I remember hearing about it also in the context of mitochondria, right? So it's like a mitochondrial protein, and in AD obviously you have like mitochondrial dysregulation. Could that be might that be what's dysregulated or somehow linked?
Yeah, I mean, it could be, but I think the primary reason is this, but I'm not really sure. I'm primarily a bioinformatician, so I don't want to talk about, exactly the molecular mechanisms in AD. So this is what we found in our analysis, and I'm just sharing what we found. Yeah. So again, you can do further studies to check.
Yeah.
Thank you for the questions. We have another question, and I guess I need to ask that. So this is from Eva Limberopoulos. I hope I pronounced the name correctly. How do you interpret the significance of finding perturbations in innate immunity in asymptomatic AD?
Yeah, so like I mentioned before, immunity is, it's well studied. So in Alzheimer's disease, in the actual Alzheimer's disease, not the asymptomatic one, again, so immunity is a very important component. We see a lot of genes and a lot of pathways involved in immunity to be perturbed. Again, so if I can show you the slides, actually I didn't mention the immunity part. In the AD table, the last table, which was the slide, it was mostly elastic fiber deformation, such things like that. But we did find a lot of pathways perturbed in the immune system as well, relating to AD, and it's been well studied. So again, it seems plausible that it is obviously affected in asymptomatic AD as well, because it's one of the reasons why the AD progression actually takes place. So it's like the immune cells start destroying the ECM and the ECM of the nerves. And yeah, so I think that's what she meant. I'm not really sure. The question again, I don't want to dive deeper into or speculate about why it's actually integral in AD or asymptomatic AD progression.
Okay, thank you. Thanks a lot. So yeah, I guess we have no other questions for this talk.
Okay.
Yeah, so I would like to thank you again for the wonderful talk, and also thanks for the excellent questions. Was very informative. So we can move on to our next speaker. By the way, feel free to use the chat if you have any other questions or follow-up questions. Okay, so the next talk is from Arjumand Farooqi. And Arjumand is going to talk about transitorin and Alzheimer's disease, a systematic review.
Hello, everyone. Am I audible?
Yes.
Hello?
Yes.
Yeah. So I'm going to talk about the role of transthyretin in Alzheimer's disease. We did a systematic review And so prealbumin or transthyretin is a protein that is found in various tissues of the body, including the brain. And studies have found a conflicting role of transthyretin in Alzheimer's disease. But no systematic review was yet done to produce high quality evidence. So our aim was to clarify the role of transthyretin in Alzheimer's disease. So the method that we used was the PRISMA checklist, and we predefined the keywords, and then on 1st of July 2020 we entered them on PubMed and got the result with 346 researchers. And after removing the duplicates one, and after title and abstract screening using our inclusion and exclusion criteria, and then full text screening, we got in finally 95 studies that were included in our qualitative analysis. So the findings after data extraction were categorized under the following headings. There were 11 main headings, and the results that did not fit in any of them were included in miscellaneous. So the function of transthyretin in the brain, we analyzed on the basis of the site of the production of transthyretin, the interaction between transthyretin and A-beta amyloid, the fluctuation in the levels of serum transthyretin, the changes in the level of transthyretin in the CSF, the action or the role of transthyretin stabilizers in the Alzheimer's disease, the Alzheimer's disease transthyretin and the sex hormones, the effect of transthyretin configurations on Alzheimer's disease, the action or the role of apolipoprotein, metallothinins, and thyroid hormones in Alzheimer's disease and their interaction with transthyretin. So the results for us follows that the function, most of the studies that we included agreed that the function of transthyretin in the brain was neuroprotective, even though there were 1 or 2 studies that did not agree. Most of the studies agreed that the transthyretin was produced in the choroid plexus, but the more recent studies also showed that it was produced in neurons, and there was also diffusion of serum transthyretin into the CSF. The transthyretin was found to clear A-beta peptide molecules and inhibit A-beta aggregation. So this was its actual role in Alzheimer's disease. Its physiological role. So, and the studies on serum found that there was an inverse correlation between the progression of Alzheimer's disease and the transthyretin levels in the serum. So, the serum transthyretin levels were found to be lowered in the patients with Alzheimer's disease. And there were also the CSF transthyretin levels were also significantly decreased in patients with Alzheimer's disease. Transthyretin was found to influence A-beta levels in a gender-specific manner. Its action was influenced by testosterone and estradiol. So here we found that both testosterone and estradiol, they were protected against Alzheimer's disease. In CSF of patients with Alzheimer's disease, we also found that there was the transthyretin that was already there, was most of it was in oxidized form, as compared to the people who did not have Alzheimer's disease. Here I would like to again add that this was not a quantitative synthesis, it was a qualitative analysis, and we only used PubMed database and not the other ones. These were 3 of the studies that I've included the reference of, which were included in the 95 studies that we had. Thank you.
Thank you very much. That was a really informative talk. So we have plenty of time for questions or comments as well. Okay, so for now, I don't see any questions. Oh, yeah, please.
Yeah, actually, I'd have a question. So amongst the studies that you looked at, have there been any upregulation studies in animals to see whether the neuroprotective function can somehow be harnessed? Because it would be interesting to see whether it's more correlational and there's a different mechanism at play, or whether it's a causal relationship, right? The neuroprotective effect and the levels of the protein? Maybe you said it and I missed it. In that case, I'm sorry.
Yeah, so there were studies that were done, but they were not the recent one. Recent ones that have been done in relation to transthyretin and Alzheimer's disease were mainly concerned with their production. So that, as I mentioned, the production is now recently been found to be in by neurons as well. Even though many studies do not agree with this, there had been studies who counterfeited this mechanism of production. But yeah, so the whole point of doing this research was because there were so conflicting ideas. So that was what inclined us to do this research. Thank you.
Sorry, actually, could I ask another question?
Yeah, sure.
So in your opinion, in order to kind of like further clarify the role, so you've done like the survey of the literature, what would be kind of like in your opinion, like a next step, kind of like either experimentally or?
Yeah, so few of the things that I found that were very, very interesting were the role of transthyretin stabilizers. So these were very scattered literature that was present on this, but it was quite informative. And I think there is a potential role of transthyretin stabilizers in the Alzheimer's disease, curing the Alzheimer's disease. So yeah.
So you mean stabilizers to, for example, maintain like the level so that it's degraded less, like, or?
Yeah, so there were even the degradation is there, but also there is the configuration, as I mentioned, that there was more oxidized transthyretin found in patients with Alzheimer's disease as compared to patients that did not have Alzheimer's disease. So yeah, so there have been researches done, there have been experimental procedures conducted on mice, but there were very few. So they were interesting, quite interesting.
Okay, nice. Thank you.
Thank you.
Yeah, thanks for the very nice questions as well. Are there any other questions for our speaker? So I am not an expert in this field. Actually, my field is really very different, so that might be a very naive question. You mentioned that there might be some sex differences or sex hormones.
Yeah.
So can you maybe elaborate a bit on that?
Yeah. So the sex hormones played a significant role in Alzheimer's disease. That's what we found from the literature, and was observed that reduced level of brain testosterone and estradiol in female mice with genetic reduction of transthyretin might underlie their increased Alzheimer's disease neuropathology. This is what they indicated. But then again, there were very few researches done. As far as I can remember, there were only 2 researches done on this topic, but I included it because I thought this was a good finding, or maybe it could promote some future innovations.
Okay. So, by the way, what was the time period of these studies? So you mentioned you started from 2020, and then like how far did you go back?
Yeah, so the inclusion criteria and the exclusion criteria was said that we had very few exclusion criteria. There was only one exclusion criteria, and it was that if the research was not done in English, it was printed in any other language. Like we had a lot of French and German researchers and Japanese as well. So that was the only exclusion criteria. Apart from that, it was even if it was done in like 1990s, we did include it because there was not a lot of researches done. As you can see, we got 346 results, but then after exclusion on the basis of the criteria, we had only 95 studies. So that is why we do not have a lot of exclusion criteria.
Okay, thank you. We have another question from Eden. Did you organize all the discussions together and compare them with the findings? Did you find interesting new questions from it, or some, I guess, implications? So I guess for future work, this is, yeah, from Eden.
So from what I'm understanding, the question is, did I compare my findings with discussion of what? I'm not able to get it.
So maybe can you clarify that, Eden, a bit? And in the meantime, you can also see it from the chat, because I also don't.
Yeah, I would like to
exactly
see
understand.
what the
Yeah.
question was.
So it's in the Q&A section. Yeah.
Just
Yeah.
Yeah, so yes, I did at the end, when I compiled the whole thing in a form of report, I did, because I got a chance through the Indian Academy of Science to do this whole research. So when I was compiling the report, I did compile it in one, and then I did compare the results. And yes, as I said, there were the whole point that I chose this topic after the primary literature review, because I saw that there were so many conflicting ideas. As I said, even though a lot of researchers did unequivocally accept that transthyretin had a neuroprotective role, there were two researchers that did not accept it. And there were recent researches that were done to say that it was the production of transthyretin was in the choroid plexus. A lot of researchers, if you search it yourself, they all agree. But then there are three researchers who say that they did not agree, and they say that their method of isolation was much superior. So according to them, their research was correct. So yes, there have been conflicting ideas, but and I have, as I mentioned, the role of one that I found most interesting was the role of transthyretin stabilizers, along with the role of apolipoprotein that was also there. That could help, if the research is done on it, then it could really help in the solving of this case, that is Alzheimer's disease. If was it okay? Was it satisfying, the answer?
I hope so. And if it's not, I think. Yeah. So she said, "Great."
Thank you.
I guess it was a satisfying answer. Yeah. So I don't see any other questions. So we can, I guess, move on to our next speaker. Okay. So this will be the last talk in this session. And the talk is from Dominique Kleppel. Dominic is a first-year PhD candidate at the Center for Data Science in Coventry University. He's working with Fei He, and his talk is titled Energy Landscape Analysis, or Analysis of EEG of Alzheimer's Patients Reveals Alterations of State Transition Dynamics. So stage is yours.
Yes, can you see the screen?
Yes, yeah.
Great. Yes, thank you for your introduction. I'm really excited to talk about this project and the results that we got. As you said, we performed the energy landscape analysis of EEG of Alzheimer's patients. And well, the energy landscape method or analysis essentially maps the brain states so that they have each a particular energy. And the lower the energy of the state, the higher is the probability that the state will occur. And this method had been applied to fMRI and MEG data in the past, but to the best of our knowledge, this is actually the first attempt to use this for EEG data. So we have a data set from 20 Alzheimer's disease patients and 20 healthy age-matched participants. And from each of these people, we have 8 10 seconds long data samples. 4 of these samples are at rest with eyes closed, and the other 4 are at rest with eyes open. And we collected these data using 22 bipolar EEG channels. So I would now just quickly summarize the method itself. So we start by selecting just 10 electrodes out of these 22. And we take the continuous signal from each electrode and binarize it so that it becomes either 1 or 0. And you can think of this as basically the brain region that's generating this signal as being either active or not active. Once we have these binary data, this is basically how they look like. So then each row becomes basically this binary state that the brain is currently in. And using these binary data, we estimate the energy using the pairwise maximum entropy model. And this model estimates the energy mainly by looking at the pairwise functional connectivity between the electrodes. And once we have the energy of all these states, we can actually construct the whole energy landscape and analyze the general shape or features of this landscape. Of course, there are many things that we could analyze about these landscapes, but we chose just 3. First, this is the number of local minima. So that's basically the number of like volleys in this landscape. Then there is the standard deviation of basin sizes, which is basically the variation in the sizes of these volleys in the landscape. And finally, we looked at how much time the system or the brain spends in the global minimum, so the deepest volley in the energy landscape. And the results that we got for local minima, we found that Alzheimer's patients have consistently more local minima. And you can think of this as basically the Alzheimer's patients oscillating between more stable states compared to the healthy participants. Then we also found that the standard deviation of basin sizes is consistently larger across both conditions and multiple frequency bands of the data. So it is larger for the healthy participants. And finally, we found, again, consistently across both conditions and multiple frequency bands, that the healthy participants spend actually more time in this global minimum. So again, we can think of this as basically the signals of healthy participants as being more stable or remaining within one region of the energy landscape. And the actual interpretation of these results is still sort of future work. But so far what this indicates is actually somewhat a disagreement with the literature. As previously, it was reported that Alzheimer's patients show reduced complexity in their EEG signals, but the results from the energy landscape actually indicate an increase in complexity. And then what we would like to do in the future as well is look at whether we can actually use the features of the energy landscapes for prediction, so for diagnosis of Alzheimer's. And finally, there are some limitations to this approach. First, that at the moment we can construct and analyze the energy landscape only with a relatively small subset of channels or electrodes. And this is due to the computational resources, since it becomes simply too expensive to actually compute the energy landscape with more channels. Then a second issue is that, or challenge is that we simplify the data to just binary activations, and we risk losing some finer details in the continuous signals. And finally, we assume that the functional connectivity remains stable across the whole 10 seconds long recording, which might not be true, but at the moment, yeah, it's just a reasonable simplification that we made. Yes, thank you.
Thank you very much, Dominic. That was a really nice and interesting talk, interesting technique as well. So if you have any questions, you can ask, either post them on the Q&A You can come up to the stage to ask it yourself. Hey.
Hey, actually, me again.
Go ahead. Yeah.
Thank you for the talk. It's super interesting. I was wondering, because it's such a huge problem in psychiatry or computational psychiatry to have like a differential diagnosis and actually kind of disentangle different diseases, whether there's any studies or research showing that there's these similar kind of changes in brain connectivity or energy, like in the way that you said you calculated it for other psychiatric conditions, and to what extent we could rely on this being something that's quite specific to, I mean, sorry, not psychiatry, neurodegenerative, oh God, like, sorry for a moment. I was wondering, to what extent can we like say that this is, for example, something that's specific to Alzheimer's, or is it more probable that it is a general like feature of neurodegeneration? What do you think? Because that would obviously be important for prediction, right?
Yeah, of course. So at the moment, research is actually quite limited in this area. In the past, the energy landscape, it has been used to like analyze the dynamics or state transitions of healthy brains. And actually, the only study that I know of that did something similar to what we did with the Alzheimer's was a study looking at the differences of epileptic patients. So yeah, at the moment we can't really say if this is just a feature of neurodegenerative diseases in general, or if it's something Alzheimer's specific. Yeah.
I see. But it would, what also would probably be interesting, right, is to look at the timing of like when these changes kind of start happening compared to when there's like actual like onset of symptoms. I guess it's hard, right, because you need to get the recordings from patients before they're gonna get it. But yeah, it would be super interesting if we could use it for prediction.
Yeah, definitely, I agree. But yeah, at the moment we don't have a data like this, so yeah.
Yeah, but cool that you're doing this. I mean, I've never heard of like the method before, and that sounds quite exciting.
Yeah, so originally it actually comes from statistical physics, so that's why we maybe didn't hear about that. Yeah, because originally it was actually developed for the study of magnets, I think. Yeah, so yeah, so it's actually an adaptation of this method to neuroscience.
Yeah, nice. There's a lot of good stuff that you can repurpose from there, so that's
yeah.
Yeah, I have a question about the selection criteria. So you said that you select from a number of electrodes. What was the criterion for you to select the specific electrode?
Yes. So the previous studies they solved this by simply looking at regions of interest. So they manually selected the regions that should be affected by the phenomena that they are studying. We use a different approach, which is completely data-driven, so automatic, and we select the channels which are which actually have the highest entropy, because we want to look at the regions where the most movement or activity is happening. So we select this by using the entropy, and another like combining with a like predictive approach, where we look at which channels actually provide the best predictive accuracy for the Alzheimer's disease, and then we simply select the overlap between these two methods.
Okay, cool. And I mean, you haven't used any task, right? It's eyes open, eyes closed.
Yeah, it's just resting.
Yeah, resting in your condition. So would it be possible to use with any other cognitive task and to like get more information on what the state transitions would be? Would it be informative?
Yeah, I think it could be definitely done. Yeah, because again, it'll be entirely possible to construct the energy landscape while doing some task. So yeah.
Okay, but what would be then the would we gain more information about the disease?
Yeah, that's really difficult to say at the moment. Yeah, honestly, I don't know.
Yeah, no, understandable. Yeah, it's already very, I guess, complicated technique.
one more question about
Yeah, sure.
how do you infer like the functional connectivity exactly from the like the EEG data? So is it kind of based on like probabilities, like that, for example, that one brain area becomes active after the other? Is it about temporal sequences or?
Yeah, so it's basically almost the same as a simple correlation. So
Okay.
simply if one brain region becomes active at the same time as the other one.
Okay, yeah, makes sense.
Hey Dominic, great talk. So I just had a question. You mentioned stability was higher in the Alzheimer's case. So what does stability actually mean in this context, and does it actually have biological significance?
Actually, it's the other way around. So the healthy were more stable, and what this means is simply that the signals are oscillating or moving around the energy landscape more for the Alzheimer's patients than for the healthy ones. And at the moment, it is difficult to essentially interpret whether there is some pathological explanation for this. Yeah, so simply with the data that we have, we can't say. Yeah.
Okay. Thank you.
Thank you very much for the questions. Maybe let me check the question part. If also anyone wants to come to the stage to ask their questions, they are definitely welcome. Okay, for now, yeah, for now we don't have any questions. So maybe just like a follow-up question. What would be the next step for you?
So the next step is looking at the predictive power of these energy landscape based features, and then also looking at or investigating whether we could actually use the continuous data instead of just these binary simplification. And finally, we would like to look at whether this assumption of the stable functional connectivity is true or not. If not, then if it would be more reasonable to construct multiple energy landscapes from the 10 seconds long recording. So instead of just one, looking at 2, 3, I don't know. Yeah.
Mm-hmm. Yeah. So this stable functional connectivity sounds a bit like resting state. Is it like that?
Yes.
Yeah.
Yes, but
okay.
There are actually studies that found that that even at rest, the functional stability connectivity is actually moving around or changing, going through like a state of connectivity to another. Yeah. So.
Okay. Yeah, I mean, it's just that because I'm in the field of fMRI more, it kind of sounds to me like a resting state. Okay, cool. Okay, so if there are no more questions, yeah, we can thank again for the wonderful talks to our speakers. Was really informative. I also want to thank the attendees for their engagement and also the questions. I would also like to thank our backend, Ariana Dorschel, who's been doing all the technical work, and she had really wonderful questions also to the speakers. Yeah, so this would be the end of our session. And I wish you a nice day or nice evening, depending on where you are. And thanks again, our speakers, for the wonderful talks. And bye, everyone.
Thanks so much, everyone. Bye.
Thanks.
Bye.
Bye.
Bye-bye.
Thank you.
Where are you speaking from?
I'm speaking from Iran, Rasht. North of Iran.
Nice.
I'm speaking from London.
Yeah. Good for you.
Well, it's very rainy at the moment.
Very rainy. And London has same weather like Rash. Rash weather is some sort of like London. Yeah, it's always cloudy and raining. Okay.
Lydan, where are you?
I'm in the US, in Nashville, Tennessee. It's weirdly rainy here too today.
Yeah, okay. Okay, I'm in Madrid and it's sunny, but it doesn't really matter because I have to stay at home. So.
All right.
Yeah.
I'm sure you can go out for your groceries and stuff, right? And enjoy the sun a little bit.
If it wasn't for Neuromatch. Yeah, I hope I have some time later. Cool. All right, we're going to start soon. I'm looking forward to your presentations. Like the topics.
So I guess I should start sharing my screen.
If you wish, yeah.
Okay.
Okay, so welcome everybody who is joining. I'm gonna wait a few seconds to let everybody join before we start. This is session 3013, room 6 of Neuromatch 3, in case someone is distracted or confused.
Whenever I can start talking and presenting, tell me to start.
I'll tell you in a second. Okay, so let's start because the time is tight. My name is Alex Hernandez.
Okay.
I'm speaking to you from Madrid, and I will be the moderator of this session. It is session 3013, room 6 of Neuromatch 3, and we're gonna hear talks about autism and other conditions in children, I think. And we have a great lineup of speakers. They are Anis Zahidefard, Rianne Haratzen, and Lydan Durham. And before we start, I want to remember the audience that they're welcome to tweet with the hashtag NMC3. And of course, you are all very welcome to engage in questions and discussion in the chat, and use the Q&A tool of Zoom for asking questions to the participants. All right, so our first speaker is Anis Zahidefard. She's a master's student at the University of Tehran in Iran, where she studied electrical engineering, but I think she's now thinking about moving into computer science and neuroscience. And I think she's on the right path because she's now combining fMRI and machine learning to classify autism spectrum disorders. And I think she started this project at the Neuromatch Academy last summer. So I'm looking forward to listening to more about this.
Yeah.
Welcome, Anis, and the stage is yours.
Thank you. Thank you everybody for listening and watching our presentation. First of all, our group is composed of 2 EC engineering students, a medical student, and a physics student. And I'm going to talk about enhancing classification of autism spectrum disorder using resting state fMRI with machine learning approach and dimensionality reduction. First of all, autism spectrum disorder, ASD, is a relatively common neurodevelopmental disorder with almost entirely unknown causes. People with autism typically show abnormal social interactions, deficit in verbal and nonverbal communication, lack of empathy, and a behavioral pattern of doing repetitive actions. Early diagnosis of ASD using non-behavioral measurements can lead to improved outcomes for children with ASD, such as specialized education, which can lead them to join the society sooner and better. Children with autism exhibit various abnormal features of brain structure, most notably an increase in cerebral volume in both gray and white matter, possibly because of accelerated growth in early childhood. In addition, post-mortem neuropathology has revealed abnormalities in cortical cytoarchitecture, and diffusion MRI results point to disorganization of white matter pathways. Some scientists suggest that autism may result from an overabundance of short-range or local cortical pathways that interfere with the functional differentiation of brain regions. Loss of function connectivity during task and rest conditions has been reported, for example, between regions of frontal and parietal cortex. Functional MRI have revealed that people with autism fail to show the characteristic deactivation of midline default network areas, such as the medial prefrontal cortex and posterior cingulate cortex when transitioning from a state of cognitive rest to an attention-demanding cognitive task. The amount of the activation in the medial prefrontal cortex was inversely correlated with the clinical measure of social impairment. This lack of deactivation may be explained by the low level of default activity during rest and autism. Finally, the scientific question that we're going to talk about is how well can we detect ASD condition based on resting state fMRI data? At first, we considered the autistic children dataset, which includes images of children's faces who are diagnosed with and without ASD. But we quickly put this dataset aside because of two main reasons. At first, it wasn't very ethical to use images of children's faces, and second, it was more of an image processing problem instead of a neuroscience problem. So we decided to work on an fMRI data, which I'll describe in a minute. To address the challenge in autism spectrum disorder research and efforts toward automatic diagnostic classification, a large-scale neurological sample, such as fMRI data, is essential. In this project, we are using available preprocessed data, ABIDE, from a great dataset called ABIDE Initiative, which stands for Autism Brain Imaging Data Exchange. ABIDE 1, which was released in August 2012, and consists of more than 16 international sites that share their collected resting fMRI and corresponding structural and phenotypical information of over 11,000 individuals, including 539 people diagnosed with ASD, and the rest of them non-autistic. Subjects were from both genders and almost all ages. In addition to the fMRI data, the dataset contains phenotypic data such as gender, age, and IQ. The dataset is well preprocessed in lots of different ways, including parcellation into 200 and 400 homogeneous brain regions in the Cradock atlases and a set of other atlases. In this study guided from previous literature, we used the 200 Cradock atlases. By finding the correlation between different relevant regions of the brain, we can reduce available data for each participant from 63,201 to only 19,900 data points. The correlation matrix for each participant has a major effect on the reduction of the data size, and that can also result in more accurate outcomes. And here we can see the correlation formula, which we can use it for the Cradock atlas. Once we framed our question and chose a data set, we began the implementation stage. To classify ASD brains from controlled brains, we had to choose from many machine learning and artificial intelligence classification tools. At first step, we started to concentrate on applying a simple machine learning algorithm, multi-layer perceptron, and use TensorFlow and Cross to help with the implementation and code. And after that, the architecture of the network is shown in the slide. Our network, it has 4 layers. In the first layer, we use 8,000 neurons with the ReLU function. The second one, 13 neurons with a ReLU function again, and a third one again with a ReLU function and 4 neurons. And in the final stage, we use a sigmoid function, which is 1 neuron in the final layer. The output layer is binary, which tells us whether someone has autism, the participant has autism or not. We train our network with the splitted data into test and train the 19% of our data to train samples and the rest 10% of the data to the test sample. And to wrap it up, we reached to 72% accuracy after training, after running our code in the first step, and the 72% according to other papers was doing so great. But the major obstacle hindering us from achieving a higher accuracy was overfitting. By taking a quick look at our sample training procedure in this image, one can determine that at very early iteration of training, our training accuracy reached to 100%. However, test result will not pass over 72%. This means that our network is overfitted by training data and cannot be so accurate for data from outside, so it will get noise easily. One of the useful methods to overcome overfitting is to decrease the network's complexity. To do so, we made our network as simple as possible. Using only one sigmoid function as our activation function, we reached to 75 accuracy with 0.66 F-bonus score. We also tried to use 400 region of interest dataset, as by considering more brain regions, it could lead us to a higher precision. In this case, we trained a network of 2 layers, including one ReLU with 4 neurons and a sigmoid layer with 1 neuron, and we reached to 76% accuracy with the F-bonus score 0.63. Due to reducing complexity in our data for having a more reliable and faster computing, we've reduced the dimension of input data to reach our goal, which is choosing data points that are more effective for our classification. At first, we separated autistic from non-autistic and drew the element by element average correlation matrix of autistics and non-autistics. After that, we have differentiated these 2 matrices from each other and sorted the result matrix ascending. Finally, chose the first and last 1,000 data, because the 1,000 data from the first 19,900 data and the last 1,000 were so important and affect our data points so much. After that, we have trained the new limited 2,000 data points, and the same simplified network of 2 layers, including one ReLU with 4 neurons and a sigmoid layer with 1 neuron is used, which is shown in the picture. And the training process using mean squared error as its last function and keeps training for 25 iterations, and the results were remarkable. 84% accuracy and 0.68 F-bonus score. That according to other papers, this result was some kind of notable result for us. In the end, as our work is not ended yet, we have some future works to do. One of the main things that we're going to do is using the CNN plus LSTM architecture or by LSTM for time series, because CNN is good for feature extraction and LSTM is good for using in time series. So we can do the feature extraction and LSTM time series prediction as our architecture, because as our data may be bigger and larger than this simple ABI data set, we should use some feature extraction that is more reliable and it works better. After that, we can use ANFIS to predict the severity of autism spectrum disorder, improve our classifier using ANFIS, such as using phasey logics. And after that, we can invest the accuracy in subsets like age and sex differences to see whether they have any connectivity to determine and whether someone has autism or not. But one of the main limitations we have for this dataset was the low number of dataset participants. Thank you for your attention. I hope you enjoyed the presentation. Here's our group, me, Mernaz, Parisa, and Youssef. Thank you all.
Thank you very much, Anes, for your presentation. We have some questions from the audience. So Priyanka is asking if how did you choose the hyperparameters of the initial network architecture?
You know, we have just trained, it was just test and training things, and it wasn't really precise using. It was just reached with test and training. It wasn't because of, for example, the 5 neuron will work better. We have just tested this 5 neuron and reached better hyperparameters.
Mm-hmm. And they're also asking if you identify, so after training, if you identified some functional connectivity features that help classification more than others, I suppose.
The functional connectivity that in the last slide, let me just check it again. In the last slide, I told that invest the accuracy in subsets like age and sex differences. We can use these columns that were in our pre-processed data to see whether if someone's sex and age has any connectivity to the results of being autistic or non-autistic. We should check this later in our future work. We haven't done it yet, but we have in our plan to check the columns of a different functional connectivity between the other features and the results in being autistic and non-autistic.
Mm-hmm. That actually has to do with, so Madine Sarbestani asked me to ask you a question on her behalf about the if you used age for improving classification. So, but yeah, you asked that, so thanks. I'll take one more question from the audience. Carlos Hernandez is asking why your approach is better if you, well, they're asking, so in your table of the results, there was a random forest approach with 90% accuracy.
Our approach is not concurrently better than the random forest method, because the random forest method used the whole 19,900 data, and this made the running slower than the usual time. But we've chosen the important features in our data, the first 8,000 feature and the last 8,000 feature, and we hope by changing the architecture we might reach more than 90%, as we have reached to more than the first accuracy that we have already get. At first we've reached to 74%, and after that we have just reached to 85%, and by then I guess if we choose the right data, and because the other papers use the whole data, we can reach better accuracy, and we can just use better dimension reduction to reach better accuracy and F1S scores.
Mm-hmm. Thank you very much. I think we need to move on now.
And after that, I just want to thank Madine. Madine was our supervisor during NeuroMatch, and she helped us a lot. She guided us a lot, and I hope she is doing well. And I'm so sorry that I have informed her late, so she couldn't maybe join our talk and presentation.
She's posting another session in another room.
Yeah, she told me.
Okay, thank you very much, Anis. You have some other questions in the chat. Maybe you can reply.
Yeah. Yes, I will reply them.
Thank you very much.
Thank you a lot.
Great to see projects from NeuroMatch Academy presented at NeuroMatch here. So now our next speaker is Rianne Hartzen. She's a postdoctoral researcher at the Center for Brain and Cognitive Development at Middelburg in London, and also part of the Bond Lab led by Professor Emily Jones. And Rianne studies individual differences in brain development and how this relates to later behavior, which I find fascinating. So I'm looking forward to hearing more about this. Welcome, Rianne, and the stage is yours.
Thank you very much. Can you all see my screen properly and not us panelists? Okay. Thank you very much for that nice introduction. So today I'm going to talk about mapping the building blocks of face processing in individuals with and without autism using marker state analysis. So as we already heard in the previous talk, autism is a neurodevelopmental disorder, and it's usually diagnosed around childhood characterized by difficulties in social interaction, social communication, also restricted and repetitive behaviors, and hyper or hyposensitivities to sensory processing. But currently the diagnosis is based on lots of interviews and questionnaires with clinicians, which are very time consuming and also laborious. So there is a need for objective biomarkers that may tell us whether a person might have autism or not. And currently there's no biomarker in the field for autism. So biomarker is an objective measure of a biological process that might indicate some normality. For instance, in diabetics, people with diabetes, we can measure their glucose with a glucose sensor to measure their sugar levels in their blood, which then is a biomarker for the sugar levels. And also in autism, there's a lot of heterogeneity, which means that different people might present different clinical profiles and also different patterns in their brain activation. But one of the main core domains of symptoms in autism are difficulties with social interactions. And a recent paper from Julian Tillman in a group of the LEAP study found that social communication difficulties relate to lower adaptive functioning during daily life. So this domain of symptoms is also really affecting the daily life of these individuals with autism. And that's what we're trying to focus on, finding a biomarker related to these social communication difficulties. So one potential thing to focus on is face processing. We know that there's a slower and less accurate recognition of faces for inverted faces compared to showing upright faces, possibly due to distorted configural information, because normally when you look at faces in upright, you would process them like an eye. The eyes are above the nose, like in relation to the different features on the face. But this information is distorted during inverted processing. And the brain measure related to that is the face-sensitive N170. The N170 is a negative response about 170 milliseconds after the stimulus onset, which is slower and more negative for inverted faces compared to upright faces. And this N170 is usually measured at the posterior electrodes around P7 and P8. And previous studies have found that this inversion effect is attenuated in autism. So the difference between upright and inverted faces is smaller in autism. So next we were wondering why this might be, and by focusing on the underlying mechanisms, we can understand more about potential biomarkers if we're looking at the N170. So the quantitative hypothesis suggests that there's a reduced engagement in regions specialized for this configural face processing in autism. So that would be a difference in the engagement, whereas the qualitative hypothesis suggests that there might be activation of different regions. So there might be an alternate recruitment of regions not specialized in configural processing in autism. For instance, people with autism might activate areas that are related to object processing instead of face processing. So in this study, we were trying to find an answer to the questions, are differences in face processing between autistic and neurotypical individuals better explained by quantitative or qualitative differences? And this is kind of hard to find out using the earpiece, because with the earpiece, we're only looking at a specific time window and only at 2 electrodes, whereas we need more better analysis looking at the temporal patterns across the whole brain to find an answer to this. So the study that we used is the LEAP study, which is the longitudinal European Autism Project. It's a big study that aims to deeply phenotype individuals with and without autism and contains about 800 participants in total. Participants were recruited into different groups, adults, adolescents, and children. And there's a lot of data, including cognitive tests, eye tracking, MRI, and also, of course, EEG that I'll be speaking about now. And here below, you can see the numbers for the groups that were included in the current study. So the task that we used is the face up and inverted EEG task. where participants would be passively viewing a fixation icon for a few hundred milliseconds. Then we would show them a face for 500 milliseconds, and then there would be a blank screen. And we showed them pictures of faces upright and inverted position while they were just watching the screen. And we segmented the whole time window from -200 to 800 milliseconds, did the necessary pre-processing, and then we used multi-channel analysis with the randomization graphical user interface across all 59 channels. So not just looking at the P7 and the P8, but looking at all the channels. And this multi-channel approach we can use to look at differences in the topography using the topographic analysis of variance. We can also look at differences in the strength of these maps using global field power. And we can also look at microstates, which are quasi-stable states of brain activity. So for this, we use clustering methods to look at patterns that are consistent over time before the brain states switch to another pattern. And then if there would be differences in time and demands, which would be testing the quantitative hypothesis, we would see differences in this global field power and also in the duration and strength of microstates. Whereas when we would see different brain areas being activated between the neurotypical and autistic groups, this would show up as topographic differences and differences in the microstate maps. So these are the results for the TANOVA and the global field power. So up here in this graph, we see the results for the children, in the middle, the adolescents, and below the adults. Then for the different rows, we can see here the effects for orientation, group, and interaction. And in blue is the time where we found the significant differences for the topography, so in the TANOVA, whereas the orange time bars relate to the differences in global field power. And the main thing I want you to take away from this graph is that we can kind of see across development that topographic differences start to emerge at earlier time points, and then also end at later time points, which might suggest that there's increased development of the brain and functional processing. But we didn't really observe any differences between group that survived the duration threshold. Except for in the adults, where we found an interaction between group and orientation for the global field power strength and also for the topography. So next, I'll show the results for the TANOVA for the topography. So here we can see all the different patterns in topographies for faces up versus inverted in the children. But what I want to focus on now is this difference here between the neurotypical group and the autistic group. Here we can see that there are stronger topographic differences in the neurotypical group between the upright and inverted faces, whereas this effect is diminished in the autistic group. So again, here we see an attenuated inversion effect in the autism individuals. So next, we look at the microstate data. And here on the left, you can see all our microstate data. Each topo plot is a different microstate map. And on the right side here, we can see the differences in timing when these microstates are active. So first, what we see is that during early stage perceptual processing in the children, so around here, there are opposite patterns. There is stronger global field power for the inverted compared to upright in the children. In the neurotypical children, where this effect is opposite in the autism group. Next, during the early stages of processing, which is here, so in this greenish microstate in the adolescents and the adults, we observe again an attenuated inversion effect. So there are distinctive microstate maps with quantitative differences around this early processing stage of phases. And then at the later stages of processing, we can see different microstate maps. So here, for instance, in the children, we see this purple microstate, which does not occur during the inverted phases in the neurotypical children, whereas it does occur during the inverted phases for the autistic children. In the adolescents, we see a completely different microstate map during this processing time in the autistic adolescents, which does not occur in neurotypical adolescents. And in the adults, we see also a differences in timing between those microstates that are present. So here we see this purple microstate, which doesn't really switch to, is not really switched to from the blue microstates as we see here in the neurotypicals, or here in the autistic adults. So we found early perceptual processing differences in the children, which may relate to perceptual processing atypicalities in autism, like the hypersensitivities in visual processing. Then during the early stage of face processing, we saw this attenuated inversion effects in the autistic adolescents and adults, which may relate to atypical specialization towards faces during adolescence. And then we also saw during the later stages of face processing these different microstates maps that may reflect the development of alternative processing pathways, also involving the frontal cortex, and may also relate to compensatory mechanisms that are being developed in the individuals with autism. So to conclude, we observed developmental pathways of face processing in autism with quantitative differences during early perceptual processing in childhood, qualitative differences during face processing in adolescence and adulthood. Now, next, we would like to know whether these atypicalities are primary or secondary processes that are cascading from those early typicalities. And longitudinal research from infancy to adulthood using the sibling autism studies would really be helpful with this. And also, what does this mean for the biomarker research? Maybe we should also focus not only on the N170, but also look at the other time windows, like the earlier processing and the later processing. And also these biomarkers might be different at different developmental stages. Something that might relate to autism symptoms in adults might not be might not be showing the same relation in children or adolescents. So I want to thank the AIMS2 trials who has made this all possible. Also, Dr. Emily Jones and Dr. Luke Mason for helping me with the pre-processing and guiding me, and all my other collaborators. And thank you all for listening. And I hope I was below the 12 minutes.
It was a little bit over it, but
oh, sorry.
If someone has any question for Rianne, please go ahead and type it in the chat or in the Q&A. Thanks for such a nice talk about a great, important topic. While the question comes, so I'm curious, is it possible actually in autism research to establish like the cause and effect in development? So are the changes due to behavior and adaptation and compensatory mechanisms, as you said, or is there anything special in the brains that develop differently? Is this a question that
it is really difficult to observe that with the cause and effect from the data that we have, of course, but also looking at these longitudinal data, we can see anything in the brain in infancy that might relate to things during childhood, or things that we see during childhood that might relate to adolescence, may inform us about compensatory mechanisms and things like that.
We have one question. Thank you very much. We have one question from the audience. Sudhakar Tumala is asking, did not see any statistic regarding the strength of your biomarker. I'm not sure if I get that correctly. Is there any statistic regarding strength of your biomarkers? I guess the question is.
So all my statistics will be in the paper, but in this case, the biomarker here will be the N170, and Luke and Emily are working on a paper on that. And this research was more focused on looking at the underlying mechanisms. I looked at correlations between my microstates and the 170 latencies, but we couldn't find any significant results. And this is possibly to do with the fact that our microstates are across the whole time period. So if we see a microstate during the early time, we also see it at later times, like during the baseline. So this might be less sensitive in that case with the biomarker stuff.
I see. Thank you very much. Thanks again for your presentation. Maybe you can share the papers in the chat with us now. And now we're going to move on to the last speaker of the session. She is Leighton Durham. She's a second-year PhD student in clinical psychology at Vanderbilt University. And today she is going to talk about how global brain volumes relate to general psychopathology in children. Right. Welcome, Leighton, on stage.
Let me go ahead and try to share the screen, make sure it works okay. Okay. Are you seeing the
Go ahead. Thank you.
slide? Okay, great. Well, hi all that are here, and thank you for your presentation. Those are awesome. And thanks for the introduction, Alex. I'm excited to have the opportunity to present today. As Alex mentioned, I'm a second-year graduate student in a PhD program at Vanderbilt University in Nashville, Tennessee in the US. And I'm working under the supervision of Dr. Antonia Kazkirkin. In her lab, we investigate neural mechanisms of dimensional psychopathology. And we're particularly interested in internalizing disorders and internalizing symptoms. So today I'll be talking about the relationship between global brain volume and psychopathology in a child sample. And those listed on the title slide here below are those that helped with these analyses that I'll be talking about. Some are from Vanderbilt, others are from University of Pennsylvania and University of Chicago. Did that switch slides okay? It's all working okay? Okay, good. All right, so just to start with a bit of background, childhood is a critical time for both brain development and for the manifestation and onset of psychopathology. And importantly, psychopathology during childhood is a considerable risk factor for later psychopathology. So it's critical to promote earlier detection and prevention of psychopathology at an early age. Also, many forms of psychopathology have high comorbidity rates, and there's an increasing body of literature showing that symptoms are often dimensional, shared across different diagnoses, and hierarchically organized. There's also a substantial degree of heterogeneity within specific forms of psychopathology, so disorders don't look the same for all individuals, and they can present very differently. However, most research surrounding psychopathology uses a more categorical approach with a case control design. So for instance, a DSM-defined diagnostic group compared to a healthy control group. And the primary issue there is that you lose a substantial chunk of the spectrum of psychopathology as it manifests in reality. So anyone who doesn't quite fit into either the case group that's specifically defined or a more clean, healthy control group, however that's defined in a given study, and whether that's due to subthreshold symptoms or comorbidities or an alternate clinical presentation, for instance, those people get lost and are not considered in those type of designs. Some prior studies that have taken a more dimensional approach have shown negative associations between psychopathology and brain volume in youth samples, but the majority of them have used samples with relatively wide ranges for age, and that can obscure developmental trajectories given the amount of change in both psychopathology and brain development that's going on during childhood and adolescence. So in the current study, we aim to examine associations between regional gray matter volumes in the brain and forms of psychopathology in children from a dimensional perspective. Our analyses build on prior work primarily through a larger sample size, a more homogeneous age group, and a dimensional approach to measuring psychopathology, which could reflect the true nature of psychopathology more effectively. Our primary hypothesis based on prior studies was that general psychopathology would be associated with smaller regional gray matter volume, and that would be the case throughout the brain in a relatively non-specific pattern. So the sample for this study came from the ABCD study, which stands for the Adolescent Brain Cognitive Development Study. If you're not familiar with it already, it's a large longitudinal cohort study of brain development and child health. And the data is collected all over the US and is publicly available. So the participants for this specific set of analyses all came from the first wave of ABCD data collection, which includes children at 9 to 10 years old. And they're recruited at 21 sites across the US. The total sample size for this baseline wave is 11,875. We ended up using the final sample for these analyses of 9,607 after rounds of exclusions for lack of imaging data or missing data on other variables we needed, like psychopathology or covariates, or failure to pass relatively stringent quality assurance measures for the imaging data that occurred on the ABCD side. And the sample was split pretty evenly across sex, and the racial ethnic breakdown is listed there as well, as about 53% white, 20% Hispanic, 14% African American, 12% other. In terms of the measures we used for these analyses, we used data from a psychopathology measure called the Child Behavior Checklist for school-age children, which is commonly known as a CBCL. And it was a parent report measure about child behavior. So the individual items ask about behaviors that could be of concern or psychopathology symptoms and are rated on a 3-point scale, from 0 being not true or never true, and 3 being often true or very true. Brain volume was measured using 3T MRI imaging, and that was conducted at the 21 data collection sites. And regional GMV was defined by automated atlases, one for cortical regions and one for subcortical regions. The image processing was performed on the ABCD side by the ABCD Data Analysis and Informatics Center, and they used centralized protocols and approaches for all sites so that it was consistent. And for statistical analyses, we started with factor analyses. An exploratory factor analysis of 66 CBCL items in a random half of the sample was performed by some of our collaborators at University of Chicago and University of Pennsylvania, Tyler Moore and Ben Lahi. And they identified 3 dimensions of psychopathology that they labeled internalizing, conduct problems, and ADHD symptoms based on the items that we're grouping together. And then a confirmatory bifactor analysis with the second half of the sample was used to model those 3 factors plus a general psychopathology factor. And that represented the shared variance across the 3 factors. For the brain volume analyses, structural equation models are performed to look at associations between volume and those 4 psychopathology dimensions. And we focused on regional volume of 68 cortical regions and 19 subcortical regions. A couple of other important things to note here are that post-stratification weights were provided by ABCD and applied to account for the stratification of the sample across different data collection sites. And data was also clustered by family, since the ABCD study includes some twins and other siblings. The model we tested for each brain region was listed, was as listed there on the slide. So the covariates of note interest that we included were age, sex, race, ethnicity, and MRI manufacturer, because there were a few different scanner models used. And then we, of course, included the psychopathology factors as well. And to control for multiple testing across all of these different brain regions that we were looking at, we controlled for the false discovery rate at 0.05. All right, as I mentioned briefly, the confirmatory bifactor analysis of the CBCL data in this case yielded 4 orthogonal factors of psychopathology. So the factors are totally uncorrelated, which allows us to look at more distinct relationships and phenotypes. General psychopathology represents the shared variance across all items. So you can think of it as representing the symptoms common across multiple disorders and domains. And then 3 specific factors for internalizing, conduct problems, and ADHD symptoms represent what's unique to that domain once that shared variance is extracted. So each CBCL item loads onto exactly 1 specific factor as well as the general factor. An example of some of the items that load onto internalizing, conduct, and ADHD are listed there for those that are interested. So now I'll just summarize some of the results, and it's organized by psychopathology factor, just for clarity. So the general psychopathology factor, as we expected, was found to be, we found that increased general psychopathology was associated with smaller regional GMV throughout the brain. And this was a pretty global effect. Out of the 68 cortical regions we tested, 54 were significant. And that's after the FDR correction. And of the 19 subcortical regions, all 19 remained significant after correction. And this figure is just a visual representation of those findings for the cortical regions. The brain map shows the majority of cortical regions were significant, and the scatter plot shows that as scores for the general psychopathology factor go up, cortical brain volume goes down. For the conduct problems factor, we also found a relatively global effect that was similar to that of the general factor. There were significant inverse associations for 52 out of the 68 cortical regions and 15 out of the 19 subcortical regions. And again, that was after correction for the multiple comparisons. And this figure again just shows the map of significant cortical regions and the scatter plot of cortical volume against extracted scores for the conduct problems factor. For the ADHD factor, there was still an inverse association, but it was not as global in nature as that of the general and conduct factors. 25 of the cortical regions were significant and 8 of the subcortical regions were significant. Interestingly, we didn't find any significant regional results at all for the internalizing factor. So we didn't see the same pattern there with that factor, which I'll mention again and explain some reasons why we might think that's the case. So just a few discussion points to go over. These results are consistent with prior work that shows general psychopathology associated with smaller gray matter volumes. And it also extends prior work by showing that these associations are apparent at this young age of 9 to 10 years old, and that they show up for specific conduct problems and ADHD factors as well. Some additional relevant points from the literature to mention are that prior work has shown a general factor of psychopathology to be associated with an array of functional deficits, so including areas like self-control, emotional regulation, and executive functions. And studies have also shown reduced functional activity at the whole brain level across multiple categories of disorders when performing tasks involving cognitive, social, and reward and threat processing. So these findings all are compatible with our observation of non-specific smaller GMV across regions involved in a wide variety of functions. And as I mentioned briefly, our lack of findings for the internalizing factor is interesting, and we suspect that this could look different at a later age. Internalizing disorders become more prominent in adolescence, so it's possible that associations between internalizing symptoms and brain volume aren't showing up quite yet and will at a later time point. It's also possible that the any associations that could be there between internalizing symptoms and brain volume are occurring through the loadings on the general factor, to the point that there's nothing left for the residual specific internalizing factor to explain. So this will definitely be an important future direction for us when we have more waves of this data to look at. Some primary limitations I just wanted to mention of this study are the cross-sectional design primarily and the parent report psychopathology measure. Fortunately, we're going to have, with regard to the cross-sectional design, we will have plenty of follow-up data later on because this sample they'll continue to collect data with this sample. So down the road, we'll be able to sort of get a better understanding of these developmental trajectories in the areas of the brain, as well as in psychopathology in their true form, and look at temporal sequences more closely. And for the parent report psychopathology measure, this is interesting because some people would argue that parent report is better and some argue that self-report's better. These children are 9 to 10, so they might be at the age where they could report accurately about their own psychopathology, but all we have access to at the moment is a parent report measure. And this slide just lists some summary points and conclusions. So I'll run through them briefly. I'm not sure how I'm doing on time, but consistent with the emerging notion that the general factor may relate to non-specific variation in the brain, these present analyses provide support that general psychopathology symptoms observed in childhood are inversely associated with global gray matter volume. Reduced gray matter volume might be a transdiagnostic risk factor across psychopathology domains and disorders, but future waves of data will allow us to delineate these relationships further and help us establish a better understanding of the timing. Smaller brain volume may also represent a risk factor for ADHD and conduct problems more specifically, but we found no evidence for internalizing symptoms in this current study. And finally, considered together, these results support and expand upon the prior work on the relationship between neurostructure and psychopathology from a dimensional perspective in childhood. This just lists some acknowledgements to contributors again for these analyses, some funding information, and the ABCD study statement. And thank you guys so much for listening. I appreciate it. And thank you for the opportunity to present today.
Thank you very much, Leighton, for your presentation. Yeah, we have just a couple of minutes for questions. I think at 50 past, we're going to be kicked out automatically because there's a keynote session starting. But we have a few questions from the audience. So the first one is by Sutaka Tumala. They are saying, nice presentation. Which region showed highest association? And what is the correlation with the amygdala?
That's a great question. So that's actually going to be my next follow-up project, is to look more closely at the regional results, because it was so global that we didn't investigate the region specifically quite at the level of detail that I would like to. So there will be more to come on that, but it's a great question. We're in the process of figuring out ways that we want to look at more like clusters of ROIs and which regions are most correlated with which dimensions, and looking at it from that direction. So that's a great question.
If there's no time, I will send you the questions later.
Okay.
So Vlad is wondering if it would be interesting to look at cortical thickness or surface area to get a feel for which accounts for the reduced gray matter volume.
Yeah, absolutely. That's also a great question. We actually had a student in our lab looked at basically the exact same thing with cortical thickness measures. and found, which this was very surprising to us because they're typically pretty highly correlated, volume and cortical thickness, and she did not find near the amount of significant results that the volume analyses did. So we're not really sure why that's the case, and it would definitely benefit from further investigation, but that's a great question. We have looked at several modalities, and volume definitely seems to be the strongest by far, which is interesting.
I will take a last question before we are kicked out. So Sin Kim is saying, if I recall correctly, doesn't GMV tend to decrease in general during adolescence? Perhaps another evidence to the hypothesis that early maturation can be harmful.
Yeah, and that's also a great question. That can be really relevant to childhood trauma as well. We have a student in our lab looking right now at the impacts of trauma on brain volume and cortical thickness at this age. So that will be interesting to see what that looks like in terms of the trajectory of gray matter volume development and then decrease in adolescence. So I think that's a great question. I think the future waves of ABCD data will be extremely helpful in looking at different trajectories like this, because it's certainly, there's certainly a wide variety of individual differences. And it's nice that we have this specific age group to sort of be able to pinpoint, you know, specific relationships at a given age where the variety might not be quite so broad. But it also means that we can't say much beyond what's happening at 9 to 10 years old. So I think having the adolescent waves will be extremely impactful, so that we can, you know, do follow-up analyses and look at what this means for adolescents in this sample.
Okay, thanks, Leighton. I need to wrap up the session. I want to thank again Anis, Riane, and Leighton for their presentations, and Mel for doing a lot of work behind the scenes. And thank you very much. I really enjoyed it. Thank you very much. I really enjoyed it.
Okay, we are good to go. I'm going behind the scenes. Good luck, guys.
Thanks.
Hello, welcome to the NeuroMatch Conference 3.0. I'm the NeuroMatch Conference MC, Sabrina. And this session, we will talk about what is free will. And neuroethics, Sarah Gordon, and will moderate the talks by the philosopher Eddie Nayamis and Arina Ruskitz. If I pronounce your name wrong, I'm sorry. And here is their time. And what we are going to do in this section is the whole talk would last about 3 hours until 11. And after their lecturing, you can use the chat box to text your questions, and our lecturers will answer them one by one. It's your time.
Okay, great. Thank you and welcome, everybody. I think, just to be sure, I think we're going for 2 hours. So I think there's a little bit of confusion on the schedule, but we'll be going for 2 hours. Each speaker will take roughly 35 to 40 minutes, and then we'll have a question and answer period right after. The first talk is by Adina Roskis. She's the Hellman Family Distinguished Professor of Philosophy and Chair of the Cognitive Science Program at Dartmouth College. She is a recipient of an NIH grant in neuroethics focused on assessing the effects of deep brain stimulation on agency, and has written a lot about agency and responsibility. And I'm just going to let you get started, Dr. Roskis.
Great. Can everybody see the screen? Yes? Okay. So I'm going to talk today mostly about the neuroscience of free will, and I'll give you a little bit of philosophy ahead of time. Hold on, I'm still struggling. So when we think about acting freely, we have sort of a folk psychological conception that what happens is that time passes. Imagine you're standing on this springboard about to dive, and you have to decide. There's no external cue to tell you when you're going to jump. You have to decide when you're ready to jump. And so time goes on, and then we imagine there's a decision, and then following the decision, and in part caused by the decision, we jump, we act. So that's the folk psychological view, but I think that neuroscience has suggested that it's not quite that simple. And my plan for the talk is really to tell you what recent neuroscience says, which contradicts a very well-known view that seems to indicate that we don't have free will. So I'm going to advocate for at least the very real possibility of free will. But in order to understand what that would require, I have to give you a little bit of philosophical background, and I'm sure Eddie will give you more philosophical background. So this will be a very quick trip through the philosophical landscape. Then I'll talk about the classic model of action initiation and a neural signal that's associated with that, the readiness potential, or RP. I'll talk about recent results about the RP and an alternative model to the classic view for thinking about what these neuroscientific results mean. Then I'll discuss finally the philosophical implications and sum up. All right, so most of the discussion in philosophy about free will and the possibility of free will revolves around the concept of determinism. And that's actually a very technical concept. One of the ways of elaborating it is that if you had a complete specification of the state of the universe at any time, and you knew all of the laws, then it would be sufficient for fixing the state of the universe at all other times. Okay, so that is what determinism entails. And philosophers have typically talked about the relationship between determinism and free will, and have fallen into different kinds of camps. So what you see up at the top, I don't know if you can see the pointer here, what you see at the top are people who think that we have free will. And there are two possibilities. There's a possibility that the universe is deterministic. People who think that even if the universe is deterministic, even if the universe is deterministic, we can still have free will. And we'll call those compatibilists or soft determinists. And then there are people down here who think that determinism is incompatible with free will. And I think that that is very much the folk view, that if the world is deterministic, there's no way that we can make a free decision or act freely. These are called hard determinists. So they're a species of incompatibilists, saying that free will and determinism are incompatible. But on the other hand, you have these other incompatibilists who say, yeah, determinism and free will are incompatible, but the universe is indeterministic, and therefore we do have free will. So those are the general, main camps in which these philosophical views fall. And I'll come back to this at the end when talking about the upshot of all this. But I'm going to very quickly dismiss the value of neuroscience for figuring out this question, which I think is an empirical question, which is whether determinism is true. And there's a lot of work in neuroscience that suggests that the work that people have done by investigating the brain shows that the brain is deterministic and shows that we have no free will. And most people think that, if we could use neuroscience to prove determinism, then the consequences for free will would be pretty clear. But I really doubt that anything that neuroscience has showed or even can show says anything about the truth of determinism. And there are two reasons, at least, to think that neuroscience can't show you anything. I call those the problem of fundamental neural indeterminacy and the problem of exhaustive neural determination. And these could be an entire talk in themselves, but I'm going to dismiss them in a couple slides just to give you a sense of why I think determinism isn't something that neuroscience can talk about or answer anyway. So the problem of fundamental neural indeterminacy is the problem that we really can't tell whether something that seems to us to be deterministic or indeterministic is really that way, metaphysically speaking, or whether we just have incomplete epistemic access to what's going on. The only operational measure that we have for determinism is predictability. If we don't know, let's say, the equations governing some kind of phenomenon, then we can only decide whether or not we think it's deterministic by figuring out whether we can predict its action or its behavior. But predictability cross-cuts the question of determinism. That is, there are deterministic systems, perfectly deterministic systems that to us from the outside look indeterministic. They're completely unpredictable, such as chaotic systems. So you can get chaotic, unpredictable behavior out of a deterministic system if you don't know what the governing equations are. And then on the other hand, there are systems that at least we think are fundamentally indeterministic, like quantum systems that at macroscopic levels generally predict, to all effects and purposes look very deterministic to us. That's why we think, classical mechanics is a good macroscopic substitution for quantum mechanics. And so in these cases, you have deterministic systems that are unpredictable and indeterministic systems that are predictable. And so just merely saying whether the brain is predictable or not doesn't tell you anything about the underlying phenomenon. The second problem is the problem of exhaustive neural determination. And that is that the brain is this massively interconnected system. On the right, you see a complete wiring diagram of the only organism that I think we have a complete wiring diagram for. So these are the 302 neurons in C. elegans and all of their connections. And you can see how massively interconnected this simple system of 300 neurons are. And now you think of a system scaled up with 10 to the 12th neurons, where each of those makes something like 1,000 connections to other neurons in the system. And it's almost mind-boggling how complex such a system is. And in order to say whether some phenomenon is the result of determinism or indeterminism, you'd actually have to know what every influence on, let's say, the neuron you were recording for came from, where that was. And only if all the information going in doesn't suffice to give you the actual behavior would you be able to say something about indeterministic neurons, for instance. And so it really has, in order to answer this question, we have a requirement of global knowledge of this incredibly complex system, and we're never going to have that kind of knowledge. So this is why I think that neuroscience won't settle the question of determinism. And you might then wonder whether neuroscience really has anything at all to say about free will, but certainly there are other approaches that people have used. And so the most famous and the one that has ramified way outside of neuroscience and psychology is the work of Benjamin Libet and his argument that conscious will, which I think you might at least argue is necessary for free will, is causally inefficacious. So here's what I call the classic approach to neuroscientific investigations of free will, and they all go by way of this readiness potential. So the readiness potential is a phenomenon that was first documented by Kornhuber and Dicke in '64, and Benjamin Libet employed it with a report about the timing of consciousness of intention that I'll explain in a minute. But it has led to a very large literature on the question of free will and the way in which neuroscience can or can't affect this, our philosophical thought. So let me explain the Libet paradigm. In this paradigm, somebody has a EEG machine recording what's happening in their brain by way of electrical potentials at the scalp. And they are also hooked up to an EMG machine, which can record muscular action. And they're asked to do a spontaneous movement so that, like whenever they feel the urge, move their finger or move their wrist. So this is supposed to be a paradigm for a freely willed movement. There's no external stimulus that's causing them to move their finger or wrist when they decide to do so. And at the same time, Libet asks them to look at a dot rotating on a clock on an oscilloscope and to report the position of the dot on the clock at the time that they feel the urge or are conscious of the intention to move their finger. And that's a way of timing that conscious intention. And so when you ask somebody to do this, and here on the left is their EEG sensors, and you record from, let's say, this central point, CZ, you get a bunch of pretty noisy trials that give you what looks like a ramp up to the point at which they move, which is the 0 time. And if you do it multiple times, you get a bunch of individual noisy traces, and that's what you see here. But if you average those traces, you get a signal that is called the RP. And Libet and Deacon Kornhuber interpreted this as preparatory activity for volitional movement. So the movement occurs at the point at which at this 0 point, everything is averaged to the point of movement. And you see that preceding the movement, there's this long ramp up of activity prior to movement, which we call the RP. Importantly, though, the RP really comes from at least about 40 individual trials of this movement, and individual signals are quite noisy and quite diverse. So as I mentioned, while doing this, while creating the RP, people were asked to say where on this clock face the dot was when they became aware of the decision or urge to move. And that the aim of that is to measure the time of consciousness of willing of an action relative to the time of the neural signals related to the action that are being recorded by the EEG. And what Libet found is that the time at which you were conscious of willing the action was about 200 milliseconds before your action. But as you can see, the time is also quite variable of when people actually report feeling aware of their intention to move. But it is not before the RP starts to exceed the baseline level. That is, the ramp of the RP begins before people are aware of willing their action. And so Libet interpreted this as follows. He said the brain evidently decides to initiate, or at least to prepare to initiate, an act at a time before there's any reportable subjective awareness that such a decision has taken place. It is concluded that cerebral initiation, even of a spontaneous voluntary act of the kind studied here, can and usually does begin unconsciously. He goes on, these considerations would appear to introduce certain constraints on the potential of the individual for exerting conscious initiation and control over his voluntary acts. So his bottom line is that your brain decides before you do that it's time to move. And if your brain is deciding and you aren't, then you don't have free will. So this is just a summary of the results. We're mostly interested in these type 2 RPs, which are generated with these spontaneous actions. But the ramp up of the RP begins approximately 500 milliseconds before action. The consciousness of willing is about 200 milliseconds before action. And there's a little lag between when your brain signals, your neurons in motor cortex start firing and when your muscle moves. But anyway, this result has been replicated multiple times. There's absolutely no question that we get RPs before we voluntarily act. So anyway, in summary, I think it's widely taken by many people in philosophy and neuroscience that we don't consciously initiate our spontaneous actions. There have been multiple attempts to sort of save that. So for instance, Libet said, well, maybe in that interval between when our brain decides to act between when we're consciously willing of acting and when we actually generate this motor signal, we could veto this action that wasn't started by us with free won't. Turns out that free won't doesn't work either for much of the same reasons that free will is called into effect, which is that also takes time. And seems to have an RP associated with it. But also Libet even himself recognized that there are many voluntary actions that aren't spontaneous and quickly performed. In fact, we voluntarily pre-plan things that don't immediately lead to action. And he says, in those in which conscious deliberation of whether to act or of what alternative choice or action to take precedes the act, the possibilities for conscious initiation and control would not be excluded by the present evidence. And in fact, many philosophers have pointed out that if you think about the kinds of things that we're really concerned about free will for instance, the kinds of things that we assign responsibility for, praise and blame, typically those things aren't very closely related in time to the action. You can premeditate a murder that isn't affected by the kind of Libet style experiments. So I want to just distill the commitments of this classic view. First of all, it's a post-decisional view in the sense that the decision happens early and the RP is taken to be neural evidence of a decision to move. It follows upon that decision. It's also taken to be an ontologically real signal. So the RP is a readout of a causally efficacious neural process. That is, interpreters of these experiments take the signal of the RP to be a commitment to action. And it has a distinct and measurable onset, which is the time at which that ramp deviates from the baseline activity. And finally, in conjunction with the willing experiments, the RP, at least in situations of spontaneous movement, precedes subjective consciousness of decision. And so this is what leads to this widely accepted doctrine that conscious will is efficacious, and because it is inefficacious, it's not free. Now, there's interesting philosophical questions about the importance of consciousness to freedom. I'm not going to go into those here. Now, needless to say, there are many, many critiques of this classic interpretation. There's a lot of variability in the W timing. You might think that the whole paradigm for the Libet experiment is the wrong paradigm for thinking about free will. In fact, it's not really you're not really looking at a decision to move or not, but rather when to follow an instruction. So it may not be the right paradigm to investigate free will from the very beginning. Similarly, other people have argued that these kind of simple motor acts like moving your finger are not the right kind of model system for investigations of free will. I think that the Libet view is that all actions are just complexes of basic actions like moving your finger. So what's true of moving your finger is true of much more complicated things. But neuroscience has shown us that that's absolutely not true. In fact, it takes more neural activity to move a single finger than to move a whole hand. And so the moving of the hand is not just the sum of individual fingers. The idea that decisions in the absence of reasons, which is what this kind of paradigm is what a free decision, sort of the paradigm of a free decision is also something that has been contested. And as Libet himself notes, many of our questions about free will involve much longer time scales. So those are, I think, classic critiques of the classic interpretation. But nonetheless, I think that there's something interesting and probably correct about the intuition that free will, or at least moral responsibility, that is linked to free will, depends upon conscious decision-making or having some kind of conscious intention. And I think that that relationship is still under-theorized in philosophy. One question you might ask is the kind of consciousness that we care about, the type that's measured by the Libet task. And I've argued that, in fact, the Libet task doesn't measure the right kind of consciousness to really make any impact on the question of free will. So it really involves two different tasks. One is spontaneously moving your finger, and the second is reporting the time of the awareness of intent. And in order to do the second, you have to recognize your own intention to move, and then you have to index that with respect to this external clock. So what you have to do is attend internally, and then shift your intention from your internal state to the outside world in order to report the time at which you are aware of the result of your measurement of W. So this attentional shift takes time, but moreover, the recognizing one's own intention is a meta state. That is, it's a conscious state with the content of an intentional state. And it therefore depends on that intentional state of willing to move, and must therefore at least be temporally post that state. It's a state about another state. And so for those reasons, I think that Libet's measurement isn't really of conscious intention, but consciousness of intention. And that is going to be something that follows upon when one actually consciously intends to move. So what he's measuring is this consciousness of intention, which is, late in this decisional process, but that leaves open the possibility that the willing could happen well before that. So that's a criticism, I think, of the standard Libet paradigm. But now I kind of want to shift gears and question the paradigm much more basically. Are we actually even looking at what really matters for free will and thinking about it in the right kind of way? So in the next section, I want to talk more about some more recent empirical findings about the RP, and then some new models that I think should make us very suspicious of the commitments that I talked about before. That is, that the RP is post-decisional and that it's ontologically real. Okay, so it's pretty clear that the RP is a real signal in the sense that we can replicate it very reliably. It's visible in the EEG prior to spontaneous voluntary movement. When these traces are time-locked to movement onset and averaged over multiple epochs. One thing we see is that there's significant variability across individuals, and it's difficult to resolve in single trials. What's going on? I'm going to show you a little bit of that. And the temporal extent of the RP actually seems to be very closely related to or influenced by planning. So for instance, if you ask somebody to plan a movement, you see a much earlier RP. Any kind of anticipatory task affects the timing of the RP. So here's just a quick slide to show how variable these RP traces are across people. The shape varies, the amplitude varies. Some people, like subject 4, don't seem to have any RP. Nonetheless, when you average across many people, you get the standard RP shape. Again, very, very difficult to resolve in single trials. This is what it looks like with one trial. This is what it looks like with 10. When you get to about 40 or 50 trials, you get something that looks like an RP. And then most of the studies report trials far in excess of that, like something like 200 trials. So I think it's puzzling if you think in the classic view that the RP is a signal that happens every time you spontaneously move prior to movement. It's weird that not all subjects exhibit an RP. They can do the task. It's a little odd that the shape and the onset of the RP seem to be highly variable. And even more importantly, you would think if the RP is the signal for a commitment to move, that you would be able to predict movement by measuring the RP. But it turns out that it hasn't been a good predictor of movement onset. And in a new study that Aaron Schurger has led, they've tried to train machine learning networks to predict when somebody's going to move. And they've used sort of standard Libet tasks, and then they've used tasks in which they've controlled for anticipation. And in the standard Libet task, you get what you might expect, which is, again, these are average trials, but you get the ability of the machine learning system to predict movement happens about 1.5 to 1 seconds. Good prediction happens 1.5 to 1 seconds before the movement occurs in all these subjects. However, if you control for anticipation, the machine learning system is essentially a chance until right before movement. And so that's another indication that the RP is not giving you information about an oncoming movement. There's also that the RP doesn't seem to reflect a commitment to move, in the sense that you can get the beginnings of an RP and then withhold movement. So even though the free won't doesn't seem to be a savior for free will according to the Libet task, it looks like the RP itself is not a one-way signal for movement. The RP is also present in tasks that don't even involve a motor component, but involve only non-motor decisions. So that also calls into question the close link between RPs and motor activity and initiation of movement. And it's present in tasks involving decisions not to move. So it seems to be present in all these decisional tasks, regardless of whether they're movement decisions or other kinds of decisions. You wonder why, if the RP is present in all kinds of these decisional tasks, why we're thinking of it as a movement initiator in particular, especially when it doesn't seem to be a good predictor of movement onset. And one of the reasons you might worry about it is because what you're doing when you create an RP is you're always averaging, you're only taking trials in which somebody has moved, right? You're not looking at trials in which they don't move because there's nothing to time lock to. You don't know what you're looking for. And that's like trying to predict the weather by only looking at what happens on rainy days. But then you can't figure out what the cues are for what makes a rainy day happen. It's just not, in other words, what you're doing is there's a sampling bias in looking at brain activity when all you're looking at is brain activity that always leads to movement. But as I mentioned, the RP seems to be present in all sorts of decisional tasks. And Aaron Sugar and colleagues recently suggested that, in fact, a basic decisional model that he calls the stochastic accumulator account is also able to produce an RP. So the basic idea is that if you have just a diffusion to bound model, a decisional system, and you average, so in these diffusion to bound models, what happens is the decision essentially is made when some kind of threshold is crossed. And there may be lots of variability prior to reaching that threshold. And that's a sort of general model that people in psychology and neuroscience have used to characterize decision-making. The RP, that the same shape, can result from averaging many trials that are time locked to movement when movement is considered the decision threshold. To just give you a sense of this, whenever you see a threshold crossing in this stochastic model, that shows you how long the time is between when someone moves and when the RP starts. And this kind of stochastic model can reproduce both the behavioral results in terms of the waiting time that people typically show between different movements of their finger when put in a Libet paradigm, and it produces the same kind of EEG trace that you get from averaging all these traces time locked to movement in real people. So the model very nicely replicates the RP results, but the interpretation of what's going on is totally different. In this model, what's happening is that there is a decision process that is going on, and the decision happens essentially when you cross the threshold, and that is very closely temporally associated to the movement. So, the post-decisional model, the classic model, has the decision occurring when the deviation from baseline activity starts. So that's 500 or more milliseconds prior to movement. On the stochastic accumulator model, what you have is just this sort of random walk, because remember, this is a situation in which there are no cues to respond to. And so the decision happens when you cross the threshold over here, right before the action occurs. So nothing in this prior period has been decided, even though if you averaged all these traces together, what you would get would look exactly like the ramp of the RP. Okay, so I think recent studies have shown that there's a converging evidence in favor of this kind of pre-decisional account. That is, the neural decision to move actually occurs over here rather than back here. There's other evidence that's consistent with this. For instance, what you get with the decision models is noise reduction that's preceding spontaneous voluntary movement, which is just a natural consequence of the averaging procedure, and that's in fact what you see. I don't think that it necessarily speaks one way or the other, but it's consistent with this. There's a study that looks at whether RPs happen all the time. And these studies, I think, are also problematic in the sense that the RP, we don't know what the individual RP-like event is going to look like if, in fact, the stochastic accumulator model is correct. And so I think all of these studies that have been interpreted to show that perhaps Libet is right, in fact, are also consistent with the stochastic accumulator model. Okay, so let me just run through the implications of the stochastic model. As I said, the stochastic models are pre-decisional. That is where the RP is already detectable and has no bearing on whether or not a decision to move has yet been made. In fact, the suggestion is that it hasn't. In fact, if the decision is at the threshold crossing, then the decision follows the W time. That is, our folk psychological concept that first we form a conscious intention to do something and then we actually do it follows naturally from the stochastic accumulator model. There is some evidence that the RP might reflect the buildup of a signal of which the agent becomes aware around the W time. And as many have suggested, that's consistent with the idea that consciousness results from a temporally extended process. So those, I think, are all very naturally compatible with our folk psychological model of free will. Another thing that this model suggests is that the RP is really an artifact. It emerges from biased sampling that comes from averaging EA traces that are time locked to action. It doesn't then necessarily reflect an underlying neural process that's present only prior to action. It's just that we only look prior to action, and we don't know what to look for in other epochs. So you might think that RP-like events occur all the time, but they don't lead to action. And that's completely consistent with all the studies that have been done. And then if this is true, then there's no physiological significance to the notion of an onset of the RP, right? The RP is just an artifact of our analysis. What is really happening is that there's ongoing fluctuations of neural activity in the areas that we're recording from, and fluctuations of a certain magnitude indicate a decision. And so if there is no onset, then the RP onset cannot cause anything or reflect a real causal process. That is also, that may be true, but it's also possible that you could get RPs from real causal neural signals, right? So you can get RP-like traces from little excursions that are related to what we think of as motivational signals, et cetera. The accumulator count is neutral with respect to whether the signals that lead ultimately to decision are driven purely by noise or whether they're driven by real but small signals that are in these traces. Okay. Pretty much to sum up what the stochastic model tells us, I think it negates the commitments of the classic view. So the classic view says that the RP is post-decisional, that it's neural evidence of a decision to move. But the accumulator model suggests that it's pre-decisional. It just reflects evidence that contributes to a decision to move. The classic model says the RP is ontologically real, that it leads to action and has a distinct and measurable onset. According to the stochastic model, it's artifactual. It is an artifact of sampling. The underlying signal may be a causally efficacious process. It's the decision process, but it has no measurable onset and may not even resemble the RP in individual trials. The RP is supposed to be non-conscious or unconscious. We have no idea whether the RP reflects a signal that gives rise to subjective consciousness of decision or intention. It seems like the data is compatible with it doing so. All right, so now that's the neuroscience. What does it mean for the free will debate? What are the philosophical implications? So first of all, if the RP onset is meaningless, then relating it to the W time is also meaningless. So it undercuts completely the Libet-style arguments for unconscious decisions. The decision in the Libet-style tasks in the Sugar interpretation of the RP is due to noise. Now that has some philosophical implications. So you might think that in cases of spontaneous action, like the Libet task, when you're asked to act essentially randomly, there are no considerations for or against when to move. It's appropriate to just let decisions happen using noise or whatever signal is in the system that can make it choose to do something. So I think that even if in Libet-style tasks, the RP is due to noise, it doesn't really threaten any conception of free will. But you might think that there are some reasons that philosophers might be worried about that. So if you are an agent causation theorist, for instance, that is that you think that the agent provides some oomph that leads to an uncaused cause that leads to action, the agent causation theorists would probably not be satisfied by the accumulator model as an account of free will. There's nothing agential about this noisy process leading to decision. If you're a libertarian and you think that really what matters is that there's indeterminism and that it's indeterminism that confers free will, then if the noise is indeterministic noise, and as I said, we don't know and we can't tell, the libertarian would be happy with that. The compatibilist would also probably be happy with that. So in the case of random decisions, I think the compatibilist would say, why not just let noise or whatever else signal work in that case? Because there's no reason for or against moving at any particular time. So I think in a Libet-style paradigm, that wouldn't be a problem. And these kinds of decisional models are exactly the kinds of models in which you might think evidence or reasons can work for making decisions in cases in which reasons do matter. Moreover, even in the Sugar model, where noise is what drives the system over threshold, he still hypothesizes that there's a deviation from the baseline that's set that allows the threshold crossing to occur. So that threshold, that deviation from baseline may still be something intentional and conscious, and that would give the agent some sort of force, some kind of motivational causal force that leads the decision to happen when it does. A source compatibilist, somebody who cares about whether the source is really you, may still be worried. There may not be enough of myself in this decision-making mechanism for it to count as free will. Of course, I have a package delivery and somebody honking at me right in the middle of my talk. So there are other stochastic models that are also able to produce RPs. So if stochastic neural excursions aren't just noise, but in fact reflect volitional processes such as motivational strength. So we've been playing with a bumps model or motor ideation. Then the threshold crossing events are not entirely due to noise and instead are causally connected to volition. And this would, I think, strengthen any compatibilist view. And a volitional component may be enough to secure agency or free will for decisions, even for an agent causation theorist. So no one has seriously suggested that we have to be aware of all the causal influences on our decisions for them to be free decisions. Sarah, how much time do I have?
You should be wrapping up shortly. Thanks for asking.
Okay. All right, I will skip the next couple slides, but the main point is that there certainly seem to be biases in this kind of decisional model, but the bias, the argument is that bias is something that you see even in the regular Libet paradigm. So it certainly isn't something that we think is incompatible with free decisions. So let me just summarize. The classic post-decisional models assume that the RP onset has physiological significance, and they open a space for comparisons between onset time and W time, and thus for arguments about inefficacious conscious will. A family of stochastic models can equally well model RP characteristics. However, under these models, the classic RP is a natural consequence of analysis techniques, but does not entail that in individual trials, there's an electrical potential with the characteristics of the RP that signifies action initiation. Instead, underlying decision processes give rise to decisions that occur approximately when we are aware of making them. None of these models challenge the efficacy of conscious will, but the model details can influence the way in which decisions and awareness thereof are related. That is, these models have philosophical implications and they interact with philosophical positions on free will in interesting ways. But at this point, I think that we can probably reject, at least in my view, reject the classic view and adopt a view of intellectual humility, because there are still many things we don't understand about how actions are initiated. That's it. And I want to thank my colleague, Aaron Scherger, for his help on this as well. Thanks.
Great. Thank you so much. And we have a great set of questions going. The first one that has the most upvotes here is from, I'll just read out the question. From a materialist or modern day neuroscience point of view, wouldn't it be the base assumption that neural activity gives rise to consciousness and therefore must precede it? So I guess this is the kind of question about, doesn't the problem go all the way down?
I'm not sure if I think, I want to say yes. I want to say yes, a materialist is perfectly fine with brain activity giving rise to both consciousness and action, and that doesn't seem to be a philosophical problem. But that is not the Libet interpretation, in part because of the inversion of the timing and the initiation. But it's only if you interpret the initiation of the ramp as a decision that that problem arises. And I think that the standard materialist view is unproblematic in that case.
Okay, maybe this one will help too. I really like the distinction between conscious intent and, oops, suddenly I lost it, sorry. Between conscious intent and consciousness of intent, but doesn't this argument also assume a straightforward representation of time? In principle, couldn't I have a representation of intent that occurs after the intent, but it represents it as if it happened before the intent?
Yes, I mean, it's definitely, it certainly is the case that there's evidence that we interpret things not necessarily according to their actual chronological place. And so we we interpret not only what is happening, but we interpret when it's happening. So I think there's always the possibility that our location of something in time is displaced in one way or another. However, if what you're measuring is this objective thing out in the world, which is the point on the clock face, then you are not subject to that problem, right? Does that make sense? I mean, you're not locating something in time, you're locating something in space in that case, and it's the spatial thing that's locating it in time.
Sorry,
But
thanks.
I guess let me just add a little bit, but what you are doing is you are identifying some internal state at a time, right? And the point at which you identify that internal state doesn't necessarily correspond to the point in time when that state has occurred, but it is, I think, impossible for it to actually be identified prior to its occurrence, if that makes sense.
Okay. Another question. Ooh, my screen moves the questions. What areas of research do you think will provide insight into free will, since you're saying that neuroscience isn't really helping?
I don't want to say that neuroscience isn't helping. I do think that understanding decision making, volitional processes, action initiation is all helping. I don't think neuroscience is going to help with the determinism question. And I don't think that reliance on the RP is very helpful as a strategy for understanding free will. But I think that understanding the underlying mechanisms of decision making and action initiation and all that helps quite a bit.
Okay, and I think we'll do at least one more question here. Machine learning is able to find some signal in the activity to predict the action. That sort of answers the objection that the RP itself is just an artifact of averaging near the event. There is something in the signal, whether RP or something else, that occurs on that time frame prior to movement. So the question is, can one extract what the feature is and maybe help us understand how accounting for anticipation changes that?
Yeah, so there is signal in there, and that's true also under the stochastic accumulator model, right? There's increased neural activity that leads to the crossing of the threshold. And so, for instance, if you're more likely to cross the threshold when you're near the threshold and you can measure being near the threshold, you're going to get information about the probability of crossing the threshold. Now, it's possible that there are also real signals buried under there that contribute to crossing the threshold. And I wouldn't be surprised if there are signals that, if you could really get the sensitivity and the specificity that you need, that you could latch onto, that would tell you something about, conscious initiation or something. But the problem is that the individual signals, if they exist, the individual signals are within the noise, same magnitude as noise. And so it makes it really difficult to pull out. And that's why I think these kinds of techniques for triggering on the basis of EEG predictions of whether somebody is going to move have been not that successful, right? So people are better than chance using these signals, but they are definitely not, even at 90% or something. People have built these real-time machine learning systems that try to predict upcoming signals, and they're somewhat successful in doing it, but you could be somewhat successful doing it on the stochastic model, even if it's just a noise model, as well as if there's real signal in there.
I think I'm going to do just one more, and then we're going to switch. So what about non-movement related RPs or ERPs? What can research in this domain tell us about that?
So that's an interesting area. The fact that you see RP-like activity in those cases, I think, is evidence for a more general decisional model that maybe is non-specific in terms of whether it applies to motor initiation or some other kind of decision. So I think that it's evidence for the stochastic model. In the literature, there have been other kind of slow wave phenomena that have been characterized that are slightly different than the RP. There's the CNV and the SPN. And the only thing that really identifies them is the experimental paradigm that generates them. And so there's a lot of discussion about whether they amount to essentially the same or overlapping underlying cortical generators. So I think that in some ways, part of the problem lies in these kind of, identifying different constructs on the basis of picking out experimental design and then doing these multiple trial averaging and then calling them different things, even though they may very well be the same thing. And I think the literature is very, very unclear about relationships between RP, CNV, and SPN. So some people will say, you just don't have an RP if there's no movement. But it's not that you don't have underlying signals that look a lot like the RP, you just call it something else, right? And so then how do we generalize across different experimental paradigms? For instance, in a paradigm that doesn't involve movement at all, are we thinking that we're looking at something entirely different because we call it something different? Or are we actually thinking that we're looking at the same kinds of processes that might vary somewhat because instead of motor cortex, you're recruiting some other part of the brain that's subserving a different function? So I don't think that the literature is clear on those parts, but I think it's something that still needs a lot more elaboration.
Okay, thank you so much, Dr. Oskis. I think I'm trying to monitor the chat too here. I think we're going to stop there and move to our second speaker for the second hour. Again, it was mistakenly listed as only 1 hour. We're going for 2 hours, but even then we want to be able to give people time at the end of the 2 hours. I think we're supposed to finish a little bit before the hour. So we'll move now to our second speaker. Dr. Eddie Namias is professor of philosophy at Georgia State University, as well as associate faculty for the Neuroscience Institute there. He's written extensively on free will, including a New York Times piece, "Is Neuroscience the Death of Free Will?" It's not. And he's done some work in experimental philosophy as well, looking at ordinary people's intuitions about free will and moral responsibility. And he's part of a Templeton Foundation grant on big questions in free will. So welcome and take it away.
Thank you. And thank you to Adina for setting up a lot of the discussion I'll be having. I'm going to try and broaden out a little bit in several ways. Thanks to Sarah for moderating and to NeuroMatch for hosting us. And thanks to those of you who are not leaving, even though you thought the session was over. Hopefully I'll be able to keep you here for the next hour or so. But I want to begin by actually testing or letting you test your own intuitions. If we had something more complicated set up on Zoom, we could take a poll of your actual responses, but I'll just let you write down what you think about this scenario I'm going to give you, which is a scenario we used in a paper that was published in Cognition a few years ago called "It's Okay If My Brain Made Me Do It." So what I want you to do, and sorry it's a bit long, and I'll read it in case the words are a little small for you to read, is just listen to this scenario about a future possibility and then answer some questions about it. And then I'll talk about what it all means afterwards. So recent brain scanning studies have shown that specific patterns of brain activity predict simple decisions several seconds before people are consciously aware of those decisions. Maybe like if Libet's experiment worked that way. Imagine that in the future, brain scanning technology becomes much more advanced. Neuroscientists can use brain scanners to detect all the activity in a person's brain and use that information to detect the activity that causes decisions and predict with 100% accuracy every single decision a person will make before the person is consciously aware of their decision. The neuroscientists cannot, however, do anything to change brain activity and hence cannot directly influence thoughts and actions. Suppose that in the future, a woman named Jill agrees as part of a neuroscience experiment to wear this brain scanner for a month. It's a lightweight cap. The neuroscientists are able to use real-time information about her brain activity to detect the activity that causes her thoughts and decisions and predict everything Jill will think or decide, even before she's aware of these thoughts or decisions. However, they can't alter her brain activity to change what she thinks or does. On election day, Tuesday, let's say, Jill is, this is in the future, Jill is considering how she will vote for president and for governor. Before she is aware of making any decisions, the neuroscientists can see, based on her brain activity, that she's about to decide to vote for Smith for president and Randall for governor. Just as the neuroscientists predicted, she votes for Smith for president, Randall for governor. As with her decision to vote for Smith and Randall, the neuroscientists are able to predict every decision Jill ends up making with 100% accuracy while she's wearing the scanner. Occasionally, Jill tries to trick the neuroscientists by changing her mind at the last second or stopping herself from doing something she just decided to do, but the neuroscientists predict these events as well. Indeed, these experiments confirm that all human mental activity just is brain activity, such that everything that any human thinks or does could be predicted ahead of time based on their earlier brain activity. So that's the scenario. Now imagine that you've got your 7-point Likert scale in front of you. From 1 to 3 is on the disagree side, 5 to 7 on the agree side. Try to avoid using 4. Come on, take a stance. So first of all, is it possible for this technology to exist in the future? So think or write down what you think about that answer. Now, whether or not you think the technology could exist, what do you think it would mean if it did exist? So, during the experiment, when the neuroscientists predicted what Jill did, she had free will. Agree or disagree? During the experiment, when the neuroscientists predicted what Jill did, her reasons had no effect on what she did. If this technology existed, then people would not have free will. If this technology existed, then people would not be morally responsible for their actions. If this technology existed, then people would not truly deserve blame for their bad actions. This is a kind of comprehension check, perhaps. If this technology existed, it would show that each decision a person makes is caused by particular brain states. All right. So I wish I could see everyone's answers summarized nicely in graphical form. But what I want to do now, before I show you the actual results, is give you a sense of 3 ways that people might predict ordinary people's responses and what those predictions show about sort of the metaphysical assumptions that those people are making when they make these predictions about what ordinary people are going to say about neuroprediction. So the first one is from people I call illusionists. These are normalist scientists and psychologists who think that free will is an illusion. And there's been a lot of those people. I'll talk about some of them shortly. here's the sort of argument that they would give as a prediction of what people will say. They're going to think most people are dualists. Most people think the mind and body are separate things and the mind is somehow non-physical. And they're going to be libertarians about free will. They think free will requires some kind of indeterminism. The mind is somehow outside of the natural laws. And so, because they think ordinary people think that, they're going to predict, most people would say neuroprediction's impossible because dualism and indeterminism should rule it out. Why is that? Well, because the neuroscientists can't see the people's soul or their non-physical mind. And if indeterminism is true, there's going to be, they're not going to be able to predict with 100% certainty what people will do based on their prior brain activity. So they should predict that most people will think these scenarios are not possible, that the neuroprediction couldn't happen. But if you asked them, well, what would it mean if it did happen? People should say that that Jill didn't have free will, that people wouldn't have free will if this scenario were presenting an actual future. So that's one possibility, the Willusionists. Another possibility, which we'll call metaphysical intrusion, is it begins with the same first premise about what ordinary people believe, but it makes it so strong that people just can't give up their dualist and libertarian ideas. They're just so strongly committed to that metaphysics that no matter what you try to tell them about the scenario, they don't really conceive of it. And so what happens is they then intrude dualism and indeterminism into the scenario. So no matter what you tell them is happening in the scenario, they don't buy it. And they think Jill somehow has a soul that the neuroscientists can't see, and she can make different decisions no matter what they predict. And on that view, even if people were to say it's possible, they don't really believe it is possible. And they don't believe that it would show people don't have free will because they can't give up on that idea. So that's a second scenario, or a second set of predictions. I think both of them are wrong. So I'll give away what I think is the right prediction to make. And I call it the natural response, to move things in my direction intuitively, but also to signal that I'm suggesting that most people are going to be okay with a naturalistic way of thinking about things, but they're going to be okay with it mostly because they're what I call theory light about the metaphysics and scientific understanding of the nature of mind and free will. So this is the way this view goes. Like the intrusion view, I do think we have some pretty non-negotiable beliefs about the way we work. But they're not about the metaphysics. They're just more about the way things seem to us and the way we think our conscious agency works. So we do believe that we're conscious agents and that we make choices. So we don't accept that ourselves could be completely bypassed, that our reasoning and planning processes make no real difference to the world. So I think people pretty strongly feel that to be the case. That would suggest if there were real bypassing results from neuroscience or from metaphysics, that would be a threat to free will. I don't think there are, but we can talk about that later. Despite those non-negotiable beliefs, people have what I'll call negotiable beliefs about the underlying metaphysics that make sense of those experiences and capacities of agency. They're theory light about those things. Now, what's important to notice is we don't yet have a neuroscientific theory that fully explains consciousness and agency. We're not there yet. We're at the point where we have really good reason, like Copernicus suggested, to think, Copernicus suggested the Earth moves really fast. It's spinning and going around the sun. And that was an interesting theory, but it didn't really make a lot of sense until we had Galileo, who explained to us why that picture of the metaphysics of the universe was not completely counterintuitive. How did he do that? by getting us to think about the fact that if we're moving on a very fast moving ship, then we don't feel like we're moving. If it's not accelerating, if it's just going very fast. You can throw a ball up in the air and catch it and nothing happens. Once you have that Galilean theory to explain Copernicus, it was okay. My view is we're pre-Galilean now, but when you give people this scenario of perfect neuroprediction, it suggests we must have reached a post-Galilean stage. We must have reached a point where the neuroscientists and philosophers probably helping have figured out how to make sense of how the brain explains consciousness and agency. So when you present people with that possibility, they assume, oh, you know, we already know the neuroscience explains lots of things about motivation and behavior. I guess by the time they get this neuroprediction device, they'll have figured out everything, including how neuroscience explains how we consciously plan and make decisions. And they can see it right before Jill does it, but that doesn't mean that Jill's not doing it. It's her brain doing it, but she is her brain, so it's all cool. So that would suggest that if you tell people this technology is possible, or if you show people this technology, they'll say, I guess that's possible, because, you know, why wouldn't it be? And if it were actual, it would suggest naturalism must be the metaphysics. But that's okay, because it would just give a naturalistic explanation of how free will and mind work. So that's the prediction that I think makes sense of the actual data I'll show you. And then in the second half of my presentation, I'll try to actually explain how to make sense of this naturalistic possibility in a way that is consistent with determinism and physicalism or naturalism about the mind. But let me begin by just pointing out that that's not the way most physicalist scientists talk about things for the last 20 or plus years, or maybe going back to Libet in the 80s. They assume that we have this sense of ourselves as agents and as causing what we do, but that's all an illusion. That's Daniel Wegner talking about this view in his book, The Illusion of Conscious Will. Another psychologist brings up the Copernican idea to say it's an illusion that we have free will, just as it's an illusion that the sun moves through the sky. Neuroscientist John Dylan Haynes says our decisions are predetermined unconsciously before our consciousness kicks in. Seems the brain's making the decision before us. Notice the dualistic language there and in most of these illusionists use. They just assume that the soul isn't doing anything because there is no soul, and that will mean free will is an illusion. Similarly, Josh Green and Jonathan Cohen write free will as we ordinarily understand it, which they think is dualistic and libertarian, is an illusion. And neuroscientist Patrick Haggard says, again, not in the sense we think. And there's my favorite illusionist of all, Sam Harris. If you want to see Sam Harris taken out by philosophers, Daniel Dennett or me, you can look up our reviews of his book, Free Will. But here's what he says. Imagine a perfect neuroimaging device that would allow us to detect and interpret the subtlest changes in brain function. Imagine what it would be like to see the time log of these mental events alongside the video of your associated behavior, demonstrating the experimenters knew what you would think and do just before you did. You continue to feel free in every present moment, but in the fact that someone else could report what you were about to think or do would expose this feeling for what it is, an illusion. So that should sound very familiar. It was the basis of the scenario we developed, one of the illusionist bases of that scenario, to test his prediction of what people would say. So let's see what people say. There's Jill's brain cap. Actually, it'd probably look a little more sophisticated than that. And I asked you and our participants in the study, which were a bunch of college students at Georgia State, is it possible for this technology to exist in the future? So I don't know how many of you said yes, but about 80% across, we did several versions of this, but about 80% of people said, yes, it's possible. So that already suggests there's something wrong with the illusionist prediction. As long as people were not intruding their metaphysics into it and they were understanding the scenario, and we had several comprehension questions like that one I asked you about whether the brain causes decisions, and most people got that right. So most people think it's possible. The ones who said it was impossible typically didn't mention souls or immaterial minds or even free will. They mentioned the sorts of things you guys as neuroscientists probably realize, which is this ain't happening. There's all kinds of technological reasons that there's no way we're going to be able to pull this off. There's also, we're in a neuroethics session, there's lots of ethical reasons why we might not ever let such technology be developed if we could prevent it. But putting aside those sort of pragmatic reasons, most people didn't seem to think there's any metaphysical reasons why it couldn't happen. And it even ends with that strong metaphysical statement of physicalism. So you can read more about these results in this cognition paper if you're really interested. Here's some other quick results. We had several scenarios, one where they, like you read, they can't manipulate, one where they can, but in some cases didn't. Can you see my arrow when I scroll on here, Sarah? Okay. And then one where they can and did manipulate. And what you see is, this is on the 7-point Likert scale, you get strong agreement that Jill had free will in general, that her votes were free, as long as she wasn't being manipulated. And then all those questions about moral responsibility and blame have the same patterns of results. And just more generally, the percentage of people who said Jill had free will in her votes is here at 90%, that her decisions in general were free. And then that question, if this technology existed, it would show people do not have free will, reverse coded. Most people said no, it wouldn't show that people don't have free will. I can come back to this bypassing mediation analysis if you want later, but very briefly, most people don't think that the technology shows bypassing, that her reasons play no role on what she did. And the people who did think it was bypassing, that explained much of those people's answers to free will and responsibility. That is, if you think it causes bypassing, you think, yeah, it threatens free will. If you don't think it causes bypassing, because maybe you take that natural interpretation I was suggesting, it doesn't take away free will. So to summarize that experimental philosophy part of the talk, it looks like most people are not answering the way the illusionists predict they would. Surely the illusionists would then turn to the intrusion answer, as some philosophers have done in response to these experiments, and said, no, people are misunderstanding. They are dualists and libertarians, but when you show them this neuroprediction, they're just intruding their metaphysics and assuming that Jill still has a non-physical mind that's somehow able to, maybe it can be predicted based on her brain, but she still has some kind of downward causal control from her non-physical mind, or something is going on that people aren't getting. And there's some interesting results from my critics suggesting some evidence for this. I have no doubt that there are some people who are so committed to dualism and libertarianism that instead of saying it's impossible and it would show we don't have free will, they say, I guess it's possible, but we have free will anyway. But for the rest of us, and when I say the rest of us, I mean, if assuming most of you are naturalists about the mind, is there a way to actually make sense of this that does not lead to the illusionist answer? And that's what I want to try and suggest for the next 15, 20 minutes. So how do we make sense of this natural response? What will our post-Galilean theory look like? Well, I think it'll suggest that we have to hold on to this non-negotiable belief that we're conscious agents who make choices among options that we imagine being open in the future, that we consciously imagine, should I go to the session on free will or should I go to the session on mice brains and what's going on with the mice brain. I don't know sessions were competing with ours, but this one's way more interesting. But anyway, you had to prospect those possibilities. That's what we call it in psychology and neuroscience, as prospection, and even mice can do a little prospection, no doubt. So you're imagining these options and then you're choosing among them. That conscious process of imagination and evaluation of alternatives better play a crucial causal role in our decisions and actions. That is, bypassing must be false. But we're naturalists, so what that will mean is that the cognitive neuroscientists are going to find the neural systems that are responsible for those capacities, and they're going to discover how they play a causal role in our actions. And I'm going to suggest that if we use the interventionist theory of causation, we can get a clear sense of how that is going to happen. And what that will show, I suggest, is that using this invariant causation, which I'll explain, there's going to be no causal factor in the past, the distant past, and typically not even the near past, nor is there going to be a causal factor described at the lower level of, say, physics or biology or even neuroscience in many cases, that will be a more invariant cause, in the technical sense I'll describe, but more intuitively, there'll be nothing that better explains our actions and our future actions than our exercising those very capacities to consciously imagine, deliberate, decide, and plan. That is, even if naturalism is true, there'll be a way to make sense of our capacities playing a crucial causal role that better explains what we do than anything in the past or anything at the non-psychological level. And even in some cases, the non-conscious level. So let's see if we can make sense of that. We'll begin with an image inspired by Josh Green and Jonathan Cohen, those illusionists, in an influential article for the Law of Neuroscience Changes Nothing and Everything, in which they argue that neuroscience will illuminate why we lack free will by making those opaque problems of determinism and physicalism more vivid. They say neuroscience holds the promise of turning the black box of the mind into a transparent bottleneck. Your brain serves as the bottleneck of all the forces spread throughout the universe. This is the funnel of forces going in the backward light cone. And your brain is the bottleneck that brings all those causes together from your past that affect who you are and what you do. Moreover, this bottleneck contains the events that are intuitively most critical for moral and legal responsibility. So, one thing that happens in that bottleneck, largely presumably in the so-called default network, is the combination and recombination of possibilities in our imagination and evaluation of future possibilities, sort of in the way the branching neuron suggests about what we're thinking about the future. I'm going to suggest we turn this sentiment on its head to recognize why neither determinism nor physicalism entails that they're factors that causally control our decisions more than processes that are within our control. If we reimagine the causal bottleneck, nothing else in the expanding light cone of causal influences will be picked out as causally predicting or explaining your decisions, for instance, to come to this session, as well as you are imagining, as well as you are exercising those capacities, of course, implemented in the relevant processes in your brain to imagine future alternatives. Alternatives which typically you can actualize if you choose them, to select among them based on your values and goals accordingly. So you're exercising your capacities, selecting alternatives, explain are in some cases what are the cause of your actions, and better explains than your internal decision making. Picture all and use interventionist of causation. I think these are ideal for thinking about neuroscience and psychology. And what they tell you is trying to figure out whether X causes some other you consider what would happen if Y, what would happen if X and nothing else is different in various ways. You kind of wiggle the value of X and see what happens to the value of Y. There are interventions on the value while controlling other causal influences on Y. When you see what happens to on X and to the value of Y. This is that point where I wish I could see your face, see if you're following, but hopefully you are, and I can't. Sarah is. I'm still visible. Thumbs up, Sarah. Okay. The more technical things get, the more you worry that you're speaking to a blank void. Okay, so here's how Jim Woodward, one of the main proponents of this view, along with people like Judea Pearl, and then some philosophers at the bottom there who have used this to talk about issues in psychology and neuroscience. Woodward says, look, X causes Y if and only if there are background circumstances C such that if there were a single intervention that changes the value of X and no other variables, if that were to occur in C, the value of Y would change. So the interventionist theory of causation is what scientists use to test and make causal claims. For instance, neuroscientists use it when they're measuring alterations in specific neural processes, MRI, causing specific alterations in specific using transcranial magnetic sorry, I'm getting weird up on my screen. I got it. Okay.
Eddie,
so
the audio is pretty messed up. I don't know if there's anything you can do about it.
It might be because my Wi-Fi is suddenly slowing down. How does it sound now?
Yeah, not great to me. Yeah. I don't know whether turning off your camera can like free up bandwidth or something, but
let me close every window I can close. And I'll turn off my camera. Okay. So I can't see you now. Why is this not? I'm trying to. Okay, there.
I think it sounds a little bit better. So we'll just keep our cameras off as you're talking. All right.
All right. And Sarah, chime in if it gets really bad. But let's see.
Okay, I'll do that.
Okay. So right at the most complicated part of the talk, of course. But let's see if you can follow what. Luckily, I have lots of words on my slides for a moment. But here's the important point I want to make, that we can use this picture to talk about relative causal importance or influence or control. And here's how I think we should do it. So consider two genes, and you can consider two neural processes if you want as well, W and X. And we're trying to figure out their influence on a phenotype P, okay? Or a behavior P if you're thinking of neural processes. And here's how I think we can measure relative causal strength or causal invariance. This looks complicated, but I think it should be pretty intuitive. So one, we can measure what people call specificity. So you hold fixed the relevant background conditions, like the rest of the organism's genome and environment. And you wiggle or intervene on the value of one gene, W, or on the other gene, X, and you see which of them have specific output changes on the phenotype P. right? So it's kind of like a volume controller on a radio. It specifically changes the value of the volume. So which of the genes that we wiggle has that more specific effect on the outcome? The other important thing to figuring out causal influence or control is what people call stability. So here, you're holding fixed the gene W or X, right? And you're measuring how much the relationship between those genes and the output phenotype P remains stable when you change various background conditions, when you change other genes in the organism or change the environment. And the more stable the relationship, the more W is a stronger cause or better controls or better explains the phenotype than X. Similarly with neuroscientific or neural processes. Varying other processes in the brain or varying the organism's environment, which process better explains the behavior. All right, so now we go back to pictures and, oh shoot. I'm trying to see if it'll be okay with my video on, just because it's weird for me to see nothing but my screen. But if my voice is not working well, let me know. So here's how I want to use intervention to explain why you or me can be the causal source of our actions, even if first determinism is true, and then if physicalism is true. So let's begin with determinism. Luckily, Adina introduced us to the definition of determinism. It says, look, you're in a closed system, a complete state of the system at one time. For instance, here in the distant past, say 30 years ago or 10 years ago or whatever you want to pick, 5 seconds ago, along with the laws that govern that system, they logically entail what happens here at this moment at time t 2020 right now. So that's what determinism says. Here's what I want to emphasize. Even if determinism is true, oh no, my screen's not moving. Uh-oh. Sarah, can you still hear me?
Yeah, I can still hear you. I was going to say maybe try stop share and then share again. If
I'm trying.
There you go.
Okay. What I want to suggest is that determinism does not entail that the entire light cone of events, or more importantly, any particular event within the light cone, such as event M back here 10 years ago or 5 seconds ago perhaps, is going to be a more invariant or more important cause of your behavior than events that happen within your brain. Specifically, events that happen, say, while that carry out the process of your imagining future possible decisions you could make and then making a decision at time t. Okay? And why is that? Well, it's because if you pick out one of these events in the distant past, like M, and you wiggle it, it's not going to have a specific impact on your action, because there's too many other causal events that play a role to specifically wiggle the value of M while holding everything else fixed and seeing a particular outcome. Similarly, it's not a very stable relationship. If you hold fixed M but change all other or various other features in the background conditions, there's no way that your decision is going to stably be maintained. Conversely, if you wiggle the value of D, your decision, holding fixed other variables, it's going to have a specific effect on the outcome. If you decided to go to the mouse session instead of the free will session, that decision will probably change where you ended up on Zoom. And if you hold fixed your decision to come to the free will session, it's going to be pretty stable, because one thing about intentions and plans is they try to get the outcome that they're aimed at across variations of background conditions. So even though Zoom messes up, or you have a hard time like Adina did finding the Zoom link, your plan to be here is going to make sure you get here across a wide range of obstacles or changes to the background conditions. So all of that is to suggest that the variable D here has a more stable and more invariant relationship with your behavior than anything in the backward light cone, at least for many cases of decision making. I'm not going to have time to go through it here, but I use this along with Oshin Giri to suggest that the manipulation argument against free will, which is probably the strongest current argument that determinism threatens free will, can be answered. I'll just tell you in case you're interested, we can come back to it if we have time, but that argument says, look, if you're manipulated in the distant past so that you will do a particular thing, then you're doing it. X is not free. And there's no difference between manipulation and determinism. So determinism rules it out. But we argue there's a big difference. The difference is M, the manipulator, is a stable and direct cause of your action in a way that the distant light cone is not in determinism. So if you wiggle the value of M, the manipulator's intention, it's going to change the value of your action because they controlled your action. But if you wiggle some value in the deterministic light cone, it won't be like that. So I can talk about that more in a second. Oh, I meant for you to be impressed by this. So now we're going to turn determinism on its head to talk about physicalism. And we're basically going to try to see whether or not the same sort of point can be made about levels of explanation that we made about the backward light cone of events. So what I want to suggest is that we're shifting now from the temporal dimension with the potential bypassing threat of determinism, which I hope I've suggested does not entail bypassing, because as long as we're naturalists, those events in your brain are crucial causes, in fact, often the most important cause of your action. Now we're shifting to a metaphysical picture that talks about a physically layered view of reality, and we're trying to answer a potential bypassing threat from below rather than from behind. But I want to suggest that if we reject the dualist metaphysics that the illusionists suggest is threatening, and we should, and we instead assume that there's this post-Galilean theory that explains how the relevant causal processes are realized in neural processes, then we can see that those processes in the bottleneck of the brain, like conscious imagining, evaluation, and deciding, are the crucial causal interventions that are most important for our actions. So how do we see that? Well, in the same way that we saw there was no causal variable in the past that's a more invariant cause of your decision than the psychological variables occurring just before the decision, in many cases, there's going to be no causal variable at, say, the neural level, certainly not at the biological level if you go lower on the scale here, or the chemical or the quantum level. There's going to be no variable that better explains your decision than the one at the relatively high level, like psychology and perhaps even consciousness. Why? Well, because the psychological variable is the one that if you alter it, while holding fixed all the other factors, will alter the action. And if you hold everything else fixed, and if you hold it fixed, it will cause the relevant action across a wide variation of background conditions, even varying some of the stuff at the lower level. Why is that? Because you can vary things at the lower level without varying things at the higher level. It doesn't matter if a few neurons change. In some cases, you may still hold fixed my decision to give this talk. Of course, some cases are going to be such that you change the neurons enough that you change my decision. But of course, those are the very things that we would think are the variations that allow us to see what happens when we change the decision at the higher level. If you change the quantum states of a few neurons or even several, that's not going to change my action. So the fact that there's a little bit of slip between the lower levels and the upper levels actually shows that the higher levels can be more important causal variables. If that notion isn't quite intuitive, just look at the picture and imagine, recognize now that I'm suggesting these ovals at the lower levels are the multiply realizing states of the higher level. So it's not that these three ovals at the cellular level are all required for this upper level. What this diagram is meant to represent is a realization relation, as it says on the right here. And this cellular level could realize my decision. It could realize the neural states that allow it, but so could this slightly different cellular arrangement. So what that shows is I could vary the cellular level slightly without varying my decision. And that means that there's a tighter causal relationship between my decision and the action than there is between these events at the cellular level and my action. That is, my psychological state, in some cases conscious, not always, has a more important, more causally sourcehood relationship with my action than the neural states sometimes, certainly than the cellular states, certainly than the lower level quantum states. So all of that is to suggest a picture whereby even though physicalism is true, everything is governed by physical laws, everything at the psychological level is composed by things at the neural level and ultimately by things at the chemical level. That's consistent with saying that the higher level can be more important. And this is a view that other people have made in similar ways, like Christian List and John Campbell. But I think intuitively, or hopefully, it can capture some of your intuitions that you expressed when you were responding to the neuroprediction scenario. Okay, with my, I have no time left, so I just want to point out that many of you who are studying neuroscience probably recognize that this is what's going on in some of your experiments anyway. I won't go through these 3 experiments in any detail, but the idea is quite often in cognitive neuroscience, you pick out the neural level by picking out the psychological variable that you're looking for, and then you recognize that you're not looking at the neural level at an incredibly fine-grained level, depending on what kind of experiment you're doing. You're recognizing that there are processes at the neural level, patterns that you're looking for. If you're doing fMRI, voxel patterns that presumably don't have to be specified at the micro level in order for the experiments to work. So in these experiments done by Kaufman and Churchland, they're looking at monkey decisions, but they're picking out the neural states using microarrays that predict the monkey's decisions based on the monkey's behavior when they decide to press certain buttons. Sarah, you still give me a thumbs up if I'm...
Yeah, we're good. I just want to remind you that we need to wrap up so we have time for questions.
I won't even talk about the next 2 experiments. People can ask me about them if they want. I will just leave it there and listen to your questions. I hope some of this made some sense for you, even though I can't see your heads nodding or your eyes glazing over if it didn't. I'll just stop sharing and only share if I have to bring it back up and listen to your questions now. Thank you so much.
Great, thank you so much. I'm going to jump right in because I know we need to be finished up in about 8 minutes, but we have some great questions in here. The first one, wouldn't the illusionist conception entail the abolishment not only of free will, but also of the self? What is the illusionist idea of the self?
Good question. I don't want to harsh on the illusionists too much, but I don't think they always have a very clear conception of what they're talking about when we talk about philosophical concepts like free will or the self. But, Sam Harris, for instance, also thinks Buddhism is right, and that makes sense because Buddhism has a sort of no self view. So these questions about free will and the self are tightly interlinked, and I certainly don't want to suggest that the picture I've presented suggests there's anything like the Cartesian view of the self as a robust entity inside the brain or attached to the brain in some way. But I think there is something like the self that is tied to our memory systems and our conscious systems that allow us to recognize who we are over time in the backward direction and in the forward direction, such that our plans, which are a part of ourself, and our values have this forward-looking impact on what we end up doing. So the illusionists might say that all those things are illusory too, to the extent they think consciousness does not play a role in our future actions.
Great, thank you. Here's another question. What I'm wondering about the bottleneck figure is that there are previous events that are incredibly influential for your future decisions, and others are obviously not, or maybe it's a combination of both. But let's say the events alter your physiology, then future processes will be influenced or not even possible anymore based on previous or lower level phenomenon. How does this fit into the model, thinking about brain tumors or other kinds of damage?
Right, very good question. The simplest answer is there are prior causes that may be the causal source of some of our actions. There may be cases where, the number of people around me in a crowd is the best causal explanation for my not helping somebody in need. Or there may be a brain tumor that affects my behavior more than anything in my conscious deliberations. If that's the case, the nice thing about this model is it gives us a picture, gives us an explanation for precisely when our free will might be limited or non-existent. And those are cases where it might be because my conscious deliberation process or my values or whatever might not play the right sort of causal role on my actions than that it needs to in order for me to be in control of my actions. It can happen to degrees on this picture. So it may be that my free will is limited in the case of there being more people around, whereas it may be that my free will is completely ruled out if I have a tumor, like the famous case that turns me into having pedophilial desires that I act on, like happened in the poor guy who had a tumor. That one's tricky too, though, because we don't know whether the tumor just released desires he already had or caused him to have new desires he didn't even have. But on both models, the idea is there may be causes in our distant past or near past that are very significant, and so significant that they limit free will. All of that is consistent with saying, look, of course I'm a product of my genes, and there are genes in my past that are crucial causes to my being the kind of person I am. But we should have never thought that wasn't true. Even on a dualist picture, that's going to be true. So it's just got to be the case that none of those genes are such crucial causes on my specific decision that they outweigh what I'm doing in here. Do we have time for a couple more questions?
Yeah, that'd be great. So, and just so people know, the panelists aren't able to somehow put questions into the question system. So I'm going to ask one from Adina. If we could design an experimental manipulation that affected behavior but didn't affect conscious reasons, would that be a way to show that there's no free will? Or would we just chalk it up to an implicit bias or something like that? Or maybe the broader question is, what would count as a defeater?
Yeah, that would, I think, in accord with my prior answer. I think if you can go in and you can manipulate someone's brain in such a way, I mean, this is what manipulation arguments are scary, are a bit scary about. If you can manipulate my brain in a way that bypasses my conscious reasoning and make me behave in a certain way, and you do it, then I think you will have taken away my freedom regarding that particular action. And it gets much trickier if you do it in a way that goes through my conscious reasoning, right? And if you do that, then I would say if you're really in control, that is, you have that kind of sensitivity over what I decide and stability over controlling what I do across a variation of other conditions, then even if you go through my conscious reasoning, in that case, you have overridden my free will. You're now the causal source of my actions. Natural causes typically don't work that way. That is, natural causes typically don't work by sensitively controlling which way I decide across background, holding fixed background conditions, or making sure that I come to this Zoom session no matter what obstacles are put in my way. Only intentional manipulators typically have that kind of power. So if you did it subliminally, it would be like other subliminal cases where I might lose conscious control and free will. If you did it through my conscious will and my conscious processes, you might override my own control. I hope that answers the question, Adina.
Yeah, that's helpful. So here's one that might try to bring together the talks a little bit. Can the idea of causal stability to wiggles in the fine neural details be tested? Does it, for instance, go against the idea that large neural circuits are borderline chaotic?
There's a lot going on there. I'm not sure I'm going to get it right what the person's asking, but I maybe Adina can help here, but my thought is the more complicated the neural process that is required for certain, say, complicated behaviors, like, say, writing a philosophy paper or doing a neuroscience experiment, right? I mean, imagine trying to map that in the brain as you're deciding how to do things. The more complicated it is, the less likely you're going to find specific neural processes that you can wiggle that will control the outcome of that process. More often, you're going to have to look to a more global system in order to control the whole process or control multiple parts of the process in order to control the outcomes in a stable and sensitive way. I don't think that means the systems are chaotic necessarily. In fact, in some cases, they're meant to be anti-chaotic, right? They're meant to be our brain having duplication of systems in certain cases so that chaotic processes don't throw us off if we're trying to make sure our hand continues on the right trajectory or our plans get carried out. But that's also consistent with there being some chaotic cases where a very small change might be the thing that throws the decision one way or the other. I hope that kind of answers the question. I don't know if that was what you're asking.
I think that helps, and I realize we're done with our time, even though there are more questions in there. Thank you to both of our speakers for great talks, and thanks to the audience for putting up the questions.
I haven't looked at the Q&A, so I hope it won't disappear immediately. I will be happy, to try to answer questions if I didn't get to one of you. But thanks so much for having us.
Similarly, I guess if your question wasn't answered, send me an email if it was for me, and I'll try to respond to it. And thanks, Sarah and Eddie.
Yeah, thanks. And I'm not sure if we can just keep going with.
We don't get forced out. I'd love to answer this one question from my own student, David Simpson, who asks, come on, how can you think people are theory light when they believe in the afterlife? So that's a great question. And a quick answer is, I actually think people are pretty theory light about the afterlife, those who believe in it. And to the extent they believe in it, they tend to think of it more in a physicalist way than they do in a truly Cartesian dualist way. They don't think that when we go to heaven, we're disembodied souls who can't recognize grandma. They think we have quasi-physical bodies such that we can see and talk with grandma. And I think there's a way to read that as, look, God is all powerful. So God can take our physical being. I mean, to be clear, Jesus rose bodily from the dead, not non-physically. So he might have had a more perfect body, and the same might be true of us if we go to heaven, but we still have something like a bodily form. And I think that's a pretty theory-like conception. We don't really know how that would work, but it would still require something like having the kinds of minds we have. And I don't know, as Richard is asking, if they would involve neurons or something that's the functional equivalent of neurons. But this view is meant to be consistent with a functionalist picture. Functionalism is theory light, actually, because it says we don't know about the underlying metaphysics.
It's interesting, actually, if we're allowed to just continue talking. You know, so I really thought it was elegant the way you flipped the cone and went from determinism to multiple realizability. And it's interesting that multiple realizability has been thought to be a challenge to mental causation, because in a way, I think it's exactly in multiple realizability that you get the ability to distinguish, through interventionism between distal causes and, the more invariant potent causes, right? If it was just a one-to-one mapping all the way up, then anything that happened in the past would affect what you do now. And so without multiple realizability, I think there's a problem with mental causation. And I guess I'd never really thought about it that way before.
Yeah.
I also, I mean, the first explicit argument to do that, I think, was Yablo, to talk about the realizers.
Determinables and determinates.
Yeah.
So there's a good tradition going back to Yablo, and then a lot of other functionalists responding to Jaegwon Kim, who put forward this kind of worry. But I think the best are John Campbell and Christian List most recently, and Peter Menzies with List. But I think, they give a way of recognizing that, look, if you think of causation, maybe in a sort of theory light way, and you just think what matters is what can you intervene to make a difference? Well, it's going to turn out that the most reliable things you intervene on are going to be at the higher level. And that's true in biology, right? It's not like if you're doing CRISPR, I meant to mention CRISPR technology, right? It's not like when you do CRISPR technology, you're targeting quantum states or even individual atoms. You're targeting genes. You don't care what the genes are realized by, what specific molecules. Of course, they're realized by atoms and molecules, but you're manipulating the molecules in the DNA, little bits of it. And you're taking out one gene and putting in another and seeing what happens at the phenotypic level. Similarly, I think if you were doing that at the neuroscientific level, you would be doing systems if it were a big complicated thing, and you would have to do a lot of work. Or if it were not so complicated, you'd still be looking at the neural processes, not the quantum processes or the cell processes. I can't see the questions. So Sarah, I don't know if we're going to keep going or if there out there anymore.
It says there are 28 people out there, but
28 people.
I see a question.
Yeah, there's one more. Can you see it now, Eddie?
Yeah,
or
but it's really hard to understand. Modification of the program. Yeah, I'm not going to be able to answer it well because there's several questions and I can't quite get the idea. But to the last thing about remodeling the free will definition, I think the answer is yes, but not as much as people like the Wolusionists think. I mean, it's not what we thought it was, because we did think it was something in a non-physical soul until we started thinking that our souls were in our brains. So, you know, pre-Copernican times, you know, you tell somebody the sun goes around the earth and people were like, what are you talking about? That doesn't even make sense. But now you tell a 3-year-old kid, hey, turns out we're spinning and the sun is still. They might say like, it doesn't look that way, but it doesn't take long. Maybe they have to be 5, but it doesn't take long for them to get the Galilean picture. I've done it with my kids. They're like, oh yeah, that's kind of cool. It's like when we're on an airplane and you don't feel ourselves moving. So we're moving really fast. So the post-Galilean neuroscientific picture will be like that. It will have to be like, it doesn't feel like your soul or your conscious mind is in these things inside the head, but here's how it works. We're not there yet. We don't have it. Even fancy theories like IIT of consciousness, they don't really get me there, and I you know, want to be there. I want to get to the post-Galilean stage, and I'm not there yet. So, but I don't think it's off limits. It might require revising some of our views for sure. And then, of course, I don't want to end by not agreeing with the point that Adina was raising in some of the questioners. There's all kinds of empirical threats that might be discovered. So nothing about this picture is meant to shield free will from empirical discoveries. Quite the contrary. There might be all kinds of things in neuroscience and psychology that show we don't have as much free will as we thought.
Well, can I follow up on my question and your answer? So I thought your answer was great, but I worry that the fact that whenever we find something like, you know, an implicit bias or something, we say, oh, okay, well, that's just a bias, or, you know, it makes free will in this case not the likely interpretation. But every other, you know, all these other cases, is there anything that we could do to convince ourselves under this picture that there is no free will?
Yeah, I think, but the only thing I can think of as a universal threat on this picture is if a very bizarre view was true, one that some of the Wolusionists present as likely, which is that there's some part of our, say, frontal cortex that receives all the information that's responsible for consciousness, and it doesn't have any causal impact on our actions. Or if it does, it's
right.
Really, you know, it just like, it's what we use then to make up stories, like Haidt's rationalization view for why we're doing what we're doing. But so it would be like universal rationalization. That would be a threat on this view. It would be a really weird way for the brain to be hooked up or to have evolved.
Yeah, or some demonstration that consciousness is never efficacious.
Yeah, but if it's natural, if on a naturalistic picture, that would mean there are a bunch of neurons in our brain that are responsible for consciousness
that do nothing.
That are one-way dead ends. That's crazy.
Right. I know, it's a crazy view.
I mean, the amount of energy our brain uses, evolution would have never allowed there to be a bunch of neurons that do nothing. It just wouldn't. It just couldn't happen. So I don't think that's going to happen. But we could definitely be rationalizers a lot more than we think we are. We know we are. We know we are. We rationalize all the time.
Yeah.
Are there any lingering questions? They're all answered.
Thank you, guys.
Thank you.
Thanks, Sarah.
Thanks, Sarah.
buddy.
Thanks to whoever's left. Thanks,
Yeah.
Adina. Good to see you, kind of.
Yeah, right.
I hope to see you all in person sooner rather than later.
Thank you, guys. Bye-bye.
Bye, everybody.
The whole time. Thanks for all the technical support.
We have a great lineup of speakers in this session, and we will learn about social behavior in animals, particularly the effects of tryptamine psychedelics on social behavior, the relationship between socializing and stress. So we will have really exciting talks. We can get started. Our first speaker is Tommy Markopoulos. He's a master's candidate at the Neurobiological Psychiatry Unit of McGill University, and he works with Professor Gabriela Gobi. His talk is titled LSD Increases Social Behavior and Bursting Activity in Medial Prefrontal Cortex Neurons. So the stage is yours, Tommy.
All right, I'll just share my screen. Thank you for that lovely introduction. Can everyone see my screen? Perfect. So I think I'd start with a brief history of LSD because I think it's quite fascinating the way this drug came to light in the world. So it was first synthesized in 1938 by a chemist named Albert Hofmann, who was working with lysergic acid derivatives in order to determine which ones can be used as a central nervous system stimulant. And then, LSD didn't serve the purpose he needed it to, so he disregarded. 5 years later, he was re-looking at some of the chemicals that he developed, and he accidentally administered to himself LSD through his fingertips. And this is the first recorded human trip of LSD. And he records his experience as having an extremely stimulated imagination, an uninterrupted stream of fantastic pictures and extraordinary shapes. And this took the world by shock. It was to discover such a potent hallucinogen was something fascinating, and its ability to modulate the psyche. So research focused until 1970 on how this drug can be used to treat things like anxiety, depression, addiction, as well at large doses as an acute model of psychosis. So its potent hallucinogenic effects took the world by storm, and this increased the recreational use of this drug. It became a very prominent drug that people would take for fun, essentially. And this ultimately led to the Controlled Substance Act in 1970, which ceased all the research being done with this drug as well as other hallucinogens. And to me, it's quite absurd that this classification took place because what's a Schedule I drug? It's a drug defined as something with no currently accepted medical use and a high potential for abuse. Which, if we look at all the science being done before this classification, it seemed like such a promising therapeutic drug. Slowly, research using psychedelics resurfaced in the world, and there was this renaissance in the 1990s. And research essentially continued and stemmed off the research that was done before the banning and the classification of these drugs as Schedule I. So some of the more famous and the pioneer research that took place included psilocybin and LSD. And in fact, what they found is that, this is the graph I'm presenting to the right, we see that a very early study in this field showed that psilocybin actually decreased anxiety scores. So if we look at this table, we see that the y-axis is anxiety levels, and we see that the black squares is the group that received psilocybin. And conversely, we see that the triangle line are people that received a placebo. But when this group was crossed over to take psilocybin, we see that their anxiety scores decreased. And in my opinion, what's most fascinating about this study is that they show that even 12 months following the administration of psilocybin is that this reduced anxiety level was maintained. So this really shows that a promising effect and therapeutic potential of this drug. Now, very quickly, I'd like to talk about the pharmacology of LSD. So serotonergic hallucinogens take their hallucinogenic effects via the serotonin receptor type 2A, and most specifically in the medial prefrontal cortex. In fact, they've developed drugs like DOI, which is a selective 5-HT2A agonist, and it's proven to be a very potent hallucinogen. Other receptors that it binds to are the dopamine type 2 receptor, as well as the trace amine associated receptor 1, so TAR1. And we see that it binds to these receptors all over the brain, which explains this very vast profile of behavioral effects. Now, very quickly, the medial prefrontal cortex is a very interconnected brain region. So it has a very diverse profile of afferent and efferent projections. It's very implicated in all sorts of behaviors, but most notably social cognition. And in fact, we see in individuals suffering from autism spectrum disorder, we see that they often show disrupted neural activity in the medial prefrontal cortex, which again links back the implication of the medial prefrontal cortex in having regular social behavior. Now, my research focuses on identifying the robust behavioral effects of LSD in mice. So we seek to determine, and the reason it's nice using mice is because it allows us to use this wide variety of established behavioral assays to look at many different things like anxiety or sociability. But the benefit of also using mice is that we can use more invasive procedures that allow us to look at the neural activity and what is actually happening at a biomolecular level. So my research also seeks to understand the mechanism of action of LSD, as well as which neural circuits are implicated. So the first test we performed for this study was the direct social interaction test. And this is a test performed by first putting the test mouse in an empty cage, letting it habituate for 10 minutes, and then putting an intruder mouse inside. And what I'm measuring is how long the test mouse spends interacting with the intruder mouse. And so on this graph here is the results of the first pilot study we did with LSD. And what we found is that 30 minutes or 24 hours post-injection of LSD, that this had absolutely no effect on the interaction time. So then we decided to see if repeated administration of LSD would create a change in social behavior. And this would make sense if LSD perhaps requires some neuroplasticity in order to take effect. So the methodology we used was to give a dose of 30 microgram per kilogram per day for 7 days. And then on the 8th day, we performed behavioral assays. And I'll explain the justification of the dose shortly. But what we first found is that this repeated administration of LSD was actually able to increase the interaction time in the direct social interaction test. And I think it's quite powerful to actually see this in action as opposed to just a graph. So to the left here, we see the direct social interaction test in vehicle-treated mice. So these are mice that did not receive LSD. And so the mouse to the right is the test mouse, and we can identify the intruder mouse as the one having a piece of tape on its tail. So as we see, it does interact a little bit, but in general, it just spends a lot of time exploring the cage, but it still spends some time interacting with the other mouse because mice are inherently social beings. And I think the contrast is quite powerful looking at LSD-treated mice. So now the mouse most front, the right in front is the mouse that was treated with LSD. And then again, the mouse with a piece of tape on its tail is the intruder mouse. And what we see here is that the mouse spends significantly more time interacting, and it essentially spends most of the time following the intruder mouse and really spending an overwhelming time interacting with it. The next test we performed was another gold standard in assessing sociability in animals. So this was the 3-chamber sociability test. And the way this test is performed by first putting the test mouse in the 3 chambers apparatus and let it habituate. And then the first testing phase of this experiment is the sociability phase. And what I do here is I introduce an empty cage and a cage with a stranger mouse inside. And what we were assessing is the mouse's preference to interact with a social object. In other words, the cage with a mouse inside, as opposed to the empty cage. And mice naturally spend more time interacting with the cage with a mouse inside than the empty cage. And we see that with this diagram here. And the next stage, which we perform right after the sociability stage, is the social novelty part of this experiment. And in this case, we replace the empty cage with another cage with a novel mouse inside. And what we're looking for here is if the test mouse prefers to interact with a novel mouse rather than the familiar mouse. And we see here that mice not treated with anything just naturally prefer to interact with a novel mouse. And what this tells us is that mice naturally have a preference for social novelty. And then we administered the same treatment, so repeated LSD for 7 days. And we'll start all the way to the left. So what we see here is first that, so in black is the vehicle-treated mice. And what we see is, as expected, they interacted significantly more with the cage with a mouse inside than with the empty cage. However, when we treated them with LSD, we see that this preference for a social object became more robust. So they interacted significantly more with the stranger mouse in a cage than the empty cage. And this is a way to normalize this data, instead to put it in terms of a sociability index. And we see that this sociability index is greater in the LSD-treated mice. Now, in the social novelty phase, we see more or less the same thing. So we see that, again, mice naturally prefer the novel mouse than the familiar mouse, but we see that this preference is, again, more robust in LSD-treated mice. So the social novelty index, again, is greater in the LSD-treated mice. Next, we perform the open field test. And the reason it was important to do this test, maybe first I'll explain what it is. So essentially, we put the mouse in an open field for 20 minutes. And what we're looking for is exploratory behavior as well as its locomotion. And the reason it's important to test this, especially when dealing with psychedelics, is because, as I mentioned before, a large dose of LSD can be an acute model of psychosis. And psychosis is one of its main characterizations is having hyperlocomotion. So in order to determine that the dose we are giving isn't in fact a psychotic inducing dose, we would have to see that its locomotion and its exploratory behavior is not impaired or altered. And this confirms that. So we see that the distance traveled or the time in the center or the entries in the center of the open field are not significant. So there's virtually no difference between vehicle-treated mice and LSD-treated mice. So this confirms our dosing. So with all these sociability results, the next logical question became, what is the neural basis of these changes in behavior? And the way we assess this was by using in vivo electrophysiology. So we put an electrode inside the medial prefrontal cortex of the mouse, specifically in the infralimbic cortex, and we recorded the neural activity. So what we found is that acute LSD, which again didn't show any behavioral effects, did not change the spontaneous neural activity in the medial prefrontal cortex. Next, we tested this with repeated LSD. And similarly, we didn't find any change in the spontaneous neural activity, but we did find is an increased bursting activity. So there were more spikes per burst in medial prefrontal cortex neurons. And this has to do with perhaps the fundamental way that LSD takes its behavioral effects. And this is where our research is heading, is to understand why do we see this change in bursting activity, but not its spontaneous firing rate. Now, future directions of my research seem to focus on the characterization of LSD's mechanism of action at a molecular level. So which receptors are needed for LSD to produce these robust behavioral effects? Next, it's to extend our results to models of mental illnesses in mice. So there are a wide variety of mental illnesses that are characterized by impaired sociability, and these mental illnesses can be easily modeled using transgenic mice in mice. And so we seek to study this. And lastly, it's to perform more electrophysiological experiments in order to further understand exactly which neural circuits and which brain regions are fundamentally involved in these sociability promoting effects of LSD. So thank you, Ezgi, for emceeing this, and thank you everyone for listening.
Thank you, Tommy, for the wonderful talk. That was really interesting. We already have 3 questions, so I will start with the first one. What was the sex distribution of the mice within the experiment, and were any analysis conducted with regard to sex-specific differences?
So that's a great
that's from Rob Taylor. Yeah.
Thank you for the question. That's a fantastic question. And because this was more of a pilot study to first identify any behavioral effects of LSD, we only used male mice in the study. But using both female and male mice is absolutely something we envision doing in our lab, because I agree that using female mice really isn't the norm necessarily in a lot of science, which is very unfortunate because there are fundamental sex differences for pharmacological effects, behavioral effects. So I absolutely take the question is well taken, and I will absolutely direct future research in that regard.
Thank you. Thanks also, Rob, for the nice question. Second question is from Marta Havenit. Marta is asking, were there any aggressive interactions at all in the social interaction test? And if so, did you see any differences between groups?
So thank you for the question. In fact, we didn't see any hyper-aggressive behavior. I mean, mice in general do fight, especially when they're put in a novel environment and they're trying to gain this, this dominance for this new cage. but LSD really didn't have any significant effect that would make it something worth exploring. It really wasn't anything robust. But that's a great question, and especially interesting because some of the models we're looking to study, hyperaggression is something fundamental in that mental illness. So it will definitely be involved in the direction of this research.
Okay, thank you. Also, thanks for the question. Another question is from Jacob Cheeseman. If mice prefer novel social interactions and LSD increases sociability, why do mice not self-administer LSD?
Very interesting. So, I mean, I don't know if there's been studies using, I forget what the behavioral assay is, where they can self-administer whichever drug. But actually, some of the old research, some of the pre-1970 research showed that LSD can actually be anti-addictive. So they were using psychedelics to actually treat addictions to drugs of abuse, so things like nicotine and alcohol. So in terms of the act, perhaps the molecular or pharmacological effect, why this happens, I mean, I don't know why it's anti-addictive or why it's not addictive, but it's absolutely, I think, something that this field, a direction that this field is heading. It's to understand why this drug can be used to combat addictions.
Okay, thank you. So there is one more question, but I will go with the next talk to be just, on time. But you can answer the question by typing your answer on the Q&A section. So thanks again.
Thank you.
And we can now move on to our next speaker. Our second speaker is Sabah Shokat Fadei. She's a research associate at the Skirball Institute and is working with Robert Fremke. Her talk is titled Socializing Reduces Distress-Associated Behavior in Mice. So the stage is yours.
Can you hear me? Perfect. Hi, everyone. So today I want to talk about the project that I'm doing at Fremke Lab. My focus is socializing and social buffering and its effects on distress or distress-like behavior in mice. Before that, I want to tell you why I got interested in this project. Well, I was reading a couple of papers talking about how social buffering can be a benefactor for the physical health, mental health, and there was a couple of papers stating that the social buffering, even for a shorter time, will reduce the risk factor of mortality. And social isolation was a major factor for mortality in mice. For studying that and also inducing the depressive-like behavior in rodents, I chose LPS. LPS is a large molecule that has two components, which is it's a polysaccharide and the lipid part. And you can find it in the outer layer of gram-negative bacteria cell walls. It is a neuroinflammatory endotoxin, and it will release couple of inflammatory cytokines such as IL-8 and it will downstream pathway of kappa B. So for studying the effects of LPS on behavior, I utilized the system that one of our previous postdocs in our lab built it. So basically, I transferred my mice into the enclosure recording, and a couple hours before I start the recording, and then I start the recording 24 hours after that. I inject LPS or saline as my control through the IP, and 2 days after that, I end the recording. After recording, I analyze the videos using deep lab called DataView and ELEN. And then I had this code with the help of the lab that we basically made this ethogram for the behaviors that we could see. The interesting fact about LPS injected mice was first, we could see the hunching posture that increased for a bit abnormal time of after post-injection. And the other thing that we could see was that the inactivity level was increased when you compare the saline-injected behaviors with LPS1. For that, the next one that I wanted to see was to actually see so many aspects of LPS will have on the mice. Then I wanted to see if LPS will decrease the mortality. With that, I injected the saline and then tracked the mice. And I had 1 hour before injection and 1 hour after injection and compared it. But there was nothing, there wasn't anything in, detectable in locomotion. However, the locomotion reduced drastically post-injection after LPS. As you can see here, the mice after injection only spend time around the nest area or near the hydrogel. Then I wanted to see what is happening actually with the social buffering and LPS injection. As you can see here, the co-house mice, when I co-house them, the partner mice trying to shepherd the injected mice. But if you see here in the single house mice, it only goes to a corner and it shows the hunched posture. Then I had this heat map of the activity level in the single house mice and the co-house mice. And as you can, if you compare it each time that I had over here, the mice started showing the recovery symptoms such as activity level when they're co-house faster than their single house. Then I focus on behaviors and I want to see what the co-housing will bring up to the behavior. If you see here, there were so many behaviors but I put a couple of behaviors together as a group. Over here we have the maintenance behaviors such as feeding, grooming, drinking, nesting behavior all together, but LPS would induce a reduction in this type of behaviors. It wouldn't, the co-housing would not change anything. But then I went to the exploratory behaviors such as foraging, moving, and exploring the cage and sniffing around. There was an interesting result of this analysis, and it was that when the mice is single house, there was a statistically significant reduction when they're single house rather than when they're co-housed. And if you remember my my first slide of my ethogram, there was a hunching posture. It is really interesting, and I'm trying to see what is the neuromechanism underlying this this phenomenon. But when the mice is co-housed, they almost hunched and show the hunching posture for half of the time when they're single house. And with that, as you can see here, the saline injected mice did not show any hunching posture. That was interesting. Then I wanted to see the difference between co-house and single house mice when I injected LPS on them. And over here, we could see that the LPS injection will result in a reduction in weight, and the mice would lose their weight. But it's a preliminary data, but I found it interesting when I co-housed the female and the male mice. I could see that the male mice would not lose any weight. However, the female mice, even when I co-housed them, they would lose about 2 to 3 grams of their weight. In conclusion, I can say that LPS injection will result in the reduction of the duration of maintenance behavior and exploratory behaviors. However, it will increase the hunching posture behavior. But when I co-housed the mice, I could see that they starting to show the recovery symptoms faster than their single house. And the mice, it's a preliminary data, but I could see that when I co-house the mice, they try to engage in social behavior and social interaction much more when they're single house. And with that, I want to thank my team from Key Lab, my students, and Ludmila Lab, and also I'm here to answer my questions.
Thank you very much, Sabah. That was really nice. Very interesting talk. So let's see if we have questions. So right now we don't have, so feel free to post your questions on the Q&A section. I have a question.
Yeah.
So, I mean, it's also, I think, a bit related to what Tommy showed with LSD. So do you think in the injection of LPS might have induced a psychosis-like behavior so that the mice were really not showing any locomotion at all?
Well, they for sure they are in pain, and also they have fever because I took the temperature before and after the injection of LPS and saline. There was the LPS injected mice. The temperature was increased by 2 or 3 degrees, but saline ones didn't. And also, when I was studying the hunching posture, it was one of the abnormal behaviors, and it's one of the signs for that mice are in pain.
Okay. There is a question from Edwin Rodriguez. So Edwin is asking, is the co-housed non-LPS mouse a litter mate or a new mouse?
They're litter mice. All of them are litter mice.
Litter mice, okay. Mm-hmm.
Yeah.
And maybe to clarify, because I'm not in the field. So the hunching posture you observed, what does this indicate in mice?
It is it's an indication of that it's actually, it was listed as underlying abnormal behavior, and there's an indication of mortality actually. When I injected LPS, I usually stick to the specific dose, but if I injected too much of LPS, one of my mouse died actually while hunching.
Okay, thank you. We have a follow-up question from Edwin Rodriguez again. So Edwin asks, follow-up question is whether it makes a difference if the co-housed mouse is a litter mate as opposed to a new mouse.
Oh, that's an interesting question, because these days I'm trying to record from I'm trying to introduce new mice together and put them in an enclosure together from like other litter mice, and I think probably I can answer that question in a couple of months, I would say. But that was an interesting question, actually.
Thank you. Also, thanks, Edwin, for your questions. And I don't see any other question. Yeah, any other question that is coming?
Could I ask a question?
Sure.
Yeah.
Yeah, I guess a follow-up from the co-housing versus litter mates. Do you think it would, the benefits of co-housing would depend on the social hierarchies between mice? Because if the mouse is a subordinate in its litter group, or even with a new mouse, it would suffer further stress and kind of physical consequences of that relationship. And do you think like that would kind of reduce the benefit for the symptoms and the hunching?
That's an interesting question. I did not think about it, and I have to think about it. Well, one thing that I would say was that I got this mice as a litter mate, but they were separated at a specific time by the time of the weaning. So I put them like all together because I didn't want them to interact too much. But I can try that, and I will email you my my data later.
Thank you. Thanks for the questions, and thanks, Sabah, for the wonderful talk. So we can, I think, move on to the next speaker. So the final talk. The final talk is from Merima Shabanovic. Merima is a second-year PhD candidate at the Wellcome Trust Doctoral Training Program in Neuroscience at University of Oxford. And she's working with David Bannerman. Her talk is titled Differential Acute Behavioral Responses to, I would read that as plus minus DOI injected in a novel or familiar environment. And the stage is yours, Merima.
Thank you, Ezgi. Thank you for the introduction. Hello, everyone. In this talk, I will present the results from an experiment in which we manipulated the environmental context during administration of a psychedelic drug DOI that you heard mentioned briefly by Tommy in this talk, and I monitored the acute behavioral responses. First, I would like to start by giving you a brief background of why psychedelics are a fascinating new area of research. If we think about the goal of any therapy, it is to help the patient by providing quick and lasting. Traditional pharmacological treatments of mental disorders remain poorly effective as they take a long time to start showing effects. They require chronic administration associated with numerous adverse side effects. And even then, they often only just mask the symptoms and don't really cure the underlying issue. In contrast, the past decade has seen clinical studies of psychedelic drugs showing unparalleled therapeutic efficacy across a wide spectrum of neuropsychiatric disorders. Just a single dose can induce rapid and lasting relief from depression, anxiety, addiction, post-traumatic stress disorder, even in treatment-resistant populations where none of the standard medication was able to work. Plus, these drugs have a good safety and tolerability profile when administered in controlled settings. So what are these magic substances? Well, you may have heard of some serotonergic psychedelics before, such as natural-based psilocybin mushrooms and ayahuasca brew, or synthetic drugs like LSD or DOI. Now, DOI's human clinical use hasn't been established yet, but it is widely used in preclinical research, such as the one I'm presenting today. What all these drugs have in common is that their method of action relies on the neurotransmitter serotonin. And just as serotonin activates a whole host of serotonin receptors across the whole brain, so do serotonergic psychedelics. And the characteristic acute psychedelic effects, such as the visual and auditory changes, the non-ordinary state of consciousness, they're believed to be mainly mediated by the 2A receptor, since blocking or ablating these receptors completely abolishes the acute psychedelic effects of these drugs, both in humans and in animals. It has also been suggested that it is precisely the activation of these receptors that underlies the potent therapeutic effects of psychedelics. So Robin Carhart Harris and David Nutt suggested that psychedelic serotonin 2A receptor signaling hypothetically opens a window of enhanced plasticity and sensitivity to the environment, which in turn then lead to enduring increased learning and well-being. Now, importance of the environment for drug action is special for psychedelic drugs. These non-pharmacological determinants of psychedelic effects are called set and setting. So set is the mental state a person brings to the psychedelic experience, like thoughts, mood, and expectations, while setting is the physical and social environment in which this experience is occurring. And while these effects are reported extensively in humans, they've been mostly overlooked in animal models, even though, for example, using rodents allows us to precisely control that environment and personal history, which is not possible in humans. And because animal models remain critical for finding the mechanisms of action of these drugs, so we wanted to do is investigate whether the quality of the environment would also impact a mouse which has been administered with a psychedelic. To establish two different environmental contexts for drug treatment, we did not want to use strong and unnatural stressors, but instead we focused on familiarity of the environment, which is greatly emphasized in human clinical contexts. So one group of animals was habituated to a recording chamber for 5 days, while the second group of animals was not exposed to the recording chamber prior to the day of injection. And all animals underwent the same injection procedure, being allowed 5 minutes of free exploration before being given a single injection of DOI, after which there was 30 minutes more allowed for free exploration. And finally, I introduced 2 novel objects in the final 15 minutes of the recording. Only male C57 mice were used in the study. And I first wanted to confirm we established familiarity by doing this habituation paradigm. So I looked at the total distance traveled during the 5 habituation days, focusing on the initial 5 minutes when the animal's exploration levels would be the highest. And distance traveled during that time starts decreasing by day 2 and is significantly lower by day 3 and throughout the remaining habituation days. Next, I also wanted to confirm that habituated mice were still able to recognize the familiarity of the chamber on the day of the injection. So I compared the distances traveled in that 5 minutes before injection. And indeed, again, habituated mice were still able to recognize the recording chamber as their distance traveled was much lower than that of non-habituated animals to whom the chamber was novel. And so this confirmed that this difference between the environmental context was evident to the animals at the time of injection. Next, I investigated whether injection context familiarity would modulate the acute psychedelic responses. So in mice, this involves head twitches, which are rotational side-to-side movements. So it's a very quick behavior, but here slow down, you can kind of see the mouse shaking its little head. And DOI also induces an ear scratch response, which is pretty self-explanatory. So a mouse would scratch its ears with front or hind paws. And here you actually saw a combination of both twitches and ear scratches. Slow down again so you can see better. The mouse will do a little scratch, another scratch, a little twitch, and then another scratch. So I quantified these behaviors manually in the 30 minutes after injection, and the total number of head twitches observed increased dose dependently compared to the vehicle injection controls in both injection contexts. However, there was a significantly higher head twitch response in non-habituated mice only at this high 2 mg/kg dose of DOI. And similarly, the total number of ear scratches also displayed dose-dependent increases with some additional dependence on context. So the total number of ear scratches for non-habituated mice was again significantly higher than that for habituated animals at the same 2 mg/kg dose. And what this signals is that the novelty of the injection context can indeed affect behavioral sensitivity to DOI, and this is more likely at higher doses. Finally, to investigate whether DOI affects animals' ability to interact with changes in their environment, I placed 2 novel objects in the recording chambers 30 minutes after injection and recorded the animal's behavior for about 15 minutes. Comparing the total percentage of time that mice spent interacting with objects, interestingly, non-habituated animals that only received the saline injections actually interacted with the object significantly lower proportion of time, but this difference was not evident under any DOI dose tested. And this is possible because the non-habituated animals were busy exploring the novel objects as well as the still novel environment, while the habituated animals were completely focused on the novel objects. But what was interesting is that the habituated animals displayed a significant reduction of object interaction times, again, only under that high 2 mg dose. And what this could be a sign, we think, is that the animal's attention has been shifted from studying its environment and externally generated stimuli to studying its internal state and mind-generated stimuli. In summary, we show that mice could distinguish between contexts both before and after drug injection, and that behavioral responses to DOI differed between the 2 drug injection contexts. What this serves is an important validation of the set and setting model of psychedelic drug action in mice. And I would just like to note that additionally, day after the drug injection, a subset of animals underwent brain perfusion for a subsequent ex vivo magnetic resonance imaging to study any putative changes in brain structure. And this analysis is ongoing, so I hope that I can talk about it some more in the near future. Finally, I would just like to use this time to let you know what our future plans are. So if you remember the window of plasticity model that I showed earlier, the second branch of that proposed model was enhanced neuroplasticity. And what neuroplasticity is the ability of the brain to change in response to experience. And this occurs at various levels. So at a molecular or a cellular level, it refers to changes in numbers of neurites and synapses. On a larger scale, it refers to structural plasticity, such as regional volume changes in the brain, or functional plasticity, referring to the organization and function of neural networks. And on the highest levels of cognition and behavior, enhanced plasticity would refer to increased cognitive flexibility and greater ability to adapt to a changing environment. Now, there's a novel integrative model that has been suggested, which predicts that if a drug were to promote molecular neuroplasticity, there should be correlated and simultaneous facilitation of all these higher levels of plasticity as well. Now, psychedelics have been found to rapidly promote functional and structural plasticity by promoting the growth of new neurites and new synapses, both in vitro and in vivo, hours after the drug injection. So this is after the drug has cleared from the system. And what I would like to know is understand how this might ultimately translate to that profound shift in behavior that's reported in clinical studies. So we propose looking at plasticity markers at the higher levels of analysis, brain structure and cognitive function, and how these are modulated by psychedelics in the long term after the drug has worn off. And we hope that this would elucidate how psychedelics can induce such fast and lasting effects on behavior. And I'd be happy to talk about this more and hear your feedback during the Q&A time. But for now, I would just like to thank the supervisors of this work and other members of the lab that have helped me along. I would also need to thank the Wellcome Institute and the Clarendon Trust for funding this work and funding me. Thank you to the organizers and for your attention, and I'm happy to take any of your questions or comments.
Thank you, Merima. That was a really nice talk. Thanks a lot. So do we have any questions? So there is one from Tommy. He's asking if DOI does in fact shift attention to an internal gaze, wouldn't you expect to find a reduced time exploring the objects in both groups and not just the familiar group?
Yeah, so I think the problem, if I just quickly go back to that slide, the thing is that because we have that reduced exploration of the objects in the non-habituated group, regardless of DOI, there were no further considerable drops in that exploration time with the addition of DOI, probably due to a floor effect because of the size of the arena and the places where objects were put. You couldn't necessarily avoid the objects. So that would explain the floor effect here in the non-habituated group. But yes, that is a fair point. I think the effect is more visible in the habituated group because they already start at high levels without the drug.
Thank you. We have two more questions. So the first one is from Rob Taylor. Is there any work done to correlate the behavioral effects of DOI to more classical psychedelics used in human clinical studies?
So like I mentioned briefly in my slide about the mechanism, because they all act via the same kind of serotonergic pathway, activating relatively the same family of receptors. And the difference really is just kind of the affinity of these drugs for different receptors is what's different. But their overall pathway and mechanism of action is quite common along a lot of these drugs. So it would be common with psilocybin or with LSD that have kind of established clinical effects in humans as well. I think the thing with DOI, it hasn't been that much used even recreationally in humans. So that's why it's kind of lagging in clinical studies as well. It is preferred because of its higher affinity for the 2A receptors. So I think that in time, it will kind of reach more, it will get more attention.
Thank you. And thanks, Rob, for the question. So the last question is from Jacob Cheeseman. Jacob is asking, can you speculate about the possible physiological mechanisms underlying these context-sensitive behavioral effects?
Yeah, I mean, it could be anything, right? Because the drug affects the whole of the brain and it's injected IP, so it reaches other parts of the body as well. So there are kind of physiological changes that are occurring in the body and then also activation across the whole of the brain that occurs as well. So there's really so many places to look at, but it could affect kind of the speed at which the drug is metabolizing, for example, and purely from a behavioral perspective, it could be that it's just affecting these attentional resources and how these are distributed, and that's why, you know, those acute behavioral responses were different. But again, it would be very difficult to narrow down that search. Good question.
Thank you. Yeah, so I think we came to the end of our session. I would like to thank again to the speakers for the wonderful talks. It was really nice. And also the attendees for their excellent questions and engagement. And I would also like to thank our backend Reka Verrier, who's been doing all the technical work. And I hope you enjoy the rest of the NMC. And have a nice day or evening or night.
And welcome to this NeuroMatch session. We have 3 great talks today from 3 students actually. And since this is an interactive session, we expect to have a great discussion as well. So for this interactive session, the format is 5 minutes of introduction and 10 minutes of questions. So I encourage all of you to keep the discussion alive. The first talk is by Soo-Jin Kim. She is a research technician in the lab of Benjamin Scott at Boston University, and she's going to tell us about an implantable kinematic head plate for submicron skull positioning in mice. And I think this is a super interesting new technology, especially for people who are doing developmental studies, and if they also want to register the same subject across multiple instruments. So take it away, Sue.
Hi, everyone. I'm Sue. As Lena said, I'm here to talk to you about my device. So let's get started. So the motivation for this project was to address challenges in head restraint for in vivo experiments, specifically for imaging experiments. Some of the cons of this head immobilization setup are that it stresses out the animal, which can cause confounds in your behavioral data, and it requires experimental oversight. One solution for this is head-mounted microscopes or miniaturized head-mounted microscopes where you can image the brain directly, but those head-mounted microscopes have some drawbacks. They don't perform as well as traditional tabletop microscopes do. Another solution for this is voluntary head restraint. Voluntary head restraint was first pioneered by a researcher named Sergei Gurman in the 80s, actually in Soviet Russia, who was trying to examine visual cortex in rats. Since then, it's had a resurgence in neuroscience experiments, especially demonstrated by work by Tim Murphy's lab at University of British Columbia, but also Ben Silvesky's lab at Harvard, and Carlos Brody and David Tank's lab at Princeton University. Specifically at the Tank and Brody labs, they developed a voluntary head fixation method using pneumatic clamps and kinematic mating principles. On the left, you can see the head implant that goes on the rat's head. There are 2 kinematic depressions on either side of the implant that mate with ball bearings attached to pneumatic pistons. On the bottom panel, you can see what this may look like. There's a head plate that is inserted into the clamp. The insertion is detected, the pneumatic pistons are activated, and it clamps onto the head implant. After the image is taken of the animal's brain, it's then released so that the animal can leave. You can see this also in this video of a rat voluntary head fixing. You see the objective go down, it takes an image of its brain, and then the objective is raised, and the animal can leave the apparatus and go back in as it wants to. So with this in mind, we wanted to apply these existing principles and then extend it to now mice, because mice are more ubiquitously used in research experiments. Obviously, we want it to be biocompatible. We want it to be light enough for a mouse to, behave normally and not be like tilting and stuff. We want it to be able to be fabricated in a standard machine shop, and we wanted to achieve submicron registration accuracy. Submicron registration accuracy is because of the existing challenges in multiphoton imaging correction. The work from David Tank and Carlos Brody's lab achieved micron scale registration accuracy, but this Z motion can still produce large fluctuations in observed fluorescence intensity, and this can interfere with your functional calcium signals that you're measuring. So the design features that we wanted to incorporate were kinematic features like the Maxwell clamp, and we wanted to use the same idea as the Tank and Brody lab where we have spheres as the contact points. We also wanted it to be magnetically driven so that it's easy to connect the animal to the device. So with that, this is a CAD schematic of what this may look like. There are the cylindrical things on the top are magnets, which are placed behind the kinematic features that the head implant will mate with. The head plate on the bottom is the animal side of this device. So the methods that we used are as follows. Basically, I have my head plate. I affixed a fluorescent sample to it, and I took my head plate out of the clamp, reinserted it, took an image with my 2-photon, removed the head plate, and put it back and took another image. And I did this 100 times. Because we're talking about the scale of, sub-micron, hundreds of nanometers, I had to use super resolution techniques to estimate the position of these fluorescent samples. Here's some data. So initially we had a head implant that had spheres milled onto it. So this is all the same part. The spheres are not glued on, for example, to the head plate. And you can see that for the displacement or the change in position across successive images, that the vertical root mean square of displacement is about 400 nanometers and the horizontal is about 460 nanometers. And so this is pretty good, but we thought, if this sphere that is our contact point is machined onto this part, then it's likely that there are microscopic aberrations on the surface of it due to the machining. And if we can remove those aberrations, then it's possible to get even better registration accuracy. So I took scanning electron microscope images of the spheres that are attached to my head implant. And you can see here that there are these tracks from the machining process. And so our next step was, well, if this is true, then if we wanted to use something perfectly spherical, then our registration accuracy should go down even further. So after these machine spheres, we then use Ruby spheres, which are used for precision applications in mechanical engineering. And as we predicted, we saw that the registration accuracy went down accuracy went up, but the root mean square went down even further. So now we have 200 nanometer scale displacement across the vertical and 100 nanometers or so in the horizontal direction. So basically, each time the head implant is placed under the device, it moves somewhere between 100 to 200 nanometers. Now, I tested this in several different head plates. So one of them is the solid machine sphere that I was talking about before, but I also tried to attach, like cheap stainless steel spheres that you might buy from your typical arts and crafts store. And I epoxied those onto a head plate. And I also used the rubies, like I said. And I did that, 100 trial succession iteration for each of these head plates 3 times. And the data shows that obviously the rubies are the best. They cost like $20 a piece, so they're kind of pricey in comparison, but that all of these types of head plates do have hundreds of nanometers in resolution, which is pretty great. So with that, I want to just talk about the future directions. First, I want to obviously test this in a head fix in vivo experiment. It's easier to do than trying to implement an entire voluntary head fixation chamber. But voluntary head fixation is also one of our goals with this device. So in the future, we also plan to implement a kinematic clamping system similar to the Tank and Brody Labs device. And with that, I would like to mention my acknowledgements. My advisor, Ben Scott, Amy, who is in our lab and has helped me a lot with this work, our lab, the Center for Systems Neuroscience at Boston University, the Neurophotonics Center, and our collaborators. And yeah, that's my talk. If you have any questions, please let me know.
Thank you, Sue. That was really cool. We have a question from Horst Obenhorst. He says, this is cool. The precision of ruby versus stainless steel, etc. is higher, but what about the overall stability? Could you test that?
Yes, right. So we're trying to test that right now in an awake animal. I've tested it in an anesthetized animal by trying to image the hippocampus, which is a pretty deep area, but I plan to test stability in an awake head fixed animal using this in the near future.
Yeah, that's really cool. So have you, I'm guessing you're going to be doing a lot more tests and stuff, but have you, or are you planning to look at developmental positionings and things like that, and whether you're able to really follow the same structures over time by replacing the...
Yeah, so luckily for me, I don't have to do this all by myself. That would be awful. My lab, there's someone in our lab who's examining developmental time points in birds. So we have zebra finches that this is light enough to be implanted on. So ideally, I can give this to her and let her use this in her experiments. There's also another person examining this CA1 region in the hippocampus over several time points across 2 weeks, I want to say, and which is not as long. I think the birds, it's much longer. It's on the order of a month or something. But that's definitely one of the main applications, is needing to go repeatedly to the same spot with high precision, which is kind of hard if you need to like match it up eye to eye, which what you might need to do with normal imaging studies.
Yeah, but given the drift that you see, it's actually very less drifty, I guess. So the precision is quite high. Do you actually want to elaborate a little bit on the overall process? Like, how would you go about the entire experiment?
Yeah.
With the positioning and what the experimental design entails, essentially.
Sorry, I'm having technical difficulties, which might not be that surprising. Okay.
Okay.
So this is what I think it would look like. I just put a mouse in here, but that doesn't really matter what the animal is because it might be applied to birds. But basically you have your objective and your clamp, and they ideally the microscope ideally doesn't move aside from when you use it. Obviously this isn't the case for everybody, but so you then position your microscope above your region of interest in a given animal, and you image it. And then after you're done, it's only moved in the z direction, a known distance, and you put a different animal in. You put the objective back down to where it was before. You haven't reset the coordinate system, and then you move it to where your region of interest is, which might be slightly different. You know, if it's like the hippocampus of a different animal by like some number of like microns or something. But like this is also for the head fix setup, right? But then you image the animal, and then you have this pipeline of like each animal is always after the other one, and you know where this is in relation to the first animal. And so that way you could even program your objective to do this for you, so that, and you can say like, hey, like, you know, go to this location after some amount of time after I put the other animal in. And this is much better than, you know, screwing in a different animal, you know, trying to find the same spot over and over again, repositioning. And I think that this is the ideal situation for a head fix setup, for example.
Yeah, that's really cool. All right, I guess we have no more questions. But if there is something else that you would like us to know about the setup, and if you want people to actually contact you in order to be able to use this and test it in their own experimental setups, that would be great. So I posted that on the chat. So
yeah, great.
Everyone, please feel free to contact Sue if you guys want to test this out.
Great, thank you.
All right, thank you, Sue, so much. That was really great. We're going to move on to the next talk. It's by Jess Minder. Jessica is a graduate student in the lab of Robert Fromke and Moses Chow at NYU School of Medicine. And today she's actually going to talk about oxytocin and embryonic diapause. It's very, I guess, interesting thing that pregnant mice can do, which I had no idea about, and Jessica's gonna tell you more about that. So go for it, Jessica.
Hi, can you see my screen and hear me okay? Okay, cool. So thanks, Lena, for the intro. So my name is Jess, and like Lena said, I'm gonna talk to you about oxytocin and embryonic diapause. And so what is embryonic diapause exactly? And so this is a mechanism that has evolved to enhance overall reproductive success during conditions that are unfavorable for pregnancy. And so one such condition is while nursing. And so this puts a very high metabolic load on the mother. And so maternal mice are referred to as dams. And so if a dam is nursing, and if pregnancy ensues, then all of the embryos within the second pregnancy will pause their development at the blastocyst stage depicted here, and implantation into the uterine endothelium will be blocked. And so once nursing ceases, then the embryos will implant and development will resume as normal. And so this is widely reported throughout the mammalian animal kingdom, but in terms of mice, how long this delay actually is not well known. And so we wanted to test this in the lab. And so we let dams deliver. We induced diapause by mating them immediately upon delivery. we breed them, we remove the male the next day, so that's when we know when the second pregnancy occurred, wean the pups at 3 weeks, and then simply just monitor the time it takes for the animal to deliver the second litter. And so on average, as you can see in the pink bars, lactation extends the mouse gestation for an average of about 10 days across several different mouse lines. And so our hypothesis, since we know oxytocin is very important for lactation, our hypothesis is that the neuropeptide oxytocin, which is released in the supraoptic and paraventricular nuclei of the hypothalamus depicted here in pink, acts to coordinate the pre-implantation embryo and the uterus to initiate diapause in mice. And so we wanted to ask, what is the necessity for oxytocin within the embryo? And so we did this sort of genetics experiment, breeding mice that are heterozygous for the oxytocin receptor, letting them deliver, inducing diapause by mating the dam with another heterozygous male, removing the male so we know when the second pregnancy occurred, wean the first litter, and then let the dam deliver its second litter. But the real experiment here was to genotype the pups and look at the distribution of the genotypes between the first and the second litter. And so typically this line breeds to Mendelian ratios, but we wanted to ask if those ratios are skewed within litters that experience diapause. And so pink is the litters that went through diapause. And so interestingly, within those litters, we do not see any pups born that are knockout for the oxytocin receptor. And so this suggests that oxytocin is essential on the embryo for the embryo to be able to go through diapause. And so just to confirm, we know that at this stage, at the blastocyst stage, the embryo oxytocin receptor is expressed on the embryo, as shown here in red. And then we turn to a method developed by the Zernicka Goetz lab to culture embryos to test what oxytocin exposure does to embryos directly. And so here you're looking at the development of a blastocyst, and this protocol enables us to culture them past the implantation stages. And so if you look in the last panel, you can see that the outer layer, the trophectoderm, so this is the layer of cells that will implant into the uterus and later become the placenta. And so what happens in vitro is that these cells begin attaching to the plate and expanding beyond the embryo proper. And so this is our in vitro proxy for implanting. And so since implanting is blocked in diapause, we then wanted to ask, is the time to implant then delayed with oxytocin exposure? And so that is exactly what we see. So in the saline treated condition in panel A, to the right, you can see that the trophectoderm cells start expanding beyond the embryo as indicated by the arrow, but that is absent in the oxytocin treated condition. And panel B is just a cumulative distribution of the percentage of embryos that implant over a time course of several hours. And so on average, there seems to be a delay of about 12 hours. And so if you just look at the embryo broadly, you can see the outer trophectoderm cells, that'll be the placenta. And then you have the inner cell mass, that's going to become the embryo proper. And then there's just this hollowed out cavity called the blastocele. And so if you want to signal for the entire embryo to enter diapause, you don't want some cells sort of pausing and other cells still growing. So we hypothesize that a good way to signal across the embryo in a synchronous manner is through this cavity, which makes contact with most of the cells. And a very quick way to do that is to modulate ion flux. And so we perform patch clamp electrophysiology on the embryos. So right now I'm currently just working on getting recordings from the trophectoderm. And it's still very preliminary, but overall oxytocin exposure as compared to baseline seems to be reducing overall ion flux. And so this makes sense when you have diapause, which is a condition of pausing, very low activity and metabolism, to have reduced ion current. And so the real take-home experiment that I'm working on now is trying to test the specificity for oxytocin and see if I can trigger the release of oxytocin in the absence of nursing to induce diapause. And so to get at this, I'm working on an optogenetic activation of oxytocin neurons. So we have oxytocin-Cree animals that we breed to channelrhodopsin animals to get channelrhodopsin expressed specifically within the supraoptic and paraventricular nuclei of the hypothalamus. And so the timeline for this experiment is implanting a fiber, letting the animals recover, hormone priming them to breed them and get them pregnant, And then after the detection of a copulation plug in pregnancy, stimulating the animals via optogenetics to trigger oxytocin release in a pattern that mimics nursing for about 1 week. And so typically in a normal pregnancy, the embryos implant between days 4 and 5, but I'm going to be harvesting the animals between days 7 and 8. So if diapause occurred, we would expect that embryos should be able to be extracted from the uterus because they did not implant. And so that's the anticipated result that we expect from this experiment. And so this is currently ongoing. And so just basically in summary, we know that on the morning of day 4 of pregnancy, there's a surge of estrogen, and that makes the uterus receptive to implanting embryos. But when a dam is nursing, for some reason, that is blocked and we don't understand how. And so we think oxytocin is playing a role in that because oxytocin has been reported to sort of reverse the maturation of the corpus luteum in bovine literature. And so that would, in effect, make the uterus unreceptive to implanting. And so what I've shown you is that lactation typically induces this 10-day delay in mice on average, and we want to more specifically induce this by triggering oxytocin release with optogenetics, and then we're also working on intranasal administration of oxytocin. And then within the embryo itself, somehow the embryo undergoes diapause, which is not well understood, but our hypothesis is that it's oxytocin that's the cue to initiate entry. And so what I've shown you is that direct exposure to oxytocin in the embryo does trigger a delay, and that the receptor is needed within the embryo for the embryo to undergo diapause. And so with that, I just want to thank the Frunking and Chow Labs, and thank you all for your attention.
Thank you, Minder. That was Minder. That was really great. So do we have any questions from the audience? Let me see. We don't have. So as we wait for the questions to come in, I actually have a question. And I was wondering, in some cases, there is an immediate delivery of the next litter if the dam has been with the male and has, so it immediately gets pregnant after delivering. So are there different signals? Like, why is it that some dams have diapause and others do not?
So the only thing that I've seen in my hands that relates to that is the size of the litter. So sometimes when the litters are very small, especially like 2 or 3 pups, and even like 7 pups still, the delay is either non-existent or pretty short, like a day or 2. So actually the delay that I've shown, most of the litters tend to be, especially in CD1 mice, they're like 14 plus pups. They're very big litters. And then usually within the black 6 mice, it's, something above 7 or so. So some of the points I have that are really small litters, the delay was only like 1 or 2 days. So that sort of like brought the average down. But yeah, so that might be sort of why in practice it doesn't look like diapause occurred, because the litters were really small.
I see. So basically the amount of oxytocin released as a result of the number of pups that there are would determine how long the diapause would be for the next litter.
In theory, yeah. So that's something I haven't like systematically gone and, added, okay, 1, 2, 3, 4, 5 pups, but that might be something that might be worth doing. And it's not reported in the literature. There are some reports, that it skews with pup size, with litter size, but it not in a very systematic way. But, it is also interesting because, so there's a 10-day delay on average. So it's not like the pregnancy starts immediately once I wean the pups, right? Otherwise we'd see a 3-week delay. So also I think something happens, there's some sort of homeostatic mechanism as nursing goes on where the mom adapts to that sort of higher metabolic load. So the pregnancy is starting to progress while she's still nursing. And also as the pups get older, they probably just don't nurse as much, so.
Yeah. So what about the effect of oxytocin on the pups after they've been implanted? is there any effect or any delay after implantation, or is it all just pre-implantation?
So in diapause, it's all pre-implantation. It varies within some species. I think it's hamsters. The embryo is actually still kind of in plant, but then they delay, then their pause is like right after implanting. So some species vary exactly as to the timing of implanting, and some species, I think it's the mink, they like half implant, there's like a semi-implanting. But basically, for the most part, it's sort of the delay is sort of at that point. Whether or not they implant might vary with the species, but that's when the delay happens. There is one species, I forget, that where the development is kind of just slowed overall. But yeah, a lot of, so in mice, this happens in a facultative manner. So basically just if there is some sort of a stressor, but some species have obligate, where every single pregnancy goes through diapause. So those are animals that, like, let's say, are hibernating animals for the most part. So this way, it times all of their pregnancies to be delivered in the spring when resources are abundant.
Very cool. I guess we don't have any more questions, so we can move on to the next speaker, Naomi Lopez Caraballo. She is also a grad student in Robert Funke's lab at NYU, and today she's going to talk about social transmission of maternal behaviors through observation. The stage is yours, Naomi.
Thank you. Trying to share screen. Okay, can you see it? Can you hear me? Okay. Thank you. As Lina said, Naomi Lopez Caraballo, has well in the Fromke lab, has just, and I'll be talking about social transmission of maternal behaviors through observation. So just as a quick intro, as I mentioned, I'm part of the branch of the lab that studies maternal behaviors and oxytocin, with the hopes that we can shine light into how modulatory systems like the oxytocinergic enables complex social behaviors. In our case, we focus on maternal behaviors, specifically pup retrieval. So what happened is that when mouse moms have, after they give birth, they keep the pups in a nest. And when a pup finds itself outside of the nest, the mom promptly fetches it back to the safety of the nest. On the other hand, inexperienced virgin females don't naturally retrieve pups. They tend to ignore them or even attack them. However, we know from literature and from our lab's work that virgin mice can learn such behaviors from living with a mom and its litter, as is shown by the black trace in the graph. And this learning process seems to be enhanced or accelerated by increasing levels of oxytocin, as shown by the red and the blue trace. So while we knew virgins are capable of learning, we didn't know what experiences during cohousing actually lead to learning. What are the experiences that are training the virgins? So we decided to videotape the 4-5 days of cohousing continuously and track the behavior of both the virgin and the dam, as shown in the ethergrams at the bottom. The dam is the mother. And one thing that brought our attention was dam self-generated retrievals, which are marked as the blue lines in the dam's ethogram. So what happens is that sometimes mom accidentally dragged the pups out of the nest. Sometimes they're lactating and she goes out of the nest and the pups fall out, which she would subsequently retrieve. So we wondered if those are opportunities that the virgin has to watch the mom retrieve pups and learn to do it herself. So to test that, we developed a behavioral setup in which virgins once a day observe maternal retrievals through a transparent wall. So the virgin is one side, the mom is in the other with the pups, and we would take pups out of the nest and the mom would retrieve them and the virgin would watch on the other side. And we would do this once per day for 4 days. And afterwards, each day after the virgin observed the maternal trials, maternal retrieval trials, the virgins were tested for pup retrieval, which means she's alone in a cage with the pups, and we take pups out of the nest 10 times, and we score her performance on how many out of the 10 trials that we do, how many times she retrieves. And when we did this experiment, as we were expecting, virgins actually learned just by observing a mom retrieving on the other side of the transparent wall. On the other hand, when the transparent wall was replaced by an opaque wall, virgins' performance decreased. Virgins did not learn. So meaning that the visual cues are essential for this type of learning. We also tested oxytocin receptor knockouts in the transparent barrier, so the visual input is present. And the performance also decreased, hinting us that oxytocin does seem to play a role in this type of learning as well. So this wall, and just a quick note. So I have Joanna Garcia keynote room 2 because these experiments are part of a paper that is already available in bioarchives, and that Joanna will be presented in the keynote talk afterwards. So if you want more details, make sure you attend to that. So this wall is not sealed, meaning that not only visual input goes through, but also olfactory and auditory stimuli can go through as well. And while we think different sensory modalities contribute to maternal behaviors, actually our lab historically has focused on the auditory part. But reviewing our previous findings, it does seem that the visual input is essential, at least in this specific paradigm. So now the question is, what are they seeing and that is informative for learning? So to answer that question, we took the setup to the virtual realm. So we could have full control of the experience the virgin gets. And so virgins were head fixed in front of a monitor from where we play 40 repetitions of a video of a mom retrieving, spaced each repetition by a few seconds of dark background. And virgins were tested for retrieval afterwards, the same way that we test them in previous for the previous experiments. And we repeated this experiment once per day for 4 days. So as a spoiler, we tried different types of video, but I'll get into this right now. So the first thing that we did was to play a video of a mom just retrieving a pup. So the pup is outside, the mom goes, fetches the pup, very cute, puts it back to the nest. And the virgin watched this clip 40 times per day. And as we expected or hoped, virgins did learn to retrieve after the 4 days of the experiment. As I mentioned, our lab has historically focused on the auditory part of pup retrieval. So we wonder, what if we add pup calls to the videos? What if we add audio? Maybe having two sensory modalities, two sensory stimuli might improve the performance of the virgin. However, that was not the case. So the purple bar is showing the spectrograms of the pup calls. So performance didn't really increase, but this might be due to the audio being distracting, and pup calls played on a speaker seem to be aversive to virgins. Can relate. Who would like to hear baby cries for 10 minutes? But on the other side, when we just presented a video of a dark background, so basically no video, virgins' performance decreased. And I guess I have a few minutes, so I'll go through this, just have a couple more slides. So the setup seems to be working. When we present a video of a mom retrieving, virgins learn. When we don't present any video, they don't. So we decided to explore the idea of what if we change the video, can we affect performance? So the first thing that we did is that sometimes moms don't retrieve correctly all the time. They might drop the pups midway and leave them out of the nest. So thinking of that, I thought that I could try to bias the virgin's performance toward making retrieval errors. So I attempted to do so by playing a video of a mom failing to retrieve the pup all the way to the nest. So she drags it, she leaves it, poor pup, and she goes inside the nest. And I have done this in two ways, with audio and without audio, but in both cases, the learning seems to be decreased and slow, and most of the time, the bridges don't even attempt to retrieve the pops. And lastly, similarly, we presented videos of just a virgin mouse hanging around the cage without any nest, without pops. And so far, it also somewhat elicits behavior as well at a slower rate. And these results might be a bit tricky to understand, but what we can say is that some social stimuli is presented, that is the connect specific, the novel connect specific, and that oxytocin has been generally related to a wide array of social behaviors. So we wonder if that is enough to turn on the oxytocinergic system, making the brain more receptive to any social cues and learning. So in summary, the possibilities we can explore with this setup and the variety of movies that we can present are kind of endless. And the behavioral results we obtain from each variation are as complex as understanding social behaviors themselves. But for my next experiments, I'm mostly focusing on the two most distinctive results, which are retrieval videos versus dark background. And we are monitoring overall activity of oxytocinergic neurons in the paraventricular nucleus of the hypothalamus using fiber photometry while the virgins watch the movies. And we're also using optogenetical inhibition to inhibit oxytocin cells while they're watching the movies and see if that decreases the performance to that of dark background. And with that, take questions.
Awesome. Thank you, Naomi. That was really cool.
Sure.
Again, we're going to be waiting for a few minutes for questions. I actually have a question about your difference between, the visual cues and the auditory cues in order to elicit this behavior. So are you planning to look at, the visual cortex projections to the hypothalamus or this specific nucleus, the oxytocinergic neurons over there, and how over time when the virgins are learning, what kind of supposedly plasticity mechanisms are occurring as a result of that pathway and not the auditory to the paraventricular nucleus pathways? Like what would be the differences? Because I mean, to me, it seems like, if there are distressed pup calls, that would kind of elicit a stronger reaction compared to, visual evoked learning. But yeah, what are your thoughts on that?
Yeah, so we plan on studying and looking at the visual cortex. Actually, some of the optogenetical stimulation that we plan to do will be in projections at visual cortex, which is similar to the experiments. I don't know if you have come across Bianca Jones-Marlin was the first student to work in our lab with this project, and she stimulated the auditory projections into the oxytocinergic projections into auditory cortex. And that's how she saw enhanced or accelerated learning of pup retrieval. Yeah, right now I'm focusing just on the visual part and the projections going from PVN oxytocinergic to visual cortex. We plan in the future to explore with the auditory part because it's such an important side, especially in our lab. We have, pretty much all of our papers, maternal behaviors are focused on the auditory side, and Joanna will be talking about it in a bit. But there is a lot of things that will be explored with the setup. But for now, I'm focusing only on the visual side.
Cool. So are there differences in projection strength between the auditory and visual, either to the oxytocin neurons or from the paraventricular nucleus?
I'm actually not sure if I have come across any information about that. Actually, I'm planning on doing a more robust characterization of the projections into visual cortex, because previous papers from another member of Chow lab and Rob Fromke's lab where they studied the receptor expression. Mariela also showed that there are projections into visual cortex, but we did at the moment we didn't get into much details. But I'm planning on at least tracing the projections. But no, I don't. I wouldn't. I don't know. I'm not recalling anything specific to answer your question.
Okay, no worries. So we have a question in the chat from Vasil. He asks, does inhibition of oxytocin neurons decrease pup retrieval?
Inhibition of pup. Yeah, so that's what we're trying to test now. So I didn't present the data because I started those experiments just now, but we're optogenetically inhibiting oxytocin cell bodies in the paraventricular nucleus using ARCH. And while they're watching the video only, and then we follow the same protocol, they're tested afterwards in the for pup retrieval every day. So the experiment goes pretty much the same. So far, we're getting a decrease in performance, but we're just 4 animals in, and we don't have the control. So we'll see. But fingers crossed. That's our hypothesis that it will lead to not to a complete, going to zero into learning, but just something more similar to what we observe in the dark background. And we're planning also as well inhibiting visual cortex to also test the same idea.
Yeah, it would also be pretty cool to image from oxytocin neurons while during learning and see how the levels of, or how active those neurons are, or whether the levels, how robustly the levels of oxytocin change across learning as well.
Yeah, and that's a cool thing because so far until now, the issue that we had with this kind of experiment is that we were doing co-housing. So we just have animals in a cage continuously for 4 or 5 days. We couldn't do fiber photometry for 4 or 5 days without stopping. We couldn't do 2-photon. So, and even with it, Bianca did some optogenetics, but it was, at certain time points. So the importance of taking this to the visual realm is not only that we finally get full control of the experience, so we can start to playing around with it and parsing out what is important, what's not, but that also we can use these techniques like fiber photometry. I actually consider 2-photon, but just because I want to do fiber photometry, I'm focusing on that. But that's a possibility as well to take it to the 2-photon, and we can do more precise optogenetics. Use optogenetics exactly when they're watching the video. So supposedly exactly when they're learning to retrieve. So yeah, it's really exciting that this works. It was kind of a crazy idea of showing movies to mice, and hopefully we can get some amazing data out of it.
Yeah, that's really cool. Well, thank you so much. I guess we have no more questions. If there are any more questions, we have a few more minutes left, so feel free to post your questions or come live to ask the questions. I guess while we're waiting, maybe I can ask one last question to Naomi as well. Actually, I can see some parallels here between Sue's and Naomi's work, where perhaps Naomi could use those kinematic head plates, removable ones, and put the virgins while learning, and then put them back in the cage and image.
Yeah, that'd be exciting.
Yeah, that would be super cool. So how do you envision this circuit works? I mean, so the visual inputs are going in to the visual cortex, or the auditory cortex, and then are there direct projections, or is it through some other type of disynaptic circuits that eventually feed onto the oxytocin neurons?
Yeah, so we know there are direct projections from oxytocin from the preventricular nucleus into visual cortex. We know that's the same for auditory. We don't know as much from projections back from these sensory areas to PVN. Actually, I don't know if Silvana from our lab presented already, but she's actually looking at the circuit back from auditory to PVN. But in the case of visual, we still don't know. That's also something that we have to look at. Yeah, but the project is quite recent, so we'll see. There are a lot of things that we have to explore. But you know, it's complicated. We have had many ideas about this because so many sensory modalities can play a role. If you know, maternal behavior is a pretty important behavior because the species depends on it. And you would think that for such as important behavior, you would try to use any cue or any sensory modality that you get to take care of the baby. So, you know, I don't know if you have babies, but if someone has a baby and, you know, you see that it is in danger, then you go aid it. If you hear it, you'll use that cue. I don't know, if you smell something, you'll use that cue. So with this, we're not saying that auditory or visual or olfactory is more important than the other. I think it's very complex how they all interact. That's also the beauty of this setup, is that we can focus on different sensory modalities more precisely and start to parse that, which we couldn't do during co-housing. But you know, we have had many theories about how they interact at what points as well. So for example, if when they're interacting with the pups, maybe smell is more important. They're exploring the new pups, getting familiar with them. Auditory, if they're emitting distress calls, obviously might be alarming. They're hearing this pretty annoying sound, and they eventually learn that to shut down that sound, they have to take the pup back to the nest. And if you see it outside, then you use visual. So it's pretty interesting and quite complex to think about it, and we'll see where it takes us.
Yeah, very cool. I mean, it is, like you said, a very natural and innate behavior. So it's very important to dissect all of the circuits associated with it. All right, I think we are right on time to end this session. So I'm going to thank the three of you once again. Thank you so much, and enjoy the rest of your day. Thank you everyone for attending.
Interactive talks. I'm a researcher at Johns Hopkins University, soon to be at the University of British Columbia, and I'll be introducing the speakers briefly. Just to let the audience know, the talks are approximately 5 minutes each, after which there'll be around 10 minutes for questions. Please ask the questions using the question and answer, or the Q&A button at the bottom of your screen. And once you ask the question, please also indicate whether you would like to come on yourself and ask the question, or whether you would like me to ask the question of the speakers. So let's get started. The first speaker is Sam Alvarez from the University of Rochester. He's a member of Ralph Hafner's lab. And Sam will be talking about a method to estimate the magnitude of task-related feedback without the need for data from a second task. And this talk is entitled "A Method for Distinguishing Between Feed-Forward and Feedback Influences on Your Responses." Take it out, Sam.
All right, let me just share my screen here. All right, can everybody see that? All right, like I said, my name is Samuel Alvarez. This is a presentation on distinguishing between feed-forward and feedback effects on neural responses. This is work done with Ralph Hafner at the University of Rochester and Richard Lange at the University of Pennsylvania. So this is a very simple representation of the neural hierarchy to help frame the problem. Lower level processing areas such as V1
do some feed-forward processing driven by an external stimulus, and also receive some feedback signal from higher level processing areas. And our goal is to distinguish this signal from the feed-forward effects. So how are we going to approach this? We have an assumption that feedback will have task-dependent effects on the covariance matrix of neural responses, while feed-forward will have task-independent effects. This is based on work done by Bondi, Cohn, and Newsom. So what do we mean by task-dependent effects? What will these task-dependent effects look like on the space of neural responses? They will change variance in some directions, which we'll call H vector, while leaving other directions, which we'll call the G vector, unchanged. And on the upper right side of the screen there is a very simple cartoon that what it would look like for the 2-neuron case. The green arrows represent our H vector. You can see in the case of feedback, variance is stretched out in the H direction, while in the case of no feedback, variance in both directions is about equal. Now, why do we call these task dependent? It's because these directions are defined by the neural responses to a task. So to give a concrete example, if you expect feedback to increase over the course of learning, the neural responses of a monkey, say, trained to do coarse discrimination in the cardinal direction, we'll call that our H vector, compared to a direction in which it is untrained. So like, say, oblique discrimination, and we'll call that our G vector. So this sort of defines our strategy for estimating the strength of feedback. We'll compare variance in the task direction in which you expect feedback to variance in a task direction in which you don't expect feedback. It's worth mentioning, what exactly do you get out of an approach like this? Previous methods of detecting feedback, they've required comparing correlation structure across different task contexts, or in other words, they've required training a monkey on a variety of different tasks, cardinal discrimination, oblique discrimination, so then you could compare across the two. With this method, you would only need to train the monkey to perform a single task, which could save a lot of time and money if you've ever trained animals to do these tasks. So, now how do we want to test this up? In the previous cartoon I showed, we assumed isotropic noise. We don't want to assume that, since we know there is correlation structure in sensory neurons, neurons with similar tuning properties. Hold on 1 second. Neurons with similar tuning properties tend to have higher correlations. We want a model that will produce realistic feed-forward correlation structures similar to what we see in the brain. And such a model exists, proposed by Ingmar Kennetschatter. I'm just going to call it the Kennetschatter model. And it's going to give you a very general idea of how it works. The firing rate is just some nonlinear function of a receptive field dot product with some noisy image. And then we're adding some strength times some feedback signal to that. In this model, feed-forward correlations are a result of sheared noise as a result of overlapping receptive fields. And we're using 2 sets of receptive fields, simple receptive fields that we're just defining as a 2D Gabor function, and learned receptive fields from Olshausen and Field. And our specific feedback assumption, feedback will add variance in the H direction. This does not necessarily have to be the case. It could decrease variance in the H direction, and this method would still work. The big idea is that it will change variance in the H direction in some way, but we're assuming that it will add variance in the H direction. What we're really testing here is how well does the assumption of equal feed-forward variance hold up. Now, these are the results for the simple model, where receptive fields are just some 2D Gabor function that varies only in orientation, tiles the task space evenly, 640 neurons. Along the x-axis, we have different G vectors generated by neural responses to a coarse discrimination task, where the grading stimuli vary only by orientation, and we're rotating from oblique discrimination to cardinal, back to oblique. And on the y-axis, we have variance in these different G vector directions. The orange line is the result of having added feedback in the cardinal discrimination direction, and the blue line is the no feedback case. So this is really just a proof of concept. You can see our assumption holds in this case. The points along the blue line are all equal in the case of no feedback because they're generated by neural responses to equivalent tasks. And the green arrow here points to the value of the variance at our H vector. and the red arrow points to what we'll be comparing to, G vector that is orthogonal to H. We can calculate a D prime between these two as an estimator of the strength of our feedback. Now, obviously, this is a very simple model with very cleanly defined receptive fields. What we really want to look at is something that's a bit closer to what we may actually sample from the brain. In addition, variance is just one measure we might use. There may be other measures that are more sensitive to the effects of feedback. So we want to do a power analysis of different measures on a more realistic sample of neurons. So what we've done here is we've used our model with learned receptive fields this time, still 640 neurons, but we've simulated recording from just 50 of these neurons at a time. We have D prime on the y-axis and increasing levels of feedback on the x-axis. The different color lines are all different measures that we're testing. In the interest of time, I won't go into detail about them, but I will point out that variance is there in green. Now, the good news is that it works. Nothing crazy happens. The method doesn't detect signal where there is no signal. It's increasingly sensitive to signal as the value of the feedback increases. The 4 out of 5 measures are pretty comparable, especially at low levels of feedback, but they begin to distinguish themselves at higher levels of feedback. Unfortunately, none of them really distinguish themselves by being sensitive to low levels of feedback for this realistic sample of neurons. So that's it for now. What's next? We're asking, are there other measures that we can throw at this model to test their sensitivity? Are there ways to make the current measures more sensitive? Can we make the model more realistic? Currently, we assume that we have perfect control over input to the model. It's not necessarily the case with real subjects, because you control the image on the screen, but not on the retina. There's currently no model of recurrent connections within this. And of course, if we can find a data set to test this on, where the feedback signal is strong enough for this method to detect, just to verify this on real experimental data, that would be great. And that's it. I will take questions now.
Thank you so much, Sam. I don't see any open questions at the moment, but please do ask questions. I will open with a question. And I was going to ask about recurrent connections, and that's what I was thinking of as you were talking. But what are your ideas about how to bring recurrent connections into this, as well as nested feedback systems?
I haven't put much thought into currently what model of recurrent connections we're going to go into. I've been looking more into trying to increase the sensitivity of the measures so we could detect these at lower levels of feedback with these realistic neuron samples. That's kind of what I was hoping to get some feedback on. Maybe some people had suggestions for recurrent models to look at to add to this model. I would say probably more than recurrent connections, what I'd really like to do as soon as possible is find a good data set of actual experimental neural data to test this out on. And then, if we can't find one, or we can't find one where we can detect signal, then maybe we need to start looking closer at recurrent connections to see how that adds to the variability in this model, so we can get a more realistic idea of, okay, exactly how much feedback are we going to need for this method to work.
Understood. We have a question from Carly Sombrick. If you want to come on and ask your question, that would be great. And this is related to what you were just saying about getting actual neural data.
Hi, thanks for your talk. I'm also a Rochester alumni, so always good to see another one. But what kinds of tasks do you think your model would generalize to?
Well, I presented this entirely in the context of orientation discrimination tasks. Now, hopefully this would generalize to any sort of task in which you had a reasonable expectation of symmetry over the variable that the task is measuring on. So obviously, human brain deals with a variety of orientations in the set of natural images. So you can have a roughly reasonable expectation that it would be have equal variance in all different orientation directions. Very roughly, like I said, that the one where it was really nice and even, that was just our simple model. But
do you think it would work for visuomotor rotations?
Yeah. I mean, again, this is all work done on a model so far. I would be a lot more comfortable with making declarations of where it was going to work if we had actual data that we showed it could work on.
Fair enough.
But,
thank you.
Related to that, how do you think the first of all, thank you so much, Carly, for asking the question. And how do you think the model will scale with dimensionality of task-dependent or non-task-dependent axes? So if we had more than 2 dimensions.
I guess could you clarify what you mean? I miss.
Yeah, so you talked about multiple task-dependent directions, right? So in this case, there is a direction and an orthogonal direction. But what if there were more than 2 axes? So what if it was a 3-dimensional space of tasks where one is a task, one is a direction that correlates with the task, and 2 directions are non-correlated with the task? I guess my question was, would the sensitivity of the model go down, perhaps, if there are more the higher dimensions you get?
I would say that as long as the other directions are orthogonal in neural space, then it shouldn't matter. Yeah, as long as they're orthogonal in neural space, it shouldn't matter. I mean, like in that graph, if I'm understanding your correction on slide 6, we did show that we had multiple different tasks. We didn't have simply one G that was orthogonal in vector space. We had multiple ones. I mean, we're just saying we're going to choose the most orthogonal.
I am sorry, I totally missed that. Okay, that clarifies it. Perfect. All right. Seeing no other open questions, let me thank Sam for his time and the wonderful talk.
Thank you.
Let's move on to the next speaker, which is Lokesh Bhuminathan from Rice University. And he is a 3rd-year PhD student with Zach Pitko. And his latest article, which I don't think this talk is about that, but his latest bioarchive paper talks about a deep learning framework for estimating crowd density from images of crowds, which is super cool, regardless of whether we are going to hear about it or not. Lokesh also wanted me to mention that he is actively looking for summer internships. So if anyone is in the market for a summer intern, please hit him up. Please take it away, Lokesh.
Thank you. So I'm going to be talking about inference as control. Just the premises of this is that sensory observations about the world are invariably ambiguous, and the inference about the world's latent variables is an important computation for the brain. However, computational constraints limit the performance of this computation, and these constraints includes energetic cost in terms of neural activity and noise across the channel. Efficient coding is a prominent theory that talks about how limited resources can be best used in order to do this. And in one incarnation of this is the predictive coding theory, which basically says that predictions are being subtracted of the signal so that we don't have to send what we already know. But the predictive coding theory, however, does not account for the cost of all the noise associated with the channels and as well as the energy consumed in sending these predictions. In this particular project, I would be talking about how we can account for all the noise and all the energy costs that are that should be accounted ideally for predictive coding theory. And we will look at what different kinds of results does this lead to. And in order to do this, we formulate this problem as an inference problem on a graph, whereby feedback is viewed as a control signal, aiming to maximize how well an inference tracks a given target state. So in order to more concretely define our problem formulation, I'll just use a few slides to do that. So let's say we have a world state xt that evolves over time given some sort of linear dynamics, where A is a known matrix. And the observation that we see is basically a corrupt version of this world state, which is corrupted by the observation noise. And it's the first is multiplied by the C matrix. And based on what we understand of the world, we make predictions of what is going to happen. And this prediction is based on our estimate of the world at the previous time point. And this prediction is also corrupted by this feedback noise, which is not accounted in the predictive coding theory. And based on this prediction that I receive and the observation at the sensory level, I come up with a linear combination of them as the residual, which I send back to the brain for inference. So one thing about this whole model is that we assume everything to be Gaussian noise and everything to be linear. So this residual is formed based on the observation and the prediction from the previous time point. And combining my previous estimate and this residual, I form the new inference. So the kind of constraints that I was talking about is first the feedback energy cost, which over here is nothing but an average L2 norm of the predictions that I send. And the second is the feed forward energy cost, which is the average L2 norm of the residuals. And our ultimate goal is to have good inference, which is the average difference between the estimate and the actual world state. And my total cost would be a linear combination of these three costs. And that's the problem setup that we are looking at. And this can be solved using the popular LQG framework, where we think of the control as the predictions. More details I can talk about during the discussion if need be. And some of the interesting things that came out of this is that although I've not fully understood everything that's happening, a few takeaway points would be that there are settings in which actually sending predictions is not optimal, because it could be because the predictions are expensive, or it could be because the feedback channel is too noisy, and so on. And another interesting thing is that there are some non-trivial trends that we can observe. For example, one particular plot over here is where on the x-axis I have the weight assigned to feedback energy cost, and on the y-axis I have how much do I multiply the predictions that I get. So there are interesting trends in which first it increases and then decreases. So the key takeaway is that it's not at all trivial, and there are a lot of interplays and trade-offs involved, and this kind of framework helps us to understand that better. I can take other questions.
Thank you so much, Lokesh. That's great. That was it's hard to explain such a dense topic in such a short amount of time, and I think it's a really good job. I don't see any questions yet, but I'm going to ask a question. And so when you're talking about a linear Gaussian states and prediction, then the prediction correction framework, obviously a Kalman filter comes to mind.
Sure.
But in that case, there is no, in a Kalman filter framework, there's no feedback cost or feed-forward cost or inference cost. So that's what you're adding over here. So just like a Kalman filter, is there one closed form solution for an optimal linear estimator in this kind of framework?
Yeah. So in fact, Kalman filter is what is exactly happening here as well. It's just that I have kind of massaged the parameters and the problem setup in a way that it mimics what happens in the linear quadratic Gaussian setup, where again the estimates are formed using the Kalman filter. So the Kalman filter is very much relevant to this, and it's not kind of straightforward as to how it should be applied, but that's what is happening here.
Got it. I'm sorry. Go on, Arthur.
Yeah, it's just I'm trying to understand. I mean, could you comment on the three costs you have and exactly what they mean, and also maybe do you know what impact each of these costs has separately on what you're looking at on the right-hand side?
Sure. Yeah. So the three costs that I have is firstly the feedback cost, which is basically how much energy do I spend in sending the predictions down the hierarchy. And I define it over here to be the average L2 norm of the prediction signal that I send. And the second cost is the feed forward cost, which is what is being sent back to the brain. So that over here is the residual, and it's defined as the average L2 norm of the residual. And the third one, the most important one, is the inference cost. I'm doing the entire exercise so as to come up with good estimates of the world. So this measures how good is my inference or the estimate of the world. And one interesting example on the right side, how this all translates is, for example, this plot over here, the first plot over here, on the x-axis, we have the weight that I assigned to the feedback. That is the WF, the coefficient of how much do I weigh the feedback energy cost. And if it's very, very less, what it means is that I can send very strong predictions that beats the noise in the channel because it doesn't cost me anything. And by sending these strong predictions, which are also reliable, I can have my residuals, which is the difference between the prediction and the actual state, to be as less as possible. So this would mean that I send very strong predictions, and then I can save on the feed-forward energy cost. And that's why the control gain, which actually in a way measures how strong your predictions are, it goes to like infinity. And that's very useful because the energy cost is not there. But if our energy cost is going to be really high, if the feedback cost is going to be really high, then it's not worth sending these predictions. That's why it goes to 0 over here. So that's just one example of what's going on.
All right.
I'm not sure if I made that clear.
We have another question from Richard Lange. And I'm going to be asking the question, what would be gained or lost by going beyond the linear Gaussian case?
Right. So much of this solution for this problem was inspired by LQG. And I think any nonlinearity that comes over there, I'm not sure how to really do it.
Yeah. Perhaps some kind of local linearization.
Yeah,
is obviously what you think of initially, right?
Right.
So I also have another question, I guess from Dimitra. Why do you have to assume linear dynamics? So that would be the similar type of answer, I would imagine.
Right. Yeah, it's just that it's easier to work with.
So just to wrap up, I had one other question that I was thinking of as I was looking at this. No, I'm good. No, I cannot remember, but I will make sure to email you. This is fascinating, and thank you so much.
Thank you.
Thank you so much, Lokesh. Are there any questions for Lokesh? Going once, going twice? No. So thank you so much. Let's move on to the next speaker. Our next speaker is Arthur Pratt-Carabin from Columbia University, and he did his PhD in cognitive neuroscience at École Normale Supérieure in Paris, and is now a postdoc in the Cognition and Decision Lab of the Economics Department at Columbia University. Arthur enjoys theoretical investigation as well as experimental work. A first axis of his work focuses on approximate Bayesian inference and decision-making under uncertainty. A second axis relates to questions of efficient coding of external information, which is what he's going to talk about today. He works with Professor Michael Woodford, and he has a recent bioRxiv paper out with the same title as the talk he's going to be giving today. Welcome, Arthur. You are muted. Dimitra, could you unmute him, please? Or
okay, I should be back on mic. Can you hear me?
Yes, we can hear you.
Okay. All right, so yeah, thank you, Manu. So indeed, I'm going to talk about efficient coding, and sorry, is this? Yeah. So more like specifically, I'm interested in why the human brain produces biases when estimating quantities or magnitudes. And in this project, we propose to an explanation that like biased judgments about quantities and magnitudes reflect optimal decoding of noisy mental representations of these magnitudes. And so we'll see that this will predict that the bias in the estimation of a magnitude or a quantity will depend on the variation of the imprecision in the representation as a function of the quantity. So we'll be interested in how the bias and the noise vary together. And in addition, we think that there is a good reason for which the imprecision of the representations might vary with the value of the quantity. And this reason is efficient coding, and which predicts that the encoding should depend on the prior expression of the quantity, of the natural statistics of the quantity. So we look at this in the context of number perception, and we design an average comparison task in which we show to subjects, so human subjects, we show 10 numbers, 5 red numbers and 5 green numbers. And then we ask the subject to choose the color with the larger average. So they need to look at these numbers and then choose the color with the larger average. So we find that there seems to be noise in the decisions of subjects, so the probability of choosing one color is a sigmoidal function of the true difference between the empirical averages and not a step function. And in addition, the weight of a number in the decision appears to be a nonlinear function of the number, while for an optimal observer, it is a linear function. So we propose a model of encoding-decoding. In the encoding step of this model, the presentation of a number x elicits in the brain a series of n random signals, or I. And then in the decoding step, an estimate x hat is inferred deterministically from R as the maximum likelihood estimator of x given R. So the MLE, so the maximum likelihood estimator, has a variance, which is the inverse of the Fisher information. But it also has a bias, which is also expressed as a function of the Fisher information. In fact, as the derivative of the inverse of the Fisher information. So this, thus we have a model in which the estimate x hat of a number x has some variance and some bias, and both of which depend on the value of the number. And in addition, you can see immediately that there is going to be a relation between the bias and the variance, which is this one, that the bias equal to 1/4 of the derivative of the variance. And so this relation, so we find evidence for this relation in our data. And it's a relation that is very close to the Weyand-Stoker law of human perception that relates the discrimination threshold in perceptual task to the derivative of the bias. So this relation has been verified in many perceptual experiments. So you might wonder, why would the variance vary with the value of the number? What would the coefficient of the encoding vary with the value of the number? And one possible answer is efficient coding. So efficient coding predicts that the precision with which you encode a number or like a stimulus should depend on the statistics of this stimulus. So one thing I didn't tell you about our task is that we run this task with 3 different conditions that differ by the prior distribution from which we sample the numbers. So this prior can be downward, and so small numbers are more likely, or it could be uniform, or it could be upwards, and large numbers are more likely in our task. So we fit our model, and so we fit the Fisher information function that appears in our model. We fit it to subject data, and we find that it seems adapted to the prior. So in the downward prior condition, the Fisher information is also downward, and in the upward prior condition, the Fisher information fitted to subject data appears to be mostly upward. So, and in addition, this is our best fitting model, and it captures the aspect of the patterns in behavior that I talked about earlier. So in summary, I introduced an encoding-decoding model in which the encoding was characterized by the Fisher information function, and the decoding makes use of the maximum likelihood estimator. This model predicts a relation between the bias and the variance of the estimates. It was our best fitting model, and it yields a Fisher information function fitted to subject data that seems efficiently adapted to the prior. Thank you.
Thank you, Arthur. That was concise and great. That was very clear. Thank you so much.
And I have like suggestions for our questions.
That's always helpful. So let me ask a question not on this list first. So how would you account for any variation from the Fisher information that is predicted by an MLE? So what other alternatives could there be? For example, in your data with the flat prior, you had Fisher information that at least on one side looked significantly different, it would seem like. So how would you, what other models could there be in order to explain that?
So, sorry, I don't know if you can hear it, but my computer is kind of stuck, but so I can't go back to the slide for some reason.
We can hear you though.
Oh, okay, so it's moving on its own. Okay, I don't know what's going on. I don't know exactly. So first of all, what I've shown here is so the Fisher information fitted to subject data. The truth is, I don't know exactly what the optimal Fisher information would be in this task. In the literature, there are like results on efficient coding that will tell you that to maximize the mutual information between the stimulus and the internal representation, you should have a Fisher information that is proportional to the square of the prior. And so this is why here I've plotted the square root of the Fisher information versus the and the prior. But in real life, no one ever asks you to maximize the mutual information. In my experiment, you were rewarded by the amount that you chose, like the red or green. So this is what you should maximize. Mm-hmm. And so that's the first thing. And the truth, it turns out that in this task, it's very hard to determine what exactly should be the optimal Fisher information. And another aspect is that to derive a result of this kind of like the relation between the square root of Fisher information and the prior, you do have to assume a small noise, and things get much more complicated if your noise is not vanishing. And so the optimal, I don't know. But in any case, what's really weird here is that it's in the middle case with the uniform prior. It should be symmetric, and any solution optimal solution would be symmetric. So we don't find that it's symmetric. So I'm not sure why. One possible interpretation is that people are used to. So the natural distribution of numbers in real life is kind of skewed, like it's log normal.
Benford's law.
Or like it's or something like this. So yeah, so that might be an explanation for this asymmetry. Yeah.
Thank you. So we have a question from the audience. Ari Benjamin, if you'd like to come on and ask your question, that'd be great.
Hi, how are you? Yeah, I was. It's actually very related to what was just asked, and I'm wondering if you ran a condition in which you did not tell the subjects what the distribution was to try to suss out what the natural prior, or to see if you could identify something of the form of Benford's law.
So I could do that. The only problem is that they might learn. I mean, so if I show them something, I have to choose the distribution from which I sample. And so eventually, I guess they will learn. So maybe if I just look at the beginning and fit only that, yeah, actually maybe, or yeah, instead of asking many questions to one subject, I can ask many subjects one question, and then the number that I show will be for that person from his natural prior. Yeah, that's a possibility. Here, let's say that I like when designing a task to make the things like very clear for the subject, and so actually we showed them, we told them explicitly what the distribution was. We showed them the PDF, but also we showed them lots of samples from the actual distribution that we were about to use, so that we hope that they learn about the statistics. What people have in their mind spontaneously like this as a prior for the distribution, I'm not sure. So you see in the literature they say it's something, yeah, so Benford, but I think Benford is for figures, right? Or for like the first figures in number.
First numbers in a set of naturally occurring numbers, typically.
Yeah, so I don't know what's, yeah, I don't know. And I don't know what's the natural distribution, and I don't know if you could actually prove that, measure that in subjects with an experiment, but maybe.
So related to that, how much experience did the subjects have with the trial before this data was collected?
So here, so each subject did like a 1 hour session, divided in 2 blocks. So they did 2 conditions out of the 3. Each subject did 2. And they had, so I showed them 200 times, I think 200 samples from the distribution. So they don't have to do anything, they just look at numbers. So hopefully they get used to it and they adapt their encoding. And then, if I'm not mistaken, I think they have 200 trials. And so in one trial, you see 10 numbers. So that's a lot of numbers, and it goes pretty fast because it's like half a second per number. So it goes pretty fast. But they are not, I mean, they are not trained to do the task, I would say, before I measure.
I see. So I guess my question was, do they do multiple trials, multiple sets of trials with the same prior?
Oh yeah, they do 200 trials with one.
200 trials.
For one prior, yeah.
Yeah, absolutely. Okay, so that was my question. Yeah, so that's great. But it's pretty amazing that you can actually reconstruct these priors relatively well from the subject's data. That's pretty impressive. Also, thank you so much, Harry, for asking the question. Are there any more questions for Arthur? Going once, twice? I don't see any. And so thank you so much, Arthur, for your talk.
Thank you.
Wonderful. And also, thank you so much to the audience. And I especially at this point want to thank Dimitra Mahotsa, who is in the back end for this room, who's been fielding all of the questions and typing all of the chats to the panelists and attendees. Thank you so much, everyone. Have a wonderful morning, evening, night, wherever you are.
Let's actually do most of the talking for today. Hopefully with some discussions going with the audience in our interactive talk. So each speaker will have about 15 minutes, and of course we're going to focus a little bit more on the actual interaction part of it, as the title implies. And first of all, we have Matthew Cook. So Matthew, if you could get your slides ready.
I will share here.
If I can remind the audience to use the Q&A as much as possible, so we can get some interesting discussions going. So Matthew is a grad student at UBC, and he is going to talk about Pathfinder, software package to analyze spatial search strategies. So please go ahead when you are ready.
Awesome. Thank you. Hear me okay?
Yep, great.
Awesome. Thanks. So Pathfinder is a open source software package that we developed in the lab. And it's kind of been a project that we've been working on for quite a few years now. And it came around because we were looking at analyzing, first of all, Morse water maze data for a rat task that we had done a few years ago. And we were looking at different ways of looking into the data that weren't just latency and like the classic measures of like latency and the distance swam by the animal. And what we found is that there were other measures out there that people were kind of using, but it turned out there was no like easy way besides trying to like code your own thing to be able to implement those measures and get an easy readout for a vast array of data. So kind of this started off as a way of us going in and trying to find these software packages that we might be able to use to analyze our data in kind of a different way, to look at like different facets of the behavior during a task. And by not being able to find anything, it kind of came out as we should probably build something. At least I have a background in computer science, so I figured, you know, it would be nice to be able to build something that would be able to help kind of the greater research pool look into originally just Morse water maze data, but it's kind of expanded past that at this point. So what it is it's a open source, mostly Python package that anyone can install. And what it does, it looks at spatial search strategy data. And as you see it here, it is set up for Morse water maze, but that's really easy to kind of configure to be able to work with other kind of paradigms. So we have people using it in humans for virtual water maze. We have eye tracking data that people are using it for, different species in Morse water maze, open field, all sorts of different tasks this has been adapted for. And kind of what we wanted to do was not try to make the most complex analysis package that goes through and tries to like define different new ways of analyzing data or anything like that. We just wanted to make something that's easy to use, easy to download and quick, so people can download a package, run their data through and just kind of get an idea of if they want to explore these type of analyses, if they're telling them something novel over their latency or distance measures, and an easy way to like be able to adapt this to multiple different behavior paradigms. So what the main thing we started looking for was a way to kind of classify the behavior of the animal in the Morse water maze a little further than just saying, okay, it got to the platform quickly and it didn't swim very far, which is kind of the traditional way. So there's a few different kind of ways of looking into that. Some of this is through measures such as like entropy, where you calculate how kind of random the behavior of the animal is in the pool with respect to a goal. And another way is to actually look at different kind of strategies that they're employing during their swim. But by taking their whole swim path and kind of analyzing what they do over time, you can classify their swims into spatial, non-spatial, and then even further down into distinct strategies that these animals are employing during their swim. So Pathfinder is able to do both of these things. Like it can analyze with entropy, it can analyze with the swim path. Some of these swim paths actually use kind of an approximate of entropy to classify them. And this way you can get like a quick look into the behavior of the animals during the task. So this is kind of on the left here, we have the traditional measures, as well as I also put entropy up there. And on the right, we have this strategy analysis over a kind of sample data set that we trained this on. So these animals are trained in the water maze 4 trials a day for 8 days. Then they have a probe trial. Then they have 3 days of reversal where the platform changes location. And then they have a probe trial after the reversal. And so these animals are overtrained pretty much. So there should be asymptotic performance by the end of this. And we can see that you get valuable information both from latency as well as distance measures to see kind of in general how are the animals doing. But we also find that you can see this like shift from non-spatial strategies, so strategies where the animal isn't employing like knowledge of the platform location, to strategies where it's known that the animal is able to kind of decide where that platform is and employ strategies that get the animal to that platform in a spatial way. So this is kind of my intro to my talk, and I'd appreciate if people had questions or wanted to know further things about the software. And we can kind of go into detail about certain aspects if they're interested. And I'd also like to thank the people who've worked on the project with me and the lab.
Thank you, Matthew. Last picture looked like it was a very recent one, or at least the last 6 months or so.
Yeah, that's too recent.
Yeah. Cool. Yeah, I'm we can start kind of chatting amongst all of us. I see there's a question. Oh, that's a question from Jim asking whether there's a GitHub link for Pathfinder.
Yes, there is. I don't know if you can see it here. The QR code will take you there. And the website right here will get you there. I can see if I can paste it in chat, if I can pull
I
up
put it in the chat for everybody. That's probably the easiest thing to do.
I can do that after. I'll do that right after my my talk. I can't pull it up right now.
Sure.
But yeah, there's the GitHub link, and then I also sent you the link to the paper as well.
Yeah,
this
I'll
is
put that in
published.
the chat too. So so just in practicalities, this interfaces with an overhead camera, or?
So we accept pretty much just raw data from almost all of the commercially available software packages, as well as Denise Kai has an open source software package for tracking animals. I kind of started working on a tracking software, and then Denise Kai released hers. So there was no real point in going further. It's open source. It works really well, and so Easy Track, you'd see it right here. Easy Track here, you can just use that to track your animals and then import it into WaterMaze, and it works seamlessly. We also have a feature here to define your own kind of package. So if your package isn't supported, you can go tell the software how your data is laid out, and it can still import the data.
Okay, so if you have a specific maze setup that's kind of not typical, then you can still put that in.
So you'd still be able to put it in. You'd probably have to do something like, if you wanted, for example, look at search strategies, you'd be able to analyze the data, but the strategies would be very weird, because the strategies are based off different types of behaviors. So you can go in here and redefine in the settings, redefine all the strategies as well. But if the more you kind of stray from a circular maze, the more you're going to have to redefine something. So like thigmotaxic behavior, where the animals are staying on the side of a maze, wouldn't be the same. And say a T maze, you'd have to redefine how that's calculated.
Sure.
But kind of as you stray from a water maze or open field type of maze, you're going to get a little more involved in terms of adapting the software.
Right. I mean, because in open space areas is kind of more, well, it's all about exploration then, right? And if you use like a T-maze, then it's a different kind of behavior that you're looking at.
Yeah.
You mentioned you could use it also for eye tracking.
Yeah. So I'm not exactly sure how the labs. People have contacted me kind of in using the software and trying to work out kind of how to do certain things with it. And one of the labs that contacted me earlier was using it for human eye tracking. Because you can analyze data with respect to infinite goal locations, they were able to set, instead of in a maze, but on a screen, different goal locations on that screen, and then analyze the behavior of the kind of pupil direction with respect to those goal locations. I'm not sure exactly what strategies they defined to do that, but they were at least able to see time between goals, as well as whatever they were able to define to analyze their data.
That's an interesting idea. I mean, sometimes you find that software you create for one thing can actually be used by a slightly different community for something else, and that's brilliant when that happens, of course.
It's great. Yeah, that's kind of where it started. With when I first presented this at a few conferences, when MCCS or SFN, people would be like, "Well, does it support this?" And I think about it, I'm like, "Well, it doesn't, but I guess you could just." I mean, it's just you put raw data in. So if you just put your data in, you kind of see how it handles it, and then you kind of adapt the strategies to fit what your needs are.
Yeah,
and that's one thing we noticed. Sorry, go ahead.
I was just gonna say, that's the great thing about putting things, online on GitHub, right? The moment you put something out there, somebody comes up with a new idea for how to use things, and then it kind of starts going from there.
And that's one of the main things with open source. I'm very happy if you just take half of Pathfinder and do your own thing with it. It's made and it's there for people to do what they need with it.
Yeah. Cool. Let me just put the link to the paper that you mentioned up here too.
Can I see chat? Oh, I can see chat right here.
Yeah.
Oh, no, I can't, even if I click it.
Yes, it seems pretty flexible what you can do with it now.
Yeah, so that was, I mean, it wasn't initially the goal, but kind of as we expanded the feature set, we kind of realized that it didn't have to just be for water maze. So we made it easier and easier for people to be able to go in, and we kind of encapsulated certain parts of it. So if you're at all proficient in even a little bit of Python, you can easily go in and just kind of change the analysis code really easily. So that even without writing your own program, you have all of the kind of setup in the GUI and all of the main features of the program, and you can still analyze data in a completely different way. And still get the output in a similar format. You can still manually categorize data if you don't want to kind of use the classification of Pathfinder. So you could, even if this cannot do anything to your data, it can still read it in, and you can go through and manually categorize it and still get a nice readout of your data that way as well.
Yeah, cool. Let me remind the audience too that, if you have any question, you can also raise your hand, and then I'm sure Siddharth can put you on to speak. And yeah, put questions in the Q&A on the chat too. Speaking of, there's a question from Brian. Any plans on classifying segments of the entire trajectory? This would be important for paths classified as random.
Yes, so for our probe trials, we do that. So what we can do for probe trials is we can segment it based on either time between so 60-second trial, we can break it in what they're doing the first 15 seconds, the next 15 seconds, or what we do for this data here is we actually cut off the classification once the animal reaches where that platform, so probe trial, just to describe, is the platform is now removed from the maze. So we see what the animal does when it swims to that platform location, but the platform's not there. But what we analyze is the strategy of the animal up to the point where it would, within a cutoff of a second or so, when it's in that platform location, and analyze that path, and then we can analyze the path after that time as well. So for example, the animal might swim directly to the platform location, realize the platform's not there, and then resort to a non-spatial or a more random strategy after the case. So Pathfinder does both. You can do it by time intervals, and you can segment based on reaching goal locations. So.
There's another question from Katie saying, is there a measure of user reliability in manually labeling this strategy?
Right, so here in the bottom boxes here are two manual classifications of this data. One is me, and one is someone who's very familiar with the data as well as coded Pathfinder. So I know all of the ways Pathfinder is going to code it as well, which is all modifiable. And another is someone who wasn't familiar at all with the data. And there's kind of two ways of looking at it. Overall, the data is very similar between the two coders, but on a trial by trial basis, there isn't a significant degree of overlap in terms of the individual strategies. So it's kind of a way of saying it might be better to do automatic classification because it is consistent on the same trial every time, where a manual classifier might not be, because there is some, you can't see all of the details, especially if you're just looking at the trace of a path, which is what this classification is on. It's not on the videos, it's on like an overview trace heat map of the path. So there's kind of two sides to it, whether or not it's better to have those like inconsistencies between raters, and if that actually tells you something, or if using these kind of fixed parameters that are going to give you consistent results, trial or time over time of analysis, is more. So as a group, we're very consistent, but on a trial by trial basis, it does differ significantly.
Did that make sense, Katie? Any follow-up questions? Or? Okay, good. All right. I don't know if anyone else has any further questions. For Matt, we have maybe like 30 seconds left for another question, if there is one. Otherwise, we will be moving on. Okay, good. Right, thank you, Matthew.
Thank you very much.
Okay, we will be moving on to next. It's not a single speaker, it's a pair of speakers actually. So that's Ankita Moss and Jim Swabel. And I think Jim will be doing the talking at first. So Jim and Ankita are from NeuralX Labs and from Sunday Health. And I believe there's also a affiliation with Emory University also too. And they are going to talk about ethical considerations for vocal biomarker research. So please go ahead.
Awesome. Thanks, guys. Super excited to be here. I'm Jim Schwabel, the VP of Data and Research of Sunday Health.
Hi, everyone, and I'm Ankita Moss. I was an Innovation Fellow this summer in the NeuralX Innovation Fellowship Program.
And we're here just to talk about the ethical implications and considerations for vocal biomarker research. A very different talk than we just saw. It's more about the ethics of work we do in vocal biomarkers. So I always like to start out with a story. About 8 years ago, I got a call from my crying mother saying that my brother had a psychotic hospitalization. As you can imagine, it was a deeply troubling experience. He was not the brother who I knew him to be. Looking back at his health record, it was obvious something was off. He went to the primary care doctor 11 times over 5 years, complaining of the same symptoms like headaches, unclear thoughts. He went to 6 different specialists, and even at the hands of a well-trained psychiatrist, he was misdiagnosed with an anxiety disorder. He spent over $15,000 in screening costs, MRI imaging, and other things to be misdiagnosed leading up to this visit. So very big problem, very common story in schizophrenia. So at Neuralex and Sond, one of the things that we asked ourselves is how can we prevent this from happening? And it looked like a lot of the early signals of my brother were through voicemails on my phone that looked for abnormalities in speech. And a lot of the leading up to his first episode, he'd use new words on voicemails on my phone. And it looked like something called word salad or abnormal use of speech and language was a common symptom leading up to his episode. And we looked further at this as could speech be used as a marker for schizophrenia onset and early stage psychosis. We got really excited when we saw this paper in NPJ Schizophrenia, which is Nature's Schizophrenia Journal, which showed that in a high risk group for psychosis, somebody like my brother could be predicted to have a psychotic break with very high accuracy in a pilot study. It is a very 2-year study, retrospective. So it's really excited looking at voice as a marker for health across many disease states. And here are some trials that were ongoing in Guillermo's lab, the lead researcher in that study, that showed that voice actually is a good marker to build machine learning models across a range of health conditions in psychiatry and neurology, ranging from Parkinson's disease to Alzheimer's with various voice tasks. And so at Neuralex and Sond, we've been using voice as a marker for health for a number of years now to collect voice samples from patients, classify diseases, streamline referrals, and create reports to help improve the process of diagnosis in psychiatry. And so in someone like my brother, you can imagine him going to the primary care doctor, getting a voice test prescribed by a primary care doctor or a provider or a specialist like a psychiatrist, do a voice sample in the web, either at home or in clinic, and provide like a samples, how's your day, and then get flagged for psychosis and get a referral using a machine learning model, and then get intervention early, like psychotherapy or low-dose antipsychotic, leading to better outcomes and ultimately lower costs to drive change in the healthcare system in a way that really helps these patients. And so we're not the only ones working in this space. It's been actually a very hot topic. You guys might have been keeping up with a lot of companies getting involved in vocal biomarkers. Facebook and Google are really driving home this year a lot of initiatives. But we really wanted to bring up in this talk, how do we keep companies like us accountable? Because there's a lot of ethical implications here, as you can imagine. And so I'm going to pass this on to Ankita to help explain some of these issues at large.
So our team proposes a voice ethics framework to assess the risks voice computing poses to end users who we see as key stakeholders in this discussion. And you can find this ethics framework in Jim's book, Introduction to Voice Computing in Python. And as you can see, risks here are stratified by the issues. The first being data collection. Data types include metadata and raw data, and the risks include privacy, for example, sending sensitive data to a third party. Another issue is access. Data can either be encrypted or open access, and risks include fraud or data loss, for example, via hacking. The next one is data publishing. Data can either be private or public, and a risk is privacy, for example, if someone publishes a voicemail without the consent of an individual. Lastly, we have data manipulation. Types include data fusion and data modeling, and risks include discrimination and emulation. For example, discrimination regarding algorithmic bias and emulation if someone uses another individual's voice to open their bank account without their consent. Now I'd like to provide an example in this voice ethics framework. Now, Google Voice is a telephone service that includes recorded messages, voicemail, and messaging. Imagine if Jim's brother left him a voicemail, and Jim decided to publish this voicemail on YouTube publicly. And at the same time, Jim's brother is on a job hunt. He's searching for a job, and a company decides to create a machine learning model to detect predictors for schizophrenia. And Jim's brother is screened out of this position. This is an issue of discrimination within this framework.
So what else do you guys think? I think this is a new emerging field. You can imagine voice data is everywhere. We are in Zoom right now. This is being recorded. You guys could all be screened for health conditions using your voice on this call. I think this opens up a window of thought. I think this is true for a lot of neuroscientific data modalities, for neuroimaging and others, but voice is particularly prolific, and we'd love to further discuss this with you all. So that's all we had prepared. Love any comments, questions, thoughts about the work we're doing here.
Great. Yeah, very relevant stuff, of course, these days, with all the focus there on a lot of these issues. So yeah, let's see if we can get discussion going. Oh, that looks like it's a question from previous talk. Yeah. Yeah, I was wondering also, I mean, so bias in machine learning is obviously a huge problem too, right? If we've seen plenty of examples where you have a machine learning being trained on only white faces, for example, and then it performs horribly on anyone with just a bit of skin color. So that's another example of certainly something that falls into some of these issues with ethics for sure.
Yeah, I think that's a huge area in this work. So if you look at a lot of the papers published in the space, there isn't a lot of diversity in recruitment. You often find a white male recruited, even in the study in schizophrenia, it wasn't really a diverse population. And I think that you get a lot of issues with that, with dialect, accent, and voice. There's a lot of implications there with aging. The power in the voice changes as you age, gender differences with frequencies. So you got to be careful to control those confounds so that you're picking up the true signal of interest. And happy to talk through that at any depth if you guys want to. It's quite an interesting field. And in automatic speech recognition, it's even more interesting when you look at Google and all the things they do. I mean, African Americans are underrepresented in speech models. You just look at them querying Alexa and other devices in that population, and it's not recognized, the accent and also the dialect. And I feel like here, this is where it's really important to build models around representative use and the population. So yeah, huge issues in algorithmic bias and speech models.
Yeah, I mean, you know, even if Alexa is not very good at recognizing your voice, that's maybe not the end of the world. But when you start discussing things like detecting different diseases, then it becomes a completely different ball game we're talking about, right? Much larger ethical issues that we run into. So yeah, again, if anyone wants to join in the discussion, please either raise your hand or put something in the Q&A. Then we can fish you out and have you join in too, for sure. Yeah, I mean, data access and data collection, of course, are issues too. That especially like those of us who in the EU, for example, we're aware of the fact that there are all these EU laws now about data access and what you're doing with the data, and then if you're doing any kind of experiments, then there's multiple pages of information that you need to give to your subjects to make them aware of what's actually happening with your data, right?
Yep.
And as annoying as that can be, certainly sometimes, I think it's hugely important nowadays knowing what happens with your data after it's being collected, for sure.
Yeah, and I think that this can open up windows for discussions, like in a terms of service, for example, when you say you're improving your services, what does that mean explicitly? Like, are you aggregating data and making models? If so, how? In what populations are you doing that? If you're using a speech model, how is it trained? I think there's a lot of transparency issues here with how we transact with business. And speaking as a CEO before all of this, I think it's not necessarily out of ill will or anything else. It's just a lot of these frameworks aren't existing that we can discuss and move it forward. And I think GDPR is a great force function for that.
Mm-hmm. So how do you see this framework being used? Sorry.
No, you go ahead.
I was just going to say, how do you see this framework being used? What's your hope for it?
I think one of the ways, and Kita can chime in too, one of the ways is I think that we can assess the risks associated with data collection, access, publishing, and manipulation. And I think that when you go deeper in how we are using data, in stratifying it by the issue, you can often see to what extent the risks are. And I think privacy risks are big risks. I mean, we brought up the case of a privacy risk that ends up leading to the data manipulation risks. And these are real risks. Like you could scrape a lot of things off YouTube, create a text-to-speech model that's synthetic in somebody's voice, and then hack a bank account. And these are like real risks that when you look at systems and especially synthetic data exist. And I think you're seeing that now with all the new synthetic media out there. But I think it's even deeper than that. Like, and I don't think users intend for you to use their information that way. And I think obviously privacy risks are big, but I think ultimately it's to stratify risk, to be proactive and not reactive. It's like anything security wise, everyone's always reactive to a breach or reactive to discrimination. I think we're trying to, with this, be proactive and kind of foresee regulation, foresee laws, frame these risks in a way that are real. You know, on YouTube, there's billions of people that say depression, my story on a blog. If you build models off of depression off of YouTube, did those users consent to that when they posted those videos? And if so, how do you get that consent if it's terms of service is a certain way? There's just a lot of pre-regulated things that you can do that is being done either by these large companies or by us. And I just think it's better to be proactive and be more active in getting user consent than it is to be assuming by default it's okay. And I think that's the status quo, as you know. And I think this is sort of, you got to put pressure for policy change to happen. And you can't really do that unless you show the risks. And Nikita, I think you can chime in a bit here too, because
yes, I was gonna speak to the policy implications for ethics frameworks and how they can inform new modern day policies, which are needed now, especially, I know we're not supposed to get political, but in the United States, there's this discourse around pre-existing conditions, and there are many ways to screen for these pre-existing conditions, as you can see in this voice ethics framework. So using something like this to inform policy to make sure everyone is treated fairly, I think is important.
Are there any issues that you see are more specific to do with voice as opposed to other types of private data? So, you know, if you're ripping voice from YouTube videos, are there any ways that that's different from taking, say, faces and visual information?
Yeah, so I think disease risks are real. So one is example, COVID-19. We're in this era of COVID. Sond has a product called Sond1 that's return to work product that screens for respiratory symptoms and asthma. If you can determine in an asymptomatic population that somebody has COVID from speech samples like phonations and things like that, then it creates all these implications about traceability and screening that not necessarily are just surveys. So I think that's super relevant today. It's hard from like a face to look at things like expiratory volume and FEV1 volume measurements. So yeah, I think like voice, there's something deeper. There's diaphragm muscles affected. There's lung capacity measurements. There's brain activity in Alzheimer's and language centers of the brain that aren't present on facial expression. So all these things really do have a deep representation for you as an individual, as an identity. And all that isn't represented necessarily in a face. And yeah, and even from voice, you can create generative faces. So there's work at MIT that shows from given input, you can actually produce a facial profile of someone based on millions of samples, voice samples, and thousands of identities off of VoxLab. So there's even implications of just from a voice file, you could reproduce and reconstruct facial features. So yeah, there's a lot of things, and those have algorithmic bias issues too, and profiling issues. And I don't think any of this is really well fleshed out from a consumer-facing public perspective yet. It's more for security purpose or surveillance. But I would really like to see more discourse to be consumer friendly. I'm a huge fan of what GDPR is doing in Europe. I think we need that in the States. We need more pressure, so to speak, as Akito was mentioning, to really drive change.
I do think it's the right way to go. Yeah, I do agree on that. Yeah. Unfortunately, we do need to stop the discourse right now about this topic and move on to the next one, I'm afraid. Thank you very much, both of you. So next up is Katie James. So Katie, if you want to get your slides ready.
Sure.
So Katie is an undergraduate at the University of New Mexico. And she will be talking about the contribution of anxiety to spatial memory deficits in the TGF344AD rat model of Alzheimer's disease. So please go ahead.
Thank you so much. So as you mentioned, my name is Katie. I'm a sophomore at the University of New Mexico, and currently we're working through this longitudinal study in this rat model of Alzheimer's disease, looking at whether or not the magnitude of anxiety is a behavior, is a later predictor of spatial performance in these rats with AD. And just a note, this is kind of a snapshot of what we're working with. We're by no means, finished. So just some background on the topic. We see in Alzheimer's disease that anxiety-like behaviors and spatial navigation impairments are early markers. For example, in this figure here, we see that the husband in this partnership is easily able to navigate himself between home and the coffee shop, and his wife doesn't seem to be able to do that as easily. And this is something that we would see in someone with Alzheimer's disease, not quite knowing where they are in their environment. And in this rat model, TGF344, we see these anxiety-like behaviors around 4 months, and then the spatial deficits later at 7 to 10 months. So still early in their lifetime, but anxiety-like behaviors a little bit earlier. And just from time and time again in humans, we see the relationship between increased anxiety, having a negative relationship on working memory capacity, and more relevant to us, spatial working memory performance. And so all of this together led us to wonder, since these rats show increased anxiety-like behaviors early on, is this a predictor of their performance in the spatial working memory task? And we think that it will be. So to analyze their spatial working memory performance, we use the match to place task. So in this task, we have a Morris-Watermaze variant where the platform is in a specific location on day 1. They have 2 trials to find that platform. We expect that they would find it quicker on trial 2. And then we move the location every day for 5 days. And we do this across 3 time points, from 4 to 5 months of age, 7 to 8 months of age, and then 10 to 11 months of age. To assess their anxiety, we use the elevated plus maze again at each time point, where time spent in the closed arm here would indicate higher anxiety, or at least what we think to be higher anxiety. Of course, the rat wouldn't be able to tell us if they're anxious or not. But, all in all, we used this simple linear regression where anxiety-like behaviors would predict spatial deficits. So looking at our results so far, we see that the rats adhere to what we would expect. Looking here at, age versus closed arm in seconds, meaning our anxiety measure, where more time spent in the closed arm is higher anxiety, we see that the TG rats are consistently spending more time in the closed arm across all time points. And then looking at their spatial performance across all time points, a different score normalized across both trials, where a negative difference score indicates finding the platform faster on trial 2. We see that nearly all of the positive difference scores are attributed to rats with Alzheimer's disease, and generally they're experiencing more spatial deficits where their scores are more positive, indicating worse performance. So this is what we would expect. We see that the TG rats show greater anxiety-like behaviors and show greater spatial deficits. However, when we plotted these together, where our anxiety measure was to predict the spatial measure, we don't see much of a relationship here. And so we conclude, based just on this, that the time spent in the closed arm was not a predictor of latency. However, we think that this might be because escape latency, as Matthew went over in his presentation, would be more of an indirect measure of the variance in the behavior. And so we want to, in our next step of the experiment or analysis, look at the direct analysis of their behavior in the form of swim trajectories. So this is a figure I made to show in part A here, the whole path of the rat broken up into different trajectories. Here in part B that we see it is executing, and then all the different kinds that we might expect. And for rats specifically with high anxiety, we might see them using paths such as thigmotaxis or chaining, where they're definitely indirectly navigating around the platform, or just kind of sticking to the walls of the platform, not really moving about, not confident in their ability to find the platform. So that's kind of the next step where we're heading, and hoping to see more of a relationship between the search strategy and the spatial performance in the rats with AD and anxiety. And so just a quick thank you to all the members in Ben Clark's lab, specifically Laura and Ryan, my direct mentors, and then the members of the Pankowski lab who helped us with the initial experiment. And I've also linked my science Twitter here. I am very new to science Twitter, looking to make connections, and obviously my email. And then a thank you to our funders. So that's all I have if there's any questions.
Great, thank you very much. Were you a bit disappointed that you didn't find the relationship you were hoping for?
Yes, obviously that's always kind of disappointing, but it's not a dead end for us, and that's something to look forward to.
Right. So can you tell me a little bit more about the TG model for Alzheimer's? So the rats, because I don't really know anything about them at all.
Sure. So so relatively new model, not as many papers on this AD model as others, but basically this is the one that we've used in our lab to analyze their performance in basic reference memory tasks. I don't know personally a ton about the difference between this model and others, as I'm fairly new to this, but this is just what we specifically work with.
Okay. No, I mean, Alzheimer's is a huge issue, and for society, if we can find ways of getting to better understand it, that would be awesome, of course. Absolutely. So I mean, I've heard ideas beforehand of this, using things like the raised maze that you're also using. But yeah, I guess you'd have to see whether using something like Matthew's software would be useful in something like the water maze instead. See if you can get better results from that.
Definitely.
There's a question, I think, in the Q&A from Jason asking, are you going to look at the brains? Maybe there are dorsal ventral differences in pathology that could explain the spatial anxiety dissociation.
I mean, definitely that's a possibility. We're kind of still in the beginning stages of this, and just kind of wanted to come here to report what we've found so far. I think it would definitely help us to look at more data, obviously, as we didn't find exactly the relationship that we were hoping for. So it's definitely a possibility, sure.
Okay. Do you mind going back to the results slide again? I just want to see it once more. Sure. That's okay.
That too, I'm not exactly sure.
Right, okay. Right. Yeah, I guess it's hard to really conclude much from that, right?
Right. The only thing that we really see when we look at this is that the TG rats really only exist in this upper range, as we saw from them having high anxiety. And we do see a good amount of rats here at the 300 seconds that just didn't move at all.
Right.
So that's something obviously to consider, that they may not have high anxiety, they just didn't move, which is always when working with the rats when they don't move.
Easy rats, yeah. So they basically just spent the entire time in the closed arm, yeah?
Yes, the total trial was 300 seconds. So that would indicate they didn't.
Okay, okay. Yeah. Okay, well, I mean, yeah. I think it might be hard to conclude very much, I guess, based on this. But maybe you'll get more of a result with the water maze instead. Yeah. Any questions from the audience? Again, if you want to raise your hand, then we can also let you ask the question live, or you can write something in the Q&A. You're welcome to do that too. So do you mind going back to the final slide, your conclusion slide? Yeah, so this one here too. Okay. Yes, it's you know, it's impressive the amount of different types of behavior that you know, behaviorists can notice in a rat, where I just see a rat that's swimming in the water.
Sure.
But yeah, I'm guessing this is where something like Matthew's software could actually be quite useful for classifying this kind of behavior.
Yeah, I had a question about that. So you have a lot of strategies here, which is something we kind of like. Obviously, I don't know if you've started doing any preliminary analyses here, but we struggled a lot with trying to dissociate what like a true strategy that they might be employing was, and like, is there a difference between something like a target search and a target scan that you have here? Like, is that a different behavior that the animal is employing versus are you just dividing strategies? And it really does make a difference when you start trying to do statistical analyses on these data. We've worked with statisticians now trying to figure out good ways of analyzing the strategy data statistically. And really some of the best advice that they had was break it into spatial and non-spatial only and just do a bivariate analysis on that. So and it's kind of just a word of caution, and kind of maybe you have some insight of being very careful and trying to dissociate like true different strategies. Like thigmotaxis, the animals do behave this way, and it's different than swimming randomly in a maze. But and chaining might be, it's like a little more central thigmotaxis. It's probably, I mean, they might know the distance of the platform from the wall, but then you start getting into like, okay, now we have like focus search versus like a focal search near the platform. Are those different behaviors? Is one slightly? So I'm just wondering if you had any input on that, and maybe how far you've gotten into this.
So we've definitely I've worked with, we have our own kind of in-house way to do this, but definitely have looked through your GitHub as well as we move into doing this more centrally. But that's kind of why I asked earlier about reliability, because, you know, when we look at, you know, as you mentioned, target search versus target scan, you know, how different really are they? And so I know that maybe if I was manually, you know, coding it, I would worry that I wouldn't treat the same path the same if I saw it twice. So I definitely, you know, appreciate your advice on that. This was just kind of based on work we had seen before with the specific rat model. So that's kind of where it came from.
Matthew also had a question about the open arms. I don't know if you want to ask that still, Matthew.
Sure, yeah, I was wondering if you tried releasing the rats for your open arm, like in the open arm of the maze, because that would kind of allow you to see if they were super anxious. I'd assume they would just move to the closed arm and then stay there, rather than if you release them in a closed arm, then they don't move, and then it's harder to dissociate whether they're just being extremely anxious and not wanting to explore at all, or whether they're just lazy and are going to lay there for 5 minutes.
Sure. This experiment was done before I even came to the university. So that's, you know, I didn't get the chance to ask at the time, but something that kind of has come up in that conversation is if the rat's not going to move, they're also not going to move in the open arm. So that would, like, the open arm and closed arm are just inverse numbers of each other, basically. So we might see the exact same thing.
Okay, We are getting close to the ending time for the whole session, unless there's a really burning question from anybody. No. All right. Good. Thank you, Katie.
Thank you.
And thank you to all the speakers for today. Very interesting stuff being discussed, of course. I'm afraid we do have to stop at this point right now. Just want to make sure to say thank you also to Siddharth Suresh, who's been sitting in the background, making sure everything is working okay. And yeah, I think this is pretty much the last session right now, except for the closing ceremony, so to speak, right after this. So I hope you have a continued good weekend, and I'm sure I'll see you around.
Thank you very much.
Thank you.
Thank you.